Very good morning to all of you. On behalf of myself and behalf of the College of Biotechnology, I welcome you all in the committee room of the College of Biotechnology. Today, we are going to have a one-day international workshop of antimicrobial resistance, the current scenario and the future prospects. So, I welcome all the DASH dignitaries, faculty, delegates, and the different participants who have joined us on the online mode in the YouTube, Facebook, and the WhatsApp. So due to the limitation of the COVID-19 restrictions, we have to have the limitation of the 50 participants in this program. With this, we have developed a module which is having both the offline and online mode. So in this way, we are having offline presentation and also the online presentation. As the program, there is always a ceremony to welcome the guests. So I request Honorable Vice Chancellor Sir to present a bouquet to the Chief Guest of the function, Dr. Parveen Malik, to welcome him. And he is Animal Husbandry Commissioner, Department of Animal Husbandry and Dairying. So I request Dean College of Biotechnology to assist VC Sir to welcome Chief Guest of the function, Dr. Parveen Malik. I once again request Vice Chancellor Sir to welcome the Managing Trustee of Dabur Ayurved, Sri Mohanji Saxena ji. Now I request PI of the project NAHEP and DES, Dr. Anil Siroy, to welcome Honorable Vice Chancellor Sir in this occasion. I request Dr. Ravinder Kumar, Dean College of Biotechnology, to please assist Dr. Now I request Dean College of Biotechnology to welcome Dr. Anush Sharma Sir, who is WHO consultant, technical point in India. Dr. Anush Sharma Sir, welcome you, sir. Now I request Dr. Pankaj Kumar, Head Department of Biochemistry and Physiology, to welcome Dr. Anup Kalra Sir, Managing Director of Dabur Ayurved India. Dr. Saraswati is a goddess of wisdom, so whenever we start any issue related with the education, we have to pray that it should get success. So I invite the chief guest of honor, Honorable Vice Chancellor Sir, and I dash dignitaries to please light the lamp with the goddess Saraswati.
करुणामयी वरदायिनी कर कमल वीणावादिनी माँ सरस्वती 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 सा तो स्वर में निवास है रे रे में धरा आकाश है सा तो स्वर में निवास है रे रे में धरा आकाश है गा गाए गुड़ी गंधर्वगण गा गाए गुड़ी गंधर्वगण गुड़ी जन की हो हितकारिणी माँ सरस्वती 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 माँ स्वर को मेरे सवार दो अब सरल का कर उतार दो माँ स्वर को मेरे सवार दो अब सरल का कर उतार दो मेरी वाणी में माँ तुम बसो मेरी वाणी में माँ तुम बसो हे शारदे हंसासिनी माँ सरस्वती 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 पवित्र पावन पावनी धवल वस्त्र सुशोभिनी पवित्र पावन पावनी धा धवल वस्त्र सुशोभिनी नीन मन कर ऋषि देव मिल नीन मन कर ऋषि देव मिल मोह लोभ निवारिणी माँ सरस्वती 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 माँ सरस्वती.
Thank you. Now I invite Dean College of Biotechnology for welcome address and about the today's workshop. Sir, please.
Good morning to all. Res- Respected Honorable Vice Chancellor of this university, Dr. R.K. Mittalji, Chief Guest of the function, Dr. Praveen Malikji, Guest of Honor, Sri Mohanji Saxena, Managing Director, Ayurved, and Dr. Anup Kalra, uh, Dr. Anu Sharma, WHO Consultant, New Delhi, India, deans and directors of this university, faculty of College of Biotechnology, and dear participants, particularly those who have come from different institutions. It is my pleasure to welcome the stalwarts of distinguished fraternity in College of Biotechnology in one-day international workshop on antimicrobial drug resistance, current scenario, and future prospects. On this occasion, I welcome our most respected Honorable Vice Chancellor, Dr. R.K. Mittalji. I also welcome Chief Guest of this workshop, Dr. Parveen Malikji, Commissioner Animal Husbandry, Department of Animal Husbandry and Dairying, Government of India, New Delhi. I also welcome Mr. Mohanji Saxena, Managing Trustee, Dabar Ayurved, and guest of honor of today's workshop. I welcome Dr. Anup Kalra, Director Corporate Affairs, Dabur Ayurved India, and Dr. Anu Sharma, WHO Consultant, New Delhi, India. I welcome Dr. Lisa Thompson, uh, uh, World Animal Health Organization Regional Representative for Asia and Pacific, Tokyo, Japan, who is joining us online directly from Japan. As we all are aware that antimicrobial resistance is a burning issue and is a priority in challenging in 21st century. And we all are also aware that it is a global health and development threats, and it requires immediate attention. And World Health Organization have also declared this antimicrobial resistance is one of the 10 uh, top 10 global public health threats. So, under this situation, when we are fighting with the COVID-19 pandemic, uh, the topic have become more important so that we can think on that and we can develop some alternative medicines and drug. With this thought, the present workshop is scheduled to bring all the stakeholders under one umbrella to discuss more on this uh, burning issues. The workshop is mainly divided in two sessions. Session 1 include the lectures of Dr. Lisa Thompson, OIE Regional Representative for Asia and Pacific, Tokyo, Japan. Dr. Anu Sharma, WHO Consultant, New Delhi, India. Dr. Anup Kalla, Director Corporate Affairs, Dabur Ayurved India, and Dr. Anu Rahal, Principal Scientist, CIRG Makhdum. In second session, we will have Professor A.P. Garg, Vice Chancellor, Soviet University, as chairman of the session, and his topic of discussion on antimicrobial drugs discovery and challenges, along with the online deliberations of Dr. Bhojala Singh, head and principal scientist, Division of Epidemiology, IVRI, Dr. Ajit Singh, scientist emeritus, Luwas, Hisar, and Dr. Mamtez Saxena, professor, animal biotechnology, Govind Ballam Pant University, Pannagar. So, the relevance of this topic is quite clear with the overwhelming response for this training. And due to the limitations and restrictions due to COVID-19, we have allowed only 50 participants in offline mode. And these include the faculty members of College of Biotechnology, College of Veterinary Science of this university, College of Agriculture, and outside representation from Soviet University, CCS University, IIMT University, and from uh, other veterinary university like Bikaner, Hisar, and Mathura, along with online participation of about 346 delegates from seven different countries, including Canada, Australia, UK, Germany, Bangladesh, Nigeria, and of course the other participants from all over this country. So, with this, I once again welcome you all, the dignitaries, faculty members, delegates, and participants in this one-day workshop on antimicrobial drug resistance. Thank you very much.
Thank you, sir. In a current scenario of antimicrobial resistance, there is always so many voices coming from the different part of world that we should go for the antimicrobial resistance by the alternative medication. And for that, we have our leading medical supporting health from the Dabur Ayurved, who have working in this field for last so many years. And they have separated out a veterinary wing also for by the name of the Ayurved. We have the managing trustee of Ayurved, Mohanji Saxena ji, with us. So we are going to make this moment memorable by signing the MOU between the university and the Dabur Ayurved. I request Dean College of Biotechnology, PI sir, and Dean College of Veterinary Sciences, Dr. Rajbir Singh sir. So please assist Honorable Vice Chancellor sir and Dr. Saxena ji sir to please sign the MOU and exchange the MOU. We are lucky enough that we have the Animal Husbandry Commissioner at this occasion, because whatsoever the proceedings will come, those to be implemented in both aspects, in a human animal health aspect and the human health aspects. So we have Dr. Anush Sharma as a consultant of focal point of WHO in India. We have ASC from the veterinary side, so we can imagine that both the side will come together and they will work in this aspect. And in coming time, we will be having some collaborative projects with the Dabur Ayurved so that we can develop certain alternative medicines in this regard. And it might be a very good, it might be very challenging, but it will be very good, inspiring moment for us, because many of us who have been entered here as a faculty of College of Veterinary Sciences, where lot many works to be done in this field. So we will have the upper hand of the Dabur Ayurved. They will, in true sense, they will lead us to the path of the development of alternative medicine in this field. So this is again a memorable moment, and I personally thank Dr. Rajbir Singh sir and Honorable Vice Chancellor sir for taking all pain for the execution of this MOU. They have taken all the efforts so that it can get approved from the academic councils and accordingly from the board of management. You might be aware about the COVID-19 pandemic, during which lot many preparations, Ayurvedic preparations were in the market. We are using them for the immunity booster purpose, and also to overcome the viral infections. So that has given an impression. That we can go ahead on this aspect. So today is going to be a landmark or a milestone in the history of the university, where we started working with we are going to start with working with the Dabur Ayurved. And as nowadays government is promoting the projects in the PPP mode, so we will have certain good projects with Dabur Ayurved, because they have all the validation and laboratory facilities to cope up with, and the scientific caliber of our university, particularly the College of Veterinary Sciences, College of Biotechnology, and College of Agriculture, can be used with them to develop certain alternative medicines in coming time. I would like to request SC sir to be the witness of this and Anush Sharma sir to be the witness of this, sir, so that we can make it memorable that all the major stakeholders are the part of this. Thank you, sir. Whenever we organize such type of scientific deliberations and the lectures, it is always mandatory to have certain concrete documentation. In this way, our team had worked on a very short notice, so we have a sort of compendium and proceedings have been prepared from this part, which have the lecture compilation and also the part of the different emerging techniques which can be used for the development of alternative medicine. So I request Dais dignitaries to please inaugurate the book, compendium book of this workshop. I request Honorable Vice Chancellor Sir, Chief Guest and Guest of Honor Sir to please release the book, and I request Dean College of Biotechnology and PI of the project, NAIP ICR, Dr. Anil Suroi Sir, to please assist our Dais dignitaries to please release the compendium of the workshop. Thank you, sir. And in this series, now we have with us Sri Mohanji Saxena ji, who is Managing Trustee of Dabur Ayurved. He is guest of honor of today's inaugural function. So I request Sri Mohanji Saxena ji to bless us with his wordings. I request you, sir. सर वहीं से कर लीजिए, माइक इधर शिफ्ट कर देंगे. हाँ, इधर आ जाए, आगे बढ़ा लीजिए सर, आगे कर लीजिए सर थोड़ा सा. आ जाइए सर. हाँ, सिखाई देंगे. थैंक यू सर.
सर्वप्रथम मैं आभार प्रदर्शित करना चाहूँगा वाइस चांसलर और डीन सर का कि आज एक नई पहल शुरू हुई है जिसके अंतर्गत ये जो विषय है AMR का, उसके ऊपर चर्चा के लिए आज ये दिन चुना गया है. जैसा कि आप सब जानते हैं और मैं इस धारणा के साथ कि आप सब वैज्ञानिक हैं और विज्ञान में विश्वास रखते हैं, ना कि व्हाट्सएप के ज्ञान पे ध्यान देते हैं. बहुत सारी भ्रांतियां हैं, बहुत सारे विषय हैं, बहुत सारे गंभीर विषय हैं. और डॉक्टर मलिक से जब इस विषय में चर्चा आज से 3 साल पहले, कोविड के पहले हुई थी कि हमें इसके बारे में कुछ करना होगा और हम केवल वेस्टर्न वर्ल्ड के ऊपर निर्भर नहीं कर सकते कि वो आके हमें बताएं कि AMR का समाधान क्या होगा. जहाँ तक इस विषय का महत्व है, आप सब जानते हैं कि विश्व को अगर खतरा न्यूक्लियर बॉम्ब से नहीं है, विश्व को अगर खतरा है तो वो रेजिस्टेंट माइक्रोबियल बॉम्ब जो है, और इसको हम रोज देख भी रहे हैं. देख क्या रहे हैं कि सर्जरी कामयाब थी, पेशेंट वार्ड में आ गया था, लेकिन बाद में लंग्स में इन्फेक्शन हो गया. डॉक्टरों ने 14 दिन रखा, 10-20 लाख रुपए का खर्चा आया. कोई भी एंटीबायोटिक ऐसा नहीं था जिसका प्रयोग नहीं किया और अंत में पेशेंट को बचा नहीं पाए. ये आज एक सामान्य विषय हो गया है और इसलिए जब इस ओर शुरुआत की तो ये चीज हुई कि क्या है ये विषय, कहाँ से ये है, कहाँ पे इसको हमें एड्रेस करना पड़ेगा। तब एक चीज ये निकली कि ना कि ये केवल वेटनरी का विषय नहीं है, ये केवल एग्रीकल्चर। तो बहुत सारे लोगों को आश्चर्य हुआ कि एग्रीकल्चर का कैसे है। तो स्ट्रेप्टोपेनिसिलिन बिकता है, फलों में इस्तेमाल होता है और उसका रेजिड्यू हमारे शरीर में पहुंचता है। और फिर बात में आ गई कि अगर हमारी सॉइल में एंटी माइक्रोबियल बैक्टीरियाज हैं जो रेजिस्टेंट हैं, तो उनकी पूरी सप्लाई चेन चलती है, उनकी पूरी साइकिल चलती है। मुझे हर्ष है कि ये एमओयू साइन हुआ है, जिसके अंतर्गत एक लॉन्ग टर्म पर्सपेक्टिव में इसका समाधान निकालने में, जिसमें सबकी जरूरत है। जरूरत किसकी है कि वैज्ञानिकों ने तथ्यों को किसानों के साथ साझा करना है, उन्हें ये बताना है कि क्या सही है क्या गलत है। डॉक्टर साहब को हर जानकारी है, डीन साहब को, जितने लोग यहाँ बैठे हैं सभी को होगी कि कई बार ऐसा होता है कि हमें बाएं हाथ को नहीं पता होता है दाहिने हाथ में क्या हो रहा है। तो उसी तरीके से एग्रीकल्चरल वालों ने स्ट्रेप्टोपेनिसिलिन एग्रीकल्चर के लिए अप्रूव किया, फरीदाबाद की लेबोरेटरी ने। बोले हमें नहीं मालूम था कि ये तो ड्रग होती है और ये तो हमें एनिमल हस्बेंड्री या ड्रग डिपार्टमेंट के पास जाना चाहिए। और तब ये चीज आई कि जो नेशनल एंटी माइक्रोबियल प्लान 2017 में बना, 5 साल का, 2022 में उसका समय पूरा हो जाएगा, वो एक अलग बात है। लेकिन आज जनमानस भी इस चीज के लिए सचेत है, हमारे संस्थान जितने हैं वो भी सचेत हैं, और जितने भी औद्योगिक इंडस्ट्रीज हैं वो भी इस बात के लिए सचेत हैं कि हमें मिल के काम करने की जरूरत है। और प्रधानमंत्री ने कई बार अपने मन की बात में इस बात का जिक्र किया कि भारत देश की संपदा है ज्ञान, ज्ञान आधारित विज्ञान, और वो विज्ञान जो आज केवल देश में ही नहीं विदेशों में भी जनमानस के काम में आ रहा है। आप सब जानते हैं कि है कहने को वही चीज काढ़ा, लेकिन कोविड के काल में इस काढ़े ने कितनों की जान बचाई, इसका कहीं पे डॉक्यूमेंटेशन नहीं है, लेकिन साक्ष्य है कि लोग आज भी विश्वास करते हैं कि अगर इसका समुचित प्रयोग किया जाता है तो निश्चित रूप से फायदा होगा। आज एंटी माइक्रोबियल ही नहीं, एंटीवायरल ड्रग्स को ढूंढने के लिए भी इस ओर अनुसंधान हो रहे हैं। माइक्रोबायोमिक्स, हर माइक्रोबायोलॉजिस्ट के लिए माइक्रोबायोलॉजी का एक्सटेंशन माइक्रोबायोडाटा एंड माइक्रोबायोमिक्स में हुआ है। और फ्यूचर हेल्थ केयर इज गोइंग टू बी डिपेंडेंट ऑन आवर हेल्दी माइक्रोबायोडाटा। तो जिस तरीके से जन्म पत्री बनती थी, आज जीनोम पत्री बन रही है, संस्थान बना के दे रहे हैं जीनोम पत्री, जिसके द्वारा आप अपने डीएनए के म्यूटेशन को वॉच कर सकते हैं एंड फोरकास्टिंग कैन बी डन, हाउ गुड योर लिवर विल बी, हाउ गुड योर पेनक्रियाज वुड बी, हाउ गुड योर सेलुलर मेटाबॉलिज्म वुड बी। आज डीएनए आरएनए मैसेंजर आरएनए के ऊपर रिसर्च हो रही है, और मेरा ऐसा मानना है और मेरा विश्वास भी है कि हम सब लोग, खास तौर से जो युवा वैज्ञानिक वर्ग है, इस दिशा में बहुत कुछ कर सकते हैं। संस्थान का भी नाम ऊपर होगा, देश का भी नाम ऊपर होगा, और विश्व को जो प्रधानमंत्री ने कहा, ये हम एज ए फार्मेसिस्ट बहुत समय से कहते आए हैं कि वी आर द फार्मेसी ऑफ द वर्ल्ड, वी कैन प्रोवाइड द हेल्थ, नॉट अबाउट द डिजीज ट्रीटमेंट। अंतर सिर्फ यह है, हेल्थ इज ऑन दिस साइड, डिजीजेज आर ऑन दैट साइड। डिजीजेज गेट अटेंशन, not the health. But now probably the time has come to make forward on understanding what is health under one health program, where soil, environment, crop, animals, and human health all is taken into account while developing the solutions for the problem we have been facing. Once again, I thank VC sir, Dean sir, and Dr. Anuj Sharma, everyone, because सबकी आहुति की जरूरत है इस अभियान में कि AMR का हम क्या सॉल्यूशन निकालते हैं। धन्यवाद सर।
धन्यवाद सर। मैं कहना चाहूँगा कि बहुत ही सुंदर और सरल शब्दों में सर ने एंटीमाइक्रोबियल ड्रग रेजिस्टेंस, उसकी महत्ता को हमारे सामने रखा। और सर, जो आपने बीड़ा उठा रखा है इस प्रकार की औषधियाँ तैयार करने का, जिनके माध्यम से हम न केवल मानव का बल्कि पशुओं का भी कल्याण कर पा रहे हैं, तो हम चाहते हैं कि आप बड़े भाई की भूमिका में रहें और कुलपति जी की और एससी साहब के नेतृत्व में हमारा कॉलेज ऑफ बायोटेक्नोलॉजी और कॉलेज ऑफ वेटरनरी साइंस दोनों मिलकर के इस क्षेत्र में आगे कल्याण करें, जिससे पशुओं के हित में भी और मानव के हित में भी हम इस प्रकार की औषधियाँ ला सकें जो आने वाले समय में एक लैंडमार्क साबित हो। तो मैं विशेष धन्यवाद करता हूं सर आपका, क्योंकि आपने समय निकाला और बहुत शॉर्ट नोटिस पे हमारे शेड्यूल चेंज होने के बावजूद भी आप हमारे मध्य उपस्थित हुए, और आपके अग्रज की भूमिका हमें सर हमेशा स्वीकार रहेगी, आपका नेतृत्व स्वीकार रहेगा। धन्यवाद सर। नाउ द टाइम हैज कम टू इनवाइट द चीफ गेस्ट ऑफ द फंक्शन, डॉक्टर प्रवीण मलिक सर, फॉर हिज इनॉग्रल एड्रेस। He has the vast experience as a veterinary microbiologist. Now he is the leading veterinary practitioner, and he is the front leader for the veterinary professionals. So we are very delighted to have Dr. Malik. Sir, please.
दिखाई तो मैं दूंगा नहीं इसके साथ। Thank you very much, Dr. Amit. Honorable Vice Chancellor of the University, Dr. Mittal sir. मोहनजी सक्सेना जी, जो मैनेजिंग ट्रस्टी हैं डाबर आयुर्वेद के, डॉक्टर अनुज शर्मा, WHO कंसल्टेंट, डॉक्टर अनूप कालरा, डॉक्टर अनिल सिरोही, डॉक्टर रविंद्र कुमार, फैकल्टी फ्रॉम द यूनिवर्सिटी, डिफरेंट कॉलेजेस, स्टूडेंट्स and participants, ladies and gentlemen. ये जो टॉपिक आप लोगों ने रखा और इस टॉपिक के मौके पे जो आप लोगों ने एक MOU करने की बात कही है ARF और यूनिवर्सिटी के बीच में, बहुत ही एप्ट टाइम और सिस्टम, और जिस टाइमलाइन से हम इस टाइम गुजर रहे हैं, कोविड-19 का एक पेंडेमिक, बीच में हमें लगा था कि इंडिया ने इसे शायद क्योर कर लिया है, काफ़ी कुछ नज़दीक आ गए हैं, आज एक सेकेंड वेव की तरह वापस से केसेस बढ़ रहे हैं। इस तरह के पेंडेमिक के सिस्टम से खाली अकेले पेंडेमिक को ना देखें और हम लोग ये महसूस करें कि अल्टीमेटली ह्यूमन हेल्थ, एनिमल हेल्थ, एनवायरमेंटल हेल्थ किस तरीके से जुड़ी हुई है, हाउ दे आर इंटरलिंक्ड। अगर हम हिस्टोरिकल व्यूज लेके देखें तो जो हेल्थ में इंप्रूवमेंट आए हैं उसमें हमारी लिविंग स्टैंडर्ड्स की बात भी है जो हमने शायद इंप्रूव किए हैं, सैनिटेशन और हाइजीन की भी बात की है हम लोगों ने, काफी इंप्रूवमेंट हुए हैं, जिसमें क्लीन वाटर भी है, हमारे रहने सहने के तरीके भी हैं। और अगर हम देखें तो पिछले 4 साल से जो ये हम स्वच्छता अभियान की बात कर रहे हैं, जिसमें none other than the Prime Minister himself was pursuing how to clean your hands. मतलब मुझे लगता है कि आज अगर हम 21वीं शताब्दी में आके लोगों से कह रहे हैं कि आप अपने हाथ धो लीजिए, आज ये जो मास्क हम यूज कर रहे हैं ये सिर्फ कोविड के लिए नहीं है। मेरे बीच में चर्चाएं चल रही थी कि क्या कोविड का इसका क्या हमें जिंदगी भर पहनना पड़ेगा। मैंने कहा अच्छी चीज है, पहन लीजिए, शायद 2 या 3 साल के बाद आपको पता चलेगा कि टीबी के केसेस कितने कम हो गए, कितने अस्थमा के केसेस कम हो गए, किस तरह के बहुत सारे आपके बैक्टीरियल इंक्लूडिंग जो आपके जिसे कॉमन कोल्ड आप कहते हैं, कितना ट्रांसमिशन कम हो गया। So it's not about COVID-19 only. It's about like how you are improving your health standards globally. और इन सारी चीजों के साथ साथ जब एंटीबायोटिक्स और एंटीमाइक्रोबियल्स का इन्वेंशन हुआ, जिस तरीके से वो डिस्कवरी हुई, और हमें लगा कि अब इन्फेक्शियस डिजीज से और खासतौर से बैक्टीरियल और फंगल डिजीज से अब हमें कोई चिंता नहीं है, क्योंकि हमारे पास सॉल्यूशन मिल गया है। लेकिन शायद हमने उस समय ये हम भूल गए कि जो अलेक्जेंडर फ्लेमिंग ने पेनिसिलिन के डिस्कवरी के साथ साथ ये कहा था कि यू हैव टू बी केयरफुल इन यूजिंग दिस, यू आर गोइंग टू हैव रेजिस्टेंस टू दिस पर्टिकुलर ड्रग ऑल्सो, एंड दिस रेजिस्टेंस हैज बीन देयर। शायद लेट 1920s में इसका डिस्कवरी हुआ और मार्केट तक आते आते 1940s के अर्ली तक हमें करीब करीब 10 साल लगे थे पहले एंटीबायोटिक को इस्तेमाल करते हुए। और जब तक हमने इस्तेमाल शुरू किया था तब तक इसके रेसिस्टेंस रिपोर्ट हो चुके थे। आज की तारीख में अगर इसी सिस्टम को हम देखें तो हमारे पास मैकेनिज्म है कि एंटीबायोटिक्स जब तक नहीं आती हैं, हाल में तो आ नहीं रही हैं, अगली पाइपलाइन में नहीं है कोई 10 साल 20 साल, और जो आ रही हैं उसका रेसिस्टेंस या तो आने से पहले है या आने के 6 महीने के अंदर अंदर रिपोर्ट हो जाता है क्या ये रेसिस्टेंस? हमें ये समझने की जरूरत है, क्या ये एंटीबायोटिक के साथ चल रहा है? मुझे लगता है कि एंटीबायोटिक के साथ रेसिस्टेंस चल जरूर रहा है, लेकिन अगर हम इसे समझें कि रेसिस्टेंस है क्या एक्चुअली, ये एक स्ट्रेटेजी है ऑर्गेनिज्म की सर्वाइवल के लिए। और इस स्ट्रेटेजी में एक सिस्टम आता है कि जहां भी उसके ऊपर प्रेशर पड़ेगा फॉर दिस पर्टिकुलर ऑर्गेनिज्म हैज टू चेंज टू सर्वाइव, और उसमें यूसेज एक बहुत इंपॉर्टेंट फैक्टर है। और इसका जो यूसेज में जो सुबह हम बात कर रहे थे मोहन जी सक्सेना साहब से, पहले हम मिसयूज की बात करते हैं, फिर अब्यूज की बात करते हैं, फिर हम देखते हैं कि किस तरीके से वो इस्तेमाल हो रहे हैं, whether they are being used in a proper way, the way we define that the antimicrobials have to be used, and then how the resistance are developing. हमने कहा एंटीबायोटिक्स हमने डिस्कवर की 1940s में 30s में, क्या एंटीबायोटिक्स इससे पहले नहीं थी? आर वी यूजिंग दोज? एक रिपोर्ट में बीच में पढ़ रहा था कि फॉसिल्स के अंदर से कुछ माइक्रो ऑर्गेनिजम्स लोगों ने आइसोलेट किए हजारों साल पहले, और उसके अंदर ड्रग रेजिस्टेंट जीन्स आइडेंटिफाई किए गए। तो ये कोई नया फिनोमिना नहीं है, ना ये एंटीबायोटिक्स के साथ आया इसे, लेकिन हमने आइडेंटिफाई किया। और एक जो प्रोसेस था इसका नॉर्मल इवोल्यूशन का, जो नॉर्मल सिस्टम ऑफ डेवलपमेंट था, उसे हमने एग्रेवेट कर दिया, हमने बहुत फास्टन कर दिया, इतना स्पीड से हमने इसे बढ़ा दिया कि हमें दिखाई देने लगा कि अब अगले 30 साल के अंदर क्या कोई एंटीबायोटिक हमारे लिए यूजेबल होगी। बहुत बड़ी चिंता का विषय है, और मैं लोगों को वार्न करते हुए कहता हूं, थोड़ा ड्रामेटिक है कि शायद हम अपनी लाइफ पूरी करके चले जाएं, अगर कोई डिजीज आती है तो शायद कोई एंटीबायोटिक हमें मिल जाए वेयर वी कैन ट्रीट आवरसेल्व्स, प्रोबेबली, बट जो हमारी नेक्स्ट जनरेशन है वो अपनी पूरी लाइफ एंटीबायोटिक्स के भरोसे तो नहीं कर सकता। इसके यूज में चाहे हम ट्रीटमेंट में करें, दो वर्ड हम कहते हैं मेटाप्रोफाइलेक्सिस और मेटाफाइलेक्सिस और प्रोफाइलेक्सिस, चाहे उस तरीके से करें या ग्रोथ प्रमोशन में करें, हमें समझना होगा कि वेदर दीज आर बीइंग यूज्ड एप्टली in proper manner, in a thoughtful manner, so that the antibiotics we are using in any of these circumstances, क्या ये ठीक है? और ये जो एक अनस्टॉपेबल फिनोमिना है, हमारे पास सिर्फ एक अल्टरनेटिव है कि वी हैव टू बेसिकली रिड्यूस द स्पीड। लीजिए सर। सो ये इमरजेंस में हमारा रोल कहां से शुरू होता है और हम कहां क्या क्या इसमें कर सकते हैं? सबसे बड़ा रीजन क्या है? सबसे बड़ा रीजन है इसमें कि हमने पॉपुलेशन इतनी बढ़ा दी है, एनवायरमेंटल डिग्रेडेशन हमने कर दिया है, और जो प्रेशर सिस्टम का आया है, जो वाइल्ड लाइफ के साथ हमारा एक सिस्टम ओवरलैपिंग का आया है, एंड वी कॉल इट वाइल्ड डोमेस्टिक कॉन्फ्लिक्ट, वाइल्ड लाइफ ह्यूमन कॉन्फ्लिक्ट, वो हमने इतने ज्यादा बढ़ा दिए हैं कि शायद जिस तरह के जो ऑर्गेनिजम्स थे, अभी मोहनजी सक्सेना साहब ने कहा कि फ्रेंडली माइक्रोब्स, एक बहुत पहले मैंने 10-12 साल पहले एक छोटा सा आर्टिकल रीडर डाइजेस्ट में पढ़ा कि हमारे पास कितने माइक्रोब्स हैं वर्ल्ड में। हर ह्यूमन के अंदर हम ऑन एन एवरेज बोलते हैं कि हर नंबर ऑफ सेल्स इन बॉडी कितनी ट्रिलियन सेल्स हैं और उसके 10 गुना मान के चलें। पता कैसे चलता है कि ये हैं कितनी? इसमें से कुछ कल्चरेबल हैं, कुछ नॉन कल्चरेबल हैं। जो कल्चरेबल हैं उनको शायद हमने पहचाना है, जो नॉन कल्चरेबल हैं अभी तक हम सारे पहचान भी नहीं सके हैं। लेकिन लोगों ने अंदाजा कैसे लगाया कि जिस टाइम ह्यूमन जीनोम का प्रोजेक्ट चल रहा था, हम लोग एक्सपेक्ट कर रहे थे कि अगर हम ग्रासहोपर के जीन्स को देखें, अगर हम माइस के जीन्स को देखें, तो उसके हिसाब से हमारे पास कम से कम 1 लाख के आसपास जीन्स लोगों ने एक्सपेक्ट किए थे कि हमारे जीनोम में निकल के आएंगे। जब जीनोम प्रोजेक्ट पूरा होने को आया तो एक टेक्निक कहती है 25,000, एक टेक्निक कहती है 33,000, तीसरी टेक्निक कहती है 27 से 30,000। तो बचे हुए फंक्शंस जो हमारे 70,000 फंक्शन बच गए, वो इज परफॉर्मिंग दोज फंक्शंस। सो इट इज एक्सपेक्टेड दैट ये जो हमारा जीनोम हमारे पास अवेलेबल है थ्रू दीज माइक्रोब्स, दे आर हेल्पिंग अस, बट थ्रू द यूज एंड अब्यूज ऑफ एंटीबायोटिक्स वी आर जस्ट किलिंग देम एंड हैविंग अ कंपटीशन। सो ये एक हमने मैकेनिज्म बना दिया कि शायद वो ऑर्गेनिज्म जो हमारे फ्रेंडली थे उन्हें भी सर्वाइवल मैकेनिज्म चाहिए था। हम सिर्फ पैथोजेंस को अभी तक टारगेट कर रहे हैं, वी आर नॉट टारगेटिंग द फ्रेंडली माइक्रोब्स। हमने ये नहीं सोचा कि हम उनके अंदर भी रेसिस्टेंस जीन्स को इनकॉर्पोरेट कर रहे हैं, दीज कैन बी ट्रांसमिटेड बैक, क्योंकि ये जीन्स चाहे प्लाज्मिड में हो चाहे क्रोमोजोमल में सिस्टम से हो, दे आर बीइंग ट्रांसमिटेड फ्रॉम वन प्लेयर टू अनदर वन। सो ये जो हमारा सिलेक्शन का प्रोसेस है, दिस वी हैव टू बी कंसिडरिंग व्हेनेवर वी आर टॉकिंग अबाउट। और ये प्रॉब्लम सिर्फ इंडियन नहीं है, इट्स अ ग्लोबल प्रॉब्लम। अनफॉर्चुनेटली हमारे जैसे डेवलपिंग कंट्रीज में it is generally considered that we are not that kind of hygienic country, that clean country कि हम बिना एंटीबायोटिक के काम चला लेंगे, but it is a myth I feel. Probably we can do so. हमें अपनी लाइफस्टाइल को थोड़ा बदलना पड़ेगा और ये देखना पड़ेगा कि whether we actually require it. और जो हमारी सबसे बड़ी बात है कि एंटीबायोटिक्स हमारे लिए वरदान थे, हम इन्हें वरदान ही रहने दें, श्राप ना बनने दें। क्योंकि अब हम एंटीबायोटिक्स की बात करते हैं, अगर हम किसी एमआर के फोरम में आके खड़े हो जाते हैं तो बोलते हैं एंटीबायोटिक्स बड़ी खराब चीज है। नहीं साहब, बहुत अच्छी चीज है, लेकिन इसका इस्तेमाल तभी हो और ऐसे ही हो जैसे इसकी रिक्वायरमेंट हो। शौकिया खाने की चीज नहीं है। वायरल फीवर है पता है, वायरल कॉमन कोल्ड है पता है, लेकिन हमने एंटीबायोटिक साथ लेते हैं क्या पता सेकेंडरी कॉम्प्लिकेशंस हो जाएं। सो दीज सेकेंडरी कॉम्प्लिकेशंस क्या पता को शायद हम रोक सकते हैं, और वो बिना एंटीबायोटिक्स के भी रोक सकते हैं। हमारे पास दुनिया भर के लेजिस्लेशंस हैं। हमारे पास इंडिया की अगर मैं सिनेरियो की बात करें तो हमने जो हमारे एक्ट में लिखा हुआ था उससे भी हमने इम्प्रूव करके बाद में शेड्यूल H1 अमेंड किए। लेकिन आज भी ओवर द काउंटर बहुत सेल है एंटीबायोटिक की, और ये एंटीबायोटिक्स की जो OTC है, जिसे हम ओवर द काउंटर सेल कहते हैं, ये सबसे ज्यादा नुकसान की चीज है, चाहे वो ह्यूमन साइड हो या एनिमल साइड हो। हमारे पास कोई रिकॉर्ड नहीं है कि कितनी एंटीबायोटिक्स किस सेक्टर में यूज हो रही है। और जब हम इस तरह की बात करते हैं MR की, तो जनरली हमारे पास दो सेक्टर समझ में आते हैं। पहला सेक्टर एनिमल सेक्टर, उसमें भी हम पोल्ट्री की तरफ खासतौर से उंगली उठाते हैं। फिर बची कुची बात हम एनिमल सेक्टर की तरफ ह्यूमन सेक्टर कर देते हैं। डाटा दोनों के पास नहीं है, और इसमें हम बिल्कुल भूल जाते हैं कि जो एनवायरनमेंट का सेक्टर है, जो वाइल्ड लाइफ का सेक्टर है, जो एग्रीकल्चर का सेक्टर है, और अभी मोहन जी साहब ने कहा एग्रीकल्चर में जिस तरीके से एंटीबायोटिक यूज हुई पेस्टिसाइड की तरह, बिकॉज इनका जो अप्रूवल है वो पेस्टिसाइड की तरह है, और पेस्टिसाइड में मार्जिन ऑफ सेफ्टी सबसे ज्यादा एंटीबायोटिक्स में है, सबसे सेफ पेस्टिसाइड्स में से है, सबसे सस्ती है, और जिस तरीके से वो इफेक्टिव हैं हमारे हॉर्टिकल्चरल क्रॉप्स के ऊपर, उसका यूज भी बहुत ज्यादा है। हमने कभी उन्हें अपने साथ इस AMR वन हेल्थ में लेके नहीं किया। तो हमें चाहिए कि हमें इस सिनेरियो को पूरा एक साथ एक सिस्टम में समझें, अपने पूरे रेगुलेशंस को समझ लें, और उस रेगुलेशंस में हमें किस तरीके से उसे इम्प्लीमेंट कराना है। बिकॉज हम लोग थ्योरेटिकल बात शायद बहुत कर चुके हैं। वी हैव बीन टॉकिंग ऑन AMR, द कॉजेज एंड द वेज फॉरवर्ड, बट एक्चुअली इम्प्लीमेंटेशन लाने के लिए हमें क्या जरूरत है कि हम इस AMR का प्रोसेस स्लो कर दें, हम एंटीबायोटिक्स को यूज की तरह रहने दें। ग्लोबल एक्शन प्लान आज से नहीं 2015 से बन गया। हमारी पॉलिसी उससे भी पहले 4 साल पहले 2011 में बन गई थी। एक्शन प्लान हमने 17 से 22 का फिर 17 में बना लिया। हेल्थ पॉलिसी बन गई, सब जगह इस चीज का जिक्र है, लेकिन अल्टीमेटली ग्राउंड लेवल पे रियलिटीज में इसकी इम्प्लीमेंटेशन की जरूरत है, और वो जरूरत में शुरुआत शायद हम लोगों को खुद से करनी है। जो मोहन जी साहब ने कहा कि यहाँ जितने लोग बैठे हैं सब साइंटिस्ट हैं, सब विज्ञान को जानते हैं, इन सबको पता है कि AMR कैसे हैंडल किया जा सकता है। सच बात यह है कि हम उसको जानकारी को इस कमरे तक रखते हैं, कमरे के बाहर निकल के जैसे ही मुझे जुकाम होगा सबसे पहले एंटीबायोटिक मैं खाऊंगा। हमें चाहिए हमारे माइंड सेट की चेंज, और ये चेंज की जो साइकोलॉजिकल बिहेवियर की हमारी रिक्वायरमेंट है। जितने प्रैक्टिशनर्स हैं, चाहे वो ह्यूमन साइड में हो, एनिमल साइड में हो, एग्रीकल्चरल साइड में हो, हमें चाहिए कि सबसे पहले हम इस बिहेवियर चेंज के लिए एक मैकेनिज्म लेके आए, और बिहेवियर चेंज के लिए जो मैकेनिज्म है OI ने इसे कंसीडर किया ऑफिशियली। इसके लिए बात कर रहे हैं कि कैसे किया जा सकता है। रिपीटेड चीजें बार बार लगातार, आपको मैं एक बार बताऊंगा शायद नहीं समझ में आएगी, एग्जाम्पल्स के साथ, मॉडल्स के साथ आप बनाइए। अगर आपको लगता है कि मेरे पास कोई एक मॉडल मैं यूनिवर्सिटी से एक गांव में एक सिस्टम में क्रिएट कर सकूं, मैं यहां एंटीबायोटिक्स यूज नहीं करता हूं, क्योंकि एंटीमाइक्रोबियल यूसेज का सबसे बड़ा एक फैक्टर हम लेके चलते हैं कि शायद रेसिस्टेंस के लिए ये सबसे बड़ा फैक्टर है। ऑल्दो ऐसी बहुत सारे हमारे पास एक जो सिस्टम है, आपको बताएं कि ICR ने FAO के साथ मिलके एनिमल साइंसेज और फिशरीज साइंसेज की कम से कम 20 लेबोरेटरीज को एक नेटवर्क किया है। इन्फार इसे बोलते हैं, इंडियन नेटवर्क फॉर फिशरीज एंड एनिमल साइंसेज AMR रिसर्च। इस इन्फार में हमें ऐसी फाइंडिंग्स मिली हैं कि जो एंटीबायोटिक्स एब्सोल्युटली यूज ही नहीं हो रही हैं किसी एक सेक्टर में। फॉर एग्जांपल फिशरीज में अगर फिश से या फिश के प्रोडक्ट से हमने किसी एरियाज में सैंपल उठाए हैं तो वहां ऑर्गेनिजम्स हमें रेसिस्टेंट मिले हैं। इसका मतलब खाली डायरेक्टली उस एनिमल पे यूज की जरूरत नहीं है, हमारे पास सिस्टम में, एनवायरनमेंट में कहीं ना कहीं उस तरह का मैकेनिज्म ऑलरेडी मौजूद है, और हमें ये देखना है कि वो मौजूद और मैकेनिज्म शिफ्ट ना हो हमारे इस तरह के नए सिस्टम में। हमारे पास जो मेजर मैकेनिज्म है, पहली चीज हम जब कहते हैं बिहेवियर चेंज, तो जो हमारे नेशनल एक्शन प्लान में लिखा हुआ है 6 पैरामीटर्स पे हमने बात की है। एक चीज है हमारी अवेयरनेस और अंडरस्टैंडिंग। ये जो मास कैंपेन करने की बात है, बिहेवियर चेंज करने की बात है, बार बार हमें माइंडसेट बदलने की बात है। पता सबको है कि करना क्या है, लेकिन वो करवाना है उनसे, उसके लिए जरूरत है कि अवेयरनेस। और अवेयरनेस सिर्फ बता के नहीं आती है, अवेयरनेस शायद दिखा के आती है। हमें मॉडल जनरेट करने पड़ेंगे। शायद हम यूनिवर्सिटी में एक पोल्ट्री फार्म बना दें, या हम किसी गांव में किसी को अडॉप्ट करके कहें कि हम आपकी प्रोडक्शन कम नहीं होने देंगे, उसकी जिम्मेदारी हम लेते हैं और हम आपको ये मॉडल तैयार करते हैं, इसमें आप एंटीबायोटिक्स बिल्कुल यूज मत करें। शायद वो कॉस्ट अगर उसकी घटेगी, और अगर हम उसकी बायोसिक्योरिटी मैकेनिज्म इंप्रूव करा के हम उसे इक्वल प्रोडक्शन पे ला सकें, तो शायद धीरे धीरे लोगों को समझ में आए कि ये चीजें हो सकती हैं, एंड वी कैन डू दैट। नॉलेज और एविडेंस जनरेट करने हैं, चाहे लेबोरेटरी सर्विलेंस के मैकेनिज्म से हो, एएमआर के अंदर और सारे सेक्टर्स में। और इस एएमआर के साथ साथ हमें नॉलेज जो जनरेट करनी है वो एएमयू की जरूरत है। एएमआर पे अब धीरे धीरे, पहले छोटे मोटे पेपर्स होते थे, और अगर हम इकट्ठा करें, प्लस अब जो सिस्टमेटिक मैकेनिज्म बना है, डॉक्टर अनुज शर्मा शायद बताएंगे कि जो ह्यूमन साइड में भी सर्विलेंस है इंसार का, इसी तरीके से इंफार का एनिमल और फिशरीज सेक्टर में है। हम इस तरह के मैकेनिज्म से हम डाटा जनरेट कर रहे हैं रेसिस्टेंस का, बट हमें चाहिए साथ में डाटा एएमयू का, द यूसेज डाटा, सो दैट वी कैन एक्चुअली कोरिलेट हाउ दिस इज अफेक्टिंग। देन प्रिवेंशन और कंट्रोल की हम बात करते हैं। हम कहते हैं कि वैक्सीन आ जाएं, यस डेफिनेटली वी नीड गुड क्वालिटी वैक्सीन इन दैट, बट उसके अलावा हम सिर्फ जब प्रिवेंशन और कंट्रोल की बात करते हैं, हम सिर्फ वैक्सीन की बात करते हैं। हमने उसमें बायोसिक्योरिटी मैनेजमेंटल प्रोसेसेस को बिल्कुल बाहर छोड़ दिया है। उस चीज को हमें बताना होगा कि शायद वैक्सीन से भी ज्यादा, जो आज हम कोविड में बात कर रहे हैं कि वैक्सीन लगाएं ना लगाएं, लेकिन मास्क और हाथ धोना और हमारा बिहेवियर कि हम मुंह को टच ना करें अपने हाथ से, शायद ये 3 चीजें वैक्सीन से भी ज्यादा इफेक्टिव होंगी। एंड पीपल आर डूइंग दैट। जो लोग कर रहे हैं वहां बच रहे हैं, और जहां हम लोगों ने छोड़ दिया, हमें बताना पड़ेगा कि मास्क लगाना कैसे है। मास्क लगाते हैं, सबको पता है कि डिजीज कहां से ट्रेवल कर रही है, कि रेस्पिरेटरी रूट से। सबके नाक खुले हुए हैं, यहां देख लीजिए, गिन लीजिए, 2-4 तो मिल ही जाएंगे, ये तो साइंस के लोग हैं। नाक सबके खुले हुए हैं, और सबको पता है कि डॉक्टर सबसे पहले सैंपल कहां से उठाएगा कोविड का, नाक से, पर नाक हमारे बंद नहीं है। तो मास्क हमारा सिर्फ दिखाने के लिए, और फैशन वाले मास्क आ गए हैं, फैशन कर रखे हैं, अच्छा लगेगा, मैचिंग बनाएं, लेकिन साथ में ये नॉलेज और ये अवेयरनेस हमें चाहिए कि मास्क अगर हमारी एक बायोसिक्योरिटी का एक मैकेनिज्म है, तो क्या मैं इसे फार्म पे, क्या मैं इसे प्रोडक्शन सिस्टम में अप्लाई करा सकता हूं। हमने बर्ड फ्लू के केसेस में देखा, बहुत बड़ी जानकारी हमारे पास आती है कि सब माइग्रेटरी बर्ड से आता है। बिल्कुल ठीक बात है, माइग्रेटरी बर्ड से ट्रांसमिशन होता है, लेकिन मुझे समझ में नहीं आया कि माइग्रेटरी बर्ड्स कितने कितने फार्म में अंदर घुस गई, और घुसी कैसे। और एक में घुस गई तो कोई बात नहीं, बस एक एरिया के सारे फार्म में अचानक घुस गई, इसका मतलब कहीं ना कहीं हमारी बायोसिक्योरिटी में चूक है। एक डिजीज हो सकती है, हम उसे वहीं के वहीं कर्व कर सकते हैं, बट कर्विंग दैट पर्टिकुलर इंसिडेंस इज वेरी वेरी इंपॉर्टेंट, और उसके लिए जरूरत है नेक्स्ट चीज, हमारे पास डायग्नोस्टिक्स अच्छे होने चाहिए, जो रैपिड डायग्नोस्टिक्स हो, पॉइंट ऑफ केयर डायग्नोस्टिक्स हो। तो हमारी डिटेक्शन, तो हम जब बात करते हैं कि सबसे पहले अगर हम डाटा जनरेट कर सके, नॉलेज जनरेट कर सके, तो वी कैन प्रिडिक्ट बेस्ड ऑन डाटा, वी कैन देन प्रिवेंट इट यूजिंग सम ऑफ दोज मैकेनिज्म्स, वी कैन डिटेक्ट इट अर्ली, और अगर नहीं तब भी कंट्रोल आता तो वी हैव टू रेस्पॉन्ड इट क्विकली, और उसमें इंक्लूडिंग एंटीबायोटिक्स, दीज आर द थिंग्स। हम इसके यूज को ऑप्टिमाइज करने की बात करें, जो रेगुलेशंस हमारे पास हैं वो बहुत आसान भी है, लेकिन बड़ा मुश्किल भी हो रहा है, क्योंकि हम उन रेगुलेशंस को मान नहीं रहे। चाहे शेड्यूल H है, शेड्यूल H1 है, ओवर द काउंटर आज भी हम खुद भी एंटीबायोटिक्स खरीदते हैं, और उसे हम बिना रेजीम पूरा किए, बिना डोसेज को देखे हुए आज भी हम यूज कर रहे हैं। सो वी हैव टू ऑप्टिमाइज द यूज। R&D की रिक्वायरमेंट है, और R&D में जब हम बात करते हैं सबसे इंपॉर्टेंट R&D का मैकेनिज्म हमारा क्या है कि हम कहते हैं एंटीबायोटिक्स ढूंढ लें। नई एंटीबायोटिक्स प्रोबेबली आसान नहीं है। सो उस R&D में हमारे पास अल्टरनेटिव्स हैं, जिसमें हम बैक्टीरियोफाजीज की भी बात करते हैं, और साथ में हम बात कर रहे हैं अभी जो हमने इसमें बात की, हमारे पास वेटनरी आयुर्वेदा का, अगर हम अपने फील्ड की बात करें लाइफ स्टॉक की, तो वेटनरी आयुर्वेदा का एक कंसेप्ट जो है वो हम शुरुआत कर सकते हैं। मिनिस्ट्री में डिपार्टमेंट ऑफ एनिमल हस्बेंड्री एंड डेयरिंग इज इन टू टॉक विद मिनिस्ट्री ऑफ आयुष टू कम इन टू एन एग्रीमेंट, और अगर वो MOU के साथ हम लोग शुरू करते हैं, बिकॉज वी हैव ऑलरेडी स्टार्टेड आर एक्टिविटीज इन NDDB, वी हैव स्टार्टेड आर एक्टिविटीज इन हरियाणा के हर गवर्नमेंट में वेटनेरियंस को ट्रेन किया गया है उस कंसेप्ट पे, and then we are talking about generating this kind of knowledge on Ayush mechanisms also systematically in the system. एजुकेशन में लेके आने की बात है। तो क्या हम इस समय कोई अगर MSV में नहीं है, वेटनरी एजुकेशन के मिनिमम रेगुलेशंस में नहीं है, तो क्या हम यूनिवर्सिटी से कोई ऑप्शनल कोर्स शुरू कर सकते हैं, एक इलेक्टिव कोर्स डालें? ये बात पक्की है कि हमारे पास मैकेनिज्म हैं। हम आज भी फील्ड में आयुर्वेदिक ड्रग्स लिख रहे हैं, यूज कर रहे हैं। क्या हमें उसका सिस्टमेटिक नॉलेज जनरेट करा के स्टूडेंट तक पास ऑन नहीं करनी चाहिए? तो अगर मैं अपने BVSC के इतने कोर्सेज में एक 2 क्रेडिट का या 3 क्रेडिट का एक कोर्स एक्सपर्ट्स के साथ, सबसे बड़ी मुश्किल हमारी क्या होती है कि हमने कोर्स शुरू किया और उसे हम कह देते हैं कि डॉक्टर अमित ऐसा करो, ये कोर्स आप ले लो। अब डॉक्टर अमित ने तो कभी आयुर्वेद पढ़ी नहीं है, तो अगर आयुर्वेद पढ़नी है तो मुझे लगता है आयुर्वेद के एक्सपर्ट्स चाहिए हमें, और उस एक्सपर्ट्स में साइंस आप अपने वैज्ञानिकों से कराएं, और जो आयुर्वेदा की इंफॉर्मेशन है, जो बेसिक नॉलेज है, वो वहां के एक्सपर्ट से लें। इस केस में VC साहब, अगर आप लोग इस तरह का कुछ इनिशिएटिव लेने की बात करते हैं तो मिनिस्ट्री ऑफ आयुष से सपोर्ट हम दिलवाएंगे। उसमें हम कोशिश करेंगे कि वहां से एजुकेशन में हैंड होल्डिंग शुरू में हमारी मिनिस्ट्री ऑफ आयुष से हो जाए, ताकि जो नॉलेज हम इम्पार्ट करना चाहते हैं वो प्रॉपर हो, और वो हम उसे ITK की तरह यूज ना करें। हिंदुस्तान में आयुर्वेद के नाम से बहुत ITK लाइफ स्टॉक में यूज हो रही है। जरूरत है उनकी वैलिडेशन की, जरूरत है उस तरह के इनिशिएटिव्स की कि वो R&D से खाली वैलिडेट ना हो, बल्कि उस सिस्टम की तरह, एक नए सिस्टम की तरह, एक प्रॉपर ड्रग की तरह। और इसमें सबसे इंपॉर्टेंट फैक्टर फिर से आ जाता है, क्योंकि वेटनरी में बाकी चीजों के लिए क्वालिटी टेस्टिंग है, आयुर्वेद में, इस आयुर्वेद में क्वालिटी टेस्टिंग की बहुत लिमिटेशंस हैं। हमें समझना पड़ेगा कि जब हम ड्रग्स की बात करें, जब हम इन चीजों की बात करें, तो ये जो क्वालिटी हम इसे मेजर करने की बात करते हैं, वी हैव टू हैव somebody who is holding our hands to understand and to set up those mechanisms. मुश्किल नहीं है, केमिस्ट्री की जरूरत है। तो जो हमारे डिपार्टमेंट ऑफ केमिस्ट्री में यहाँ लोग काम कर रहे होंगे, HPLC से, चाहे GC से, MS से, वो लोग इसे निकाल सकते हैं कि whether it has got those components which are required in this medicine, आयुर्वेदिक में भी। तो वो जो R&D का सिस्टमेटिक कंपोनेंट है, इट इज रिक्वायर्ड। और लास्ट में मुझे जो लगता है कि हमारी जो लीडरशिप है, अगर हम वर्ल्ड को बात करें तो हमें कहते हैं साउथ एशिया में आप लीडर बन जाइए। अगर मैं कहता हूं कि इंडिया में है, तो मुझे लगता है कि जहां ये काम शुरुआत हो जाए वहां से हम लीडर बन सकते हैं। और अगर मैं वेस्टर्न यूपी में एक वेटनरी यूनिवर्सिटी या एग्रीकल्चरल यूनिवर्सिटी या वेटनरी कॉलेज या बायोटेक्नोलॉजी कॉलेज के अंदर खड़ा हूं, तो मुझे लगता है इससे अच्छा मौका नहीं होगा कि ये लीडर बनने, लोगों को दिखाएं what we can do, how we can change the scenario in AMR in this region। हम ज़्यादा लंबी चौड़ी बात ही ना करें, एक छोटा सा एरिया पकड़ लें, एक ब्लॉक पकड़ लें, एक डिस्ट्रिक्ट पकड़ लें, और शायद हम अगर इसमें कुछ कर सकें, so we can show that this is possible and this then can be percolated down to all other places। और ये जो मॉडल बना के एक हमारे उसकी सिनेरियो होगा, शायद हम कुछ बदल सकें। और लास्ट में मैं ये जरूर कहना चाहूंगा कि इट्स नॉट माय रेस्पॉन्सिबिलिटी, इट्स नॉट द रेस्पॉन्सिबिलिटी ऑफ डॉक्टर मित्तल अलोन, इट्स आवर शेयर्ड रेस्पॉन्सिबिलिटी, स्टार्टिंग फ्रॉम द टॉप पॉलिसी मेकर्स to everybody whoever is working in this field, in the last stakeholder, we call it last mile stakeholder। so इनके लिए हमें जो गवर्नेंस में ज़रूरत है, लेजिस्लेशन में ज़रूरत है, इंफॉर्मेशन जनरेशन में ज़रूरत है, कैपेसिटी बिल्डिंग की इसमें ज़रूरत है, ट्रेनिंग की ज़रूरत है, हैंड होल्डिंग की ज़रूरत है, रिस्क असेसमेंट की ज़रूरत है, या कॉपरेशन की ज़रूरत है। ये सब चीज़ें बैठ के एक प्रॉपर, अगर हम एक डॉक्यूमेंट क्रिएट करें, यूनिवर्सिटी में हम क्या कर सकते हैं, शायद ये डॉक्यूमेंट को हम एक्स्ट्रापोलेट करके नेशनल पॉलिसी की बात कर सकते हैं। तो वो सारे मैकेनिज्म हम यहाँ से शुरू कर सकते हैं, और अगर मैं अपनी यूनिवर्सिटी से शुरू कर सकूं तो शायद ये जो मेरा मैकेनिज्म है ये AMR में हम लोग हेल्प कर सकेंगे, एंड इट विल बी रियली यूजफुल. So with these words, I would like to thank the organizers, Dr. Mittal and his team, for inviting me here and giving me this opportunity to talk to you and basically convey my sentiments to you. Thank you very much.
थैंक यू सर. सर आपने जितनी बातें कही उनमें से कुछ बातों पे हमने पहले से कुछ काम करना शुरू किया हुआ है. 2-3 चीज़ें मैं आपसे रिक्वेस्ट करना चाहूँगा इस फोरम के माध्यम से. पहला है इन्फार का. तो जब पहली मीटिंग इन्फार के लिए हुई थी जिसमें 17-22 का प्लान बना था, आई वॉज देयर एज ए असिस्टेंट प्रोफेसर फ्रॉम मथुरा. So we requested that we have in 25 universities, we have the TVCC complex, and where we have the veterinary laboratory diagnostics which is working. So those should be incorporated in this INFAR project, so that the data of the state agriculture universities from the 25 states, at least that should come in the part of the INFAR. That time fund limitation was there, so only the ICR institutes were involved in this process. So I hope that 2022-27 5-year plan will include these veterinary complexes and our veterinary laboratory diagnostic labs in that project, so that we have the comprehensive data. As far as the second concept is there, sir, we have invited Dr. Dinesh Katos, sir, advisor to Ayush Ministry for this program, and the next session he is going to join us as on online mode, because he was having another assignment on 18th. Otherwise on 16th he would have been here with us. So we are already taking some steps toward this approach, that we are having a WHO representative, OI representative, Ministry of Animal Husbandry, then the Ayush Ministry. And as far as the facilities are concerned, sir, in short time we are going to have a lab here in which we will having the ICPMS, LCMS, GCMS. All those facilities have been are in the process. So what you have suggested that we should take the lead. So we are already have taken certain steps on that path. What we need the leadership like you and Honorable Vice Chancellor sir and the Saxena sir. That's why we have signed this MOU, so that we will in short time we will be having certain positive responses from our side. With these words, it is a ceremonial always to facilitate the Dais dignitaries. So I request the Honorable Vice Chancellor sir to present a memento to the chief guest of the function and to felicitate him for his presence. Dean sir and Dean Veterinary Dr. Rajbir Singh sir, please. Now I request Honorable Vice Chancellor sir to please felicitate Mohanji Saxena sir, the guest of honor of today's function. Now I request PI of the project, Dr. Srohi sir, to felicitate Honorable Vice Chancellor sir for gracing us this occasion and sparing this much of time for us. Thank you, sir. Now the time has come to have the motivational words of in the presidential address of Honorable Vice Chancellor sir, in which leadership we are organizing this program. I invite Mittal sir to please address the gathering.
Honorable this Animal Husbandry Commissioner, Dr. Praveen Malik Sahab, Shri Mohan Ji Saxena, Managing Trustee Dabur Ayurved, as guest of honor, Dr. Anu Sharma, WHO consultant from New Delhi, Dr. Anup Kalra, Director Dabur Ayurved, my colleagues, Director Research, Dr. Sirohi, Director Extension, Dr. Sachan, Dean Director NS Rana, Dr. Ravindra Kumar, Dr. Rajbir Singh, faculty members of this university, distinguished participants from other institutions in this workshop, dear students, ये आज बहुत ही शुभ अवसर है कि हम इस मॉर्निंग में इस सभागार में यहाँ उपस्थित हुए हैं। इसके कई रीज़न हैं। पहली बात तो यह है कि ये नाहप जो नेशनल एग्रीकल्चर हायर एजुकेशन प्रोजेक्ट वर्ल्ड बैंक फंडेड स्कीम है, ICR के द्वारा इसको संचालित किया जा रहा है, उसके अंतर्गत इस हॉल का रिनोवेशन किया गया है और ये फर्स्ट मीटिंग आई थिंक उसके बाद में डीन बायोटेक्नोलॉजी के आयोजन में यहाँ आयोजित की गई है। तो इसके लिए मैं डीन बायोटेक्नोलॉजी डॉक्टर रविंद्र कुमार उनकी फैकल्टी को बहुत बहुत बधाई देता हूँ। दूसरी बात यह है कि आज इस अवसर पर एक बहुत ही इम्पॉर्टेंट टॉपिक जो कि एंटी माइक्रोबियल ड्रग रेजिस्टेंस है, उस पर इस वर्कशॉप का, एक दिन की वर्कशॉप का आयोजन किया गया है। और उससे भी अधिक खुशी की बात यह है कि इस कंट्री के लीडर एनिमल हस्बेंड्री कमिश्नर डॉक्टर प्रवीण मलिक हमारे बीच उपस्थित हैं और साथ साथ उनकी उपस्थिति के साथ हमें सौभाग्य मिला कि हम मोहन जी सक्सेना जी के साथ में इस प्लेटफॉर्म को शेयर कर सके। और उस अवसर पर हमने जो डाबर आयुर्वेद के साथ में अभी हाल में MOU फाइनल किया था, उसको आज हम साक्षात रूप दे सके। ये बहुत ही खुशी का अवसर है कि हमने अपने इंस्टीट्यूशंस, अपने एकेडमिक इंस्टीट्यूशंस, रिसर्च इंस्टीट्यूशंस जैसे कि IVRI, NDRI, CICR, NIH बागपत और भी बहुत सारे जो इंस्टीट्यूशंस हैं जो ऑलरेडी हम MOU कर चुके थे, उसके साथ साथ हमने अदर इंस्टीट्यूशंस के साथ भी, जैसे अभी हाल में हमने किया था दयाल फर्टिलाइजर्स ग्रुप के साथ में, और आज ये खुशी का मौका है कि हम डाबर आयुर्वेद के साथ भी हमने MOU साइन किया है। और इसमें कोई संदेह नहीं है, क्योंकि मैं आपको बताना चाहूँगा कि कोई भी MOU जब आता है तो बिफोर साइनिंग इट मैं उसकी डिटेल्स पहले पूरी देख लेता हूँ कि क्या क्या हमारी एक्टिविटीज़ हैं और कहाँ कहाँ हम कोलैबोरेट कर सकते हैं। तो आई वॉज हाईली इम्प्रेस्ड with the activities of Dabur Ayurved, and I think our faculty will benefit with their experiences and their facilities, and same is the case vice versa also. तो इसकी हम उम्मीद करते हैं और इसका जो हमारा आज ये कोलैबोरेशन हमने साइन किया है, इसको आगे लेके चलते हैं। जहाँ तक एंटी माइक्रोबियल रेजिस्टेंस की बात आती है, तो मेरे से पहले मोहन जी सक्सेना ने और डॉक्टर प्रवीण मलिक ने इसके बारे में इतना कुछ बता दिया है कि मैं महसूस करता हूँ कि अब क्या गज़ल सुनाऊँ तुझे देखने के बाद। इन लोगों ने उसको सबको कवर कर दिया है फ्रॉम A to Z, और मेरे ख्याल से जो बाकी चीज़ हैं वो आप आने वाले जो लेक्चर्स हैं उसमें सब कवर करेंगे। आई एम नॉट ए मैन ऑफ veterinary or animal sciences, but I know of course that this is a very serious issue. It's a global issue, and I think the main issue of the 21st century. And if I am not wrong, the World Health Organization has kept it in top 10 issues, major challenges. And this is very much required for addressing the sustainable goals also. जैसे कि बताया कि एंटीबायोटिक्स का जो रोल है इसमें बहुत ज़्यादा पहले होता रहा, और डॉक्टर मलिक साहब ने और सक्सेना साहब ने बताया कि पहले, और वास्तव में है कि आज भी हम देखते हैं कि कोई भी बीमारी होती है, किसी को छींक आ जाती है, किसी को ज़रा सिर में दर्द हो जाता है, हल्का सा फीवरिश होता है, तो सबसे पहला ध्यान जाता है कि क्रोसिन के साथ में आप एंटीबायोटिक शुरू कर दीजिए, कहीं ये डिवास्टेटिंग ना हो जाए। लेकिन उसके साइड इफ़ेक्ट्स बहुत ज़्यादा हैं, और मैं देख रहा था कि पहले जैसे यूरिनरी ट्रैक्ट इन्फेक्शन, जस्ट फॉर एन एग्जांपल, उसमें सिप्रोफ्लॉक्सासिन बहुत यूज़फुल होती थी, एंड नॉर्मली वी यूज्ड टू गेट रिजल्ट इन 5 days, बट नाउ मेरे ख्याल से, जो इफ आई एम नॉट रॉन्ग, इसकी जो इफिकेसी है वो वर्ल्ड ओवर 4.9 to 92% इसमें रेजिस्टेंस डेवलप हो चुका from different countries. So why is that? So we have to analyze the reasons कि कैसे हम जो antimicrobial resistance है, same is the case with this microbes, I mean virus also, we are passing through this COVID pandemic. We at a time we felt that we are out of that, but again the second phase is coming, and in many parts it is going to be, it is expected to be काफ़ी सीरियस उसके आने के चांसेस हैं। और जैसे मलिक साहब ने बताया कि मैं खुद भी उस टाइम कल्पित था, मैंने भी नाक के नीचे अपना मास्क लगाया हुआ था, बट that was occasional, because so we have to be aware about those things. We have to take care of our hygiene, we have to take care of our sanitation, and I mean these are the contributing factors which contribute to the antimicrobial resistance also, directly or indirectly. I know in 1984, I'm basically by training plant pathologist, so that time I was in active research. तो इंडोनेशिया ने 1984 to 86 वहाँ पर डिक्री की थी कि जो सिंथेटिक पेस्टिसाइड्स हैं उनका यूज बंद कर दिया था उन्होंने, एंड दैट वाज द रीज़न कि उससे प्लांट्स में रेजिस्टेंस आ रहा था उन डिजीज़ के अगेंस्ट. सो देयर वाज अ टाइम, आई कैन सी अबाउट 35 years ago, they declared it, and that's why in India also, later on, this National Center for Integrated Pest Management, with a broader mandate to cut down the use of the chemicals, was created. तो वही चीज़ें जब हम देखते हैं कि एनिमल साइंस और फिशरीज में भी आ रही हैं, जैसे कि डॉक्टर मलिक ने बताया कि लगभग 20 इंस्टीट्यूट्स में ये जो प्रोजेक्ट है इसको लॉन्च किया गया है बाय ICR, दैट दे हैव बीन इंक्लूडेड एज पार्ट ऑफ दैट. सो आई थिंक देयर इज a need for greater awareness and taking a multi-sectoral approach to address this issue. It's not a issue जो आज डेवलप हो रहा है. We know that during last about 2 decades, only 5 antibiotics or so they have come out, and probably in the next decade we don't foresee any development. But of course, for this COVID virus, the country and the world has shown a promise that they could develop this vaccine. So there are opportunities, we have to encash them, and of course we have to be prepared for that, develop our infrastructure, facilities, open-mindedness, then database management. All these things are necessary and they should be coordinated in association with private and public sector. That's very, very important. So I think enough is said about antimicrobial resistance, and we look forward to having the recommendations of this workshop, and I think they will be printed later on and should be circulated to all state and central department for their comments and input, and which should again be incorporated in the proceedings. And we should I mean, like stand up to fight this microbial resistance and prepare an action plan, because we get so many recommendations in a workshop or in a this symposium, but the implementable recommendations are very much required, and along with that, the action plan that how we are going to address those things. Of course, there are many agencies for funding our programs if it is soundly prepared to be implemented in a phased manner. So that's my advice to all the organizers. I wish them all the success for conduct of this workshop and look forward to having the fruitful recommendations. And once again, I thank Dr. Praveen Malik, Mohanji Saxena, then Anu Sharma ji, Anup Kalra ji, and I know that in the list of people joining through email, we have Dr. Thompson, then Dr. Anu Rahal, then Dr. Abhijit Mitra also listed there. So I think all the participants will benefit from their participation and having good advice or guidance from all of you. Thank you very much.
Thank you, sir. We have already started working on these MOU aspect. In the last 10th of March, one of batch of our students have already visited the training facility and the R&D facilities of the Dabur Ayurved. So they have already started supporting us. And in future, we also hope that it will continue. So in this continuation, I would like to invite my colleague, Dr. Jiten Singh, who is the co-organizing secretary of this workshop, to please deliver his vote of thanks. Accept.
Thank you, Dr. Amit. I thankfully acknowledge and thank Honorable Vice Chancellor, sir, under whose guidance this program is being conducted, the ages of ICR and NAP institutional grant. I thank you, sir, for your constant support and guidance and giving us time to chair this inaugural function of one-day workshop. I'm thankful to the chief guest of today's function, Dr. Parveen Malik, Animal Husbandry Commissioner, Government of India, for giving us time and coming to this university to inaugurate one-day workshop. I am thankful to the guest of honor, Mr. Mohanji Saxena ji, Managing Trustee, Dabur Ayurved, Dr. Anuj Sharma, WHO Consultant, and Dr. Anup Kalra, Director Corporate Affairs. I thank Director Research and Project Investigator, Dr. Anil Siroi sir, and I thank Dean College of Biotechnology, Dr. R. Kumar, who has been guiding force in bringing the college to new heights. I thank Dr. S. K. Sajan sir, Director Extension, Dr. N. S. Rana sir, Dean Agriculture, Dr. Rajiv Singh sir, Dean College of Veterinary and Animal Husbandry. I thank all the head of the departments present here, faculty members, staff of the college, and students. I thank the organizers and all who are directly or indirectly involved to organize this function. Thank you.
Thank you, sir. And with this, I also thank all those who have joined us on the online mode on the Facebook, Twitter, and this. With this, we will go for the national anthem, and then Haiti is outside the syllabus. So I request all the participants to please stand up at their seat for the national anthem. Yes, sir. अधिनायक जय हे भारत भाग्य विधाता, पंजाब सिंध गुजरात मराठा द्राविण उत्कल बंगा, हिंद हिमाचल न गंगा उच्छल जलधि तरंगा, तब शुभ नामे जागे तब शुभ आशीष मांगे,
गावे तब जय गाथा। जनगण मंगलदायक जय हे भारत भाग्य विधाता, जय हे जय हे जय हे जय 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 हे।
Thank you, sir. The inaugural tea is outside in the library hall. And as we are running short of time and the session is to be started by 11:30, so I request some of the participants to be here with us so that we can start the session, because Dr. Lisa Thompson is going to join us from the Tokyo directly. So sir, thank you, sir. A very good morning to all of you. We are a little bit late. I personally apologize for the delay in the program, and especially to Dr. Lisa Thompson, who is waiting from the Tokyo, Japan, to deliver her talk on this workshop. So I request in this session, we have with us Animal Husbandry Commissioner, Dr. Praveen Malik, as a chairman of the session. We are going to have Dr. Anu Sharma in this session, who is a WHO consultant, who will be the co-chair of the session, and along with Dr. Anu Sharma, Dr. Anup Kalra, the managing director of Dabur Ayurved, he is also going to co-chair this session. The two reporters will assist to prepare the report of this session. Dr. Harshit Verma is here, and another Dr. Akshay Garg. And this session is going to have one outside presentation from Dr. Lisa Thompson. So with this, I request Dr. Malik to please chair the session. Sir.
Thank you, Dr. Amit. And this session, इसका कोई नाम दिया है क्या? Just two. Okay. So where is this? So thank you very much, Dr. Amit, for this one. So in this session, we have got four speakers. Dr. Lisa Thompson from OIE Regional Representation for Asian Pacific, Japan. Dr. Anu Sharma, WHO Consultant, Dr. Anup Kalra, and Dr. Anu Rahal. So we'll be touching the topics related to the antimicrobial use, the global perspective from the human side, from the WHO side, the policies on that. Dr. Anup Kalra will be talking on AMR mitigation, the role of herbals, it means the alternatives. And Dr. Anu Rahal will be talking about ethnopharmacological interventions. So we are already delayed in that. So may I now, without wasting time, Dr. Kalra, would you like to say something before we start? Okay. So without waiting, may I invite Dr. Lisa Thompson to come and start her deliberations on OIE antimicrobial use in animals data collection. So the OIE perspective on data collection. Hi Lisa, how are you doing?
I'm very well. How are you?
Fine, thank you.
Nice to see you.
Over to you. Please, you can come up on that now.
Okay, thank you very much. Let me just share my screen here. Okay, can you see that okay?
Yeah, it's very clear.
Okay, wonderful. Thank you so much for the invitation to speak at the workshop today. It's my pleasure to be here to represent the OIE. So I am a regional project officer and I'm working with One Health issues. So that obviously includes antimicrobial resistance. Today I'm going to really focus on antimicrobial use in animals because that's really something that's quite important for the OIE. For those of you in the audience who may not know too much about the OIE, just as a little bit of background, the OIE, which was originally the Office International des SVPUAT, was created in 1924. This was in response to rinderpest causing massive problems. It became known as the World Organization for Animal Health in 2003, but we still retain the OIE as our main name. So the OIE is an intergovernmental organization. Our mandate is to improve animal health, welfare, and veterinary public health. And the OIE sets international standards that are recognized by the World Trade Organization. You'll see from the schematic there that there are 182 members, and that's including 325 reference centers and 75 partner organizations. For each member country, we have one national delegate to the OIE and 8 national focal points on specific subject matters. And that includes one covering veterinary products, including antimicrobial resistance. Headquarters is still in Paris in France, and we have 13 regional and sub-regional representations. So obviously, to cover that number of members, we're quite spread out around the world. You'll see that number 32 there is for the Asia and the Pacific region. And these are the other regional and sub-regional representations. We are under the direct authority of the Director General of the OIE. In this region, we have the regional office in Tokyo, and the sub-regional office for Southeast Asia is based in Bangkok. So for the OIE, what we want to do is protect animals to preserve our future. But we do realise that everything is really very integrated. So we know that human health, animal health, and environmental health are all interconnected. So we're not really just focusing on animals, but we realise that there is a great deal of interconnectivity. And that really helps us when we try to understand how to use the One Health approach. So One Health is when we use collaborative efforts to address diseases that pose potential threats to both animals and humans. And when I say diseases, I actually don't just mean infectious diseases. We also include things like antimicrobial resistance in that, because it's something that's a risk to both animals and people. And when we talk about One Health, we're really talking about using a multi-sectoral approach to tackle these issues. And that includes a number of different things. So we need to have strategic planning and emergency preparedness. We need to consider surveillance for zoonotic diseases and obviously sharing information that we find out from that surveillance. We need to have a coordinated investigation and response. Joint risk assessment is important for those threats. And then to reduce the risk, we need risk reduction, risk communication, and very importantly, community engagement. And all of these obviously aren't possible if we don't include workforce development. So the One Health concept is really a global strategy for managing risks at this animal-human ecosystems interface. And the Food and Agriculture Organization, FAO, the OIE, and the World Health Organization, WHO, has a tripartite agreement. So the 3 directors general have an official agreement to work together. And the 3 priorities identified at this moment in time are zoonotic influenzas and rabies, and you'll see in the middle there, antimicrobial resistance. So recognizing that this is an issue that really needs to be addressed using One Health. So back to the OIE and what specifically have we been doing for AMR. In May 2015, at the annual general session, Resolution 26 was on combating AMR and promoting the prudent use of antimicrobial agents in animals. So the OIE develops a procedure and standards for data quality for collecting data annually from OIE member countries on the use of antimicrobial agents in food-producing animals, with the aim of creating an OIE global database. So this is really when it started for the OIE back in 2015. In 2016, at the next general session, Resolution 36 was on combating AMR through a One Health approach, actions and OIE strategies. So really figuring out what we needed to do to combat AMR. And the OIE actions to be compiled and consolidated within the OIE strategy on antimicrobial resistance include the establishment and the management of a database for the collection of data on the use of antimicrobial agents in animals, as well as the development of interpretation indicators. So I'm going to talk a little bit more in detail later on about this database for antimicrobial use in animals. So obviously a lot of work, a lot of guidelines and plans have been produced for antimicrobial resistance. And these documents are really to protect animal health and welfare and also human health, and supporting global efforts to combat antimicrobial resistance. So looking first at the Global Action Plan on Antimicrobial Resistance. So this was developed by the World Health Organization and published in 2015. And the goals here were to improve awareness and understanding of antimicrobial resistance through effective communication, education, and training, to strengthen the knowledge and evidence base through surveillance and research, to reduce the incidence of infection through effective sanitation, hygiene, and infection prevention measures, to optimize the use of antimicrobial medicines in human and animal health, and to develop the economic case for sustainable investment that takes account of the needs of all countries, and increase investment in new medicines, diagnostic tools, vaccines, and other interventions. So I think already in your opening comments, there's been some talk of the need to have alternatives for antimicrobials, and that's something that obviously needs investment. Then the OIE strategy on antimicrobial resistance and the prudent use of antimicrobials was published in 2016. And this is obviously in agreement with the WHO global strategy. But the specific aim of the OIE strategy is protecting animal health and welfare by supporting global efforts to combat AMR. And just looking at the areas that focuses on, the four main objectives are improving AMR awareness and understanding, strengthening knowledge through surveillance and research, supporting good governance and capacity building, and encouraging implementation of international standards. And from this, it's obviously very important that we think about creating awareness and communicating that with a number of different stakeholders. And we need to consider carefully what's appropriate messaging for different stakeholders. So the OIE has developed these 5 only rules. So number 1, only use antimicrobials when prescribed by veterinarians. Number 2, only when needed. So antimicrobials do not cure every infection. 3, only obtain antimicrobials from authorized sources and retailers. 4, only use the dosage and follow length of treatment and withdrawal periods as prescribed. And number 5, only use antimicrobials when associated with good animal health care. So following these 5 rules, then hopefully we can improve antimicrobial use. And obviously we think about how we can display these messages. And so we have a variety of social media tools that can be used for various campaigns. Obviously, posters and leaflets can be used. These can be found on the Trello board at the address here. If anyone's interested, I can share this presentation afterwards if you want to follow up on this. But importantly for our region, where we have very varied countries and different people here, it's very important that we think about local languages. So we have lots of these materials that are also available in different languages, and these are all accessible via the regional OIE website. So we've got for Bhutan, Cambodia, Indonesia, Malaysia, Mongolia, Myanmar, Nepal, Sri Lanka, Thailand, and Vietnam. So this is something that we're very happy to help support, to have these kind of materials available to everyone where they might be needed. And then also we have some videos and infographics and try to follow up with social media. I think this is becoming even more important now, where we have got less access to physical meetings and face-to-face events, but we need to think how we can spread the message and communicate the messages for antimicrobial resistance to many people. On the global OIE website, there is a portal for AMR. So you can click, and that has access to lots of information. Just to let you know that the new OIE global website will be launching very soon. So there will be a little bit of change in Outlook for the images here. Hopefully it will be a lot more user friendly, but please do check that out. And then on the regional website, we also have a dedicated page for AMR. And we use that mainly to highlight materials from events that we've conducted. So for example, there have been 13 in-country workshops on monitoring the quantities and usage patterns of antimicrobial agents in animals. There have been 9 seminars on containment of antimicrobials, particularly looking at veterinary education establishments and veterinary statutory bodies, also livestock and aquaculture industries. Also, we have links to things like the database on antimicrobial agent use in animals. We assist reviews of implementation of action plans for antimicrobial resistance. So we try to have materials there that are very relevant for the region and to share materials with our members. A couple of other things, obviously, last year in World Antimicrobial Awareness Week, which runs from the 18th to 24th of November, we were restricted to virtual events. So we had these 2 webinars on responsible and prudent antimicrobial use. And we decided to focus on aquatic animals and companion animals because very little attention has been paid to those, but we're obviously very aware that we need to include all animals within the OIE mandate for looking at antimicrobial use. And also supporting in-country activities. So again, we translated some more of those materials I showed you earlier, and we were able to have a regional tripartite webinar. So within the region, we do some joint activities with our tripartite members. So we have the FAO and WHO combining with OIE to have events like this webinar. So back to this global data collection. So as I mentioned, this was decided way back in many years ago at the general session to develop this global database on antimicrobial agents intended for use in animals, and it's evolving over time. So the first round covered the period from October 2015 to May 2016, with the report published in December 2016. And that point, because it was the first of the reports, it covered data ranging from 2010 to 2015. And then these have been an annual event. So we had the 4th round published in February last year. The 5th round, the report will be published soon, and we obviously are looking forward to seeing what changes have been made and what information has been collected in that latest report. And also the 6th round, because there's an overlap between collecting data and publishing the report. So the 6th round of data collection has also been started. So members have been very helpful in reporting antimicrobial use in animals. So this slide is just from some data from the 4th report. So this was published early last year. And you'll notice that on the x-axis, we have the different regions. So Africa, Americas, Asia, Far East and Oceania, and Europe. And then on the y-axis, we have the milligrams of antimicrobial agents per kilogram of animal biomass. And this is the amount of antimicrobial agents that are used in animals. Unfortunately, the global consumption of antimicrobials is quite high, so averaging around 144 milligrams per kilogram of animal biomass. This covers terrestrial and aquatic food-producing animals at this stage. You may have noticed that unfortunately the Asia-Pacific region has the highest antimicrobial use in animals. So really there is quite a lot of room for improvement. This particular slide is looking at the use of antimicrobial growth promoters. So in the whole of the report, 118 countries did not use these growth promoters, but 35 countries still do, and 9 of those were in Asia and Pacific region. So the chart on the left shows a list of different antimicrobials that are used for growth promotion, and then the length of the bar shows the number of countries reporting their use. And obviously that's a broad range of different antimicrobials that are used for promoting growth in livestock. The OIE is encouraging countries to phase out the use of antibiotics for growth promotion in the absence of risk analysis. And this is particularly true for those antimicrobials that are deemed to be of importance for human health, and also those that are important for animal health. So if we know that we have the potential to use some specific antimicrobials for a disease that is resistant to many antimicrobials, then obviously that's something that we want to remove from use in growth promotion. So let's look a little bit more detail about the reporting of these antimicrobial agents for this report. So we call the sections in the template or reporting options. So we have 1, 2, and 3. And these collect the quantities of antimicrobial agents that are intended for use in animals. We start off with the blue box, so that's qualitative data, really just baseline information that we hope all members will be able to respond to. And then in the green box, we have quantitative data with these 3 reporting options. For reporting option 1, we're really just looking at the antimicrobial classes and the type of use. So that would be whether it's for veterinary medical use or growth promotion. So that's the big differentiating point there. For reporting option 2, we're also, on top of the antimicrobial class and type of use, we're also looking at animal groups. And for this, we're looking at whether it's terrestrial food-producing animals, aquatic food-producing animals, or companion animals. And obviously the ability to be able to report this extra level of data differentiation depends very much on the member country and whether they're able to collect the data. Reporting option 3 is obviously what we're hoping that we will be able to get from everyone, and this also includes the routes of administration. So really trying to understand better how these antimicrobial agents are used in animals. So you'll have seen from the earlier slide that we're looking at antimicrobial data, and obviously that's not easy to calculate or to get. So how do we find out what the antimicrobial quantities are? So if we just follow this chart down on the left, we have some mandatory data. Obviously, knowing the active ingredients is very important. So that would include the molecule name. So for example, is it enrofloxacin, is it ampicillin, and also the concentration and units used. And this will obviously help with the calculation of how much of the molecule is within the antimicrobials used. And then the package size. So if we have a 1 liter package or 1 kilogram, then that obviously helps to get that calculation. And then on the right-hand side, we have this optional data, and that will include, as I said, the target species, for example, if we have something that's specifically for use in poultry, and the routes of administration, so if it's oral or injectable. In 2018, there was a second global conference on antimicrobial resistance. And at this conference, it was recommended to further develop the OIE data collection on antimicrobial agents intended for use in animals, converting the current spreadsheet format to a database system. So this initial format was quite difficult to use, and obviously it made it quite cumbersome for members to complete. But hopefully by using a database system, we'll be able to accommodate data submissions by animal species. It can also be connected to the World Animal Health Information System, or WAHIS, and also allow addition of data from field studies. So really quite a big task to take on. So I'm going to just give an outline of this project. So obviously we need to set up the software, the tool to help countries with the OIE annual data collecting on antimicrobial use. We need to involve the countries in developing that tool because it needs to be useful for their needs. And we need to improve data quality and accuracy. So we need to be sure that we have the information that we need for the report, but obviously that needs to be of high quality, and to make sure that the calculations are done correctly, and if there's an error that we can detect it easily and check the data. And then we want to improve countries' ownership of their data. So this is quite important. We don't just want to produce this report that is useful for the OIE, but not for the members. So as a timeline, so back from 2018 was when this recommendation to improve the system started. Excuse me. In June 2020, the project was kicked off. And then in October 2020, it started to be developed. January 2022 is the goal for the pilot phase, when hopefully we will be able to have test runs in different countries, with a provisional launch in September later that year. And just looking at the future, so the plan for the portal that countries will be able to see, they will be able to have lots of information that they can view and use. So data analysis, so looking at key data, monitoring the use of antimicrobials, analysing the data and being able to use that to support decision making. So something that will be useful for countries looking to implement antimicrobial plans for combating AMR. Calculation module, as I've said, to assist in those calculations that are always a bit tricky. And that will be available for countries to collect data 24/7. Now we're hoping the countries don't have to work 24/7, but obviously that means we're available for data to be input around the world. And obviously having accurate data is very important. There will be a round of data collection, and that can be either online or using a template. There will be clear progress and stages in that process. And obviously, if member countries have any questions, they can contact the antimicrobial use team at headquarters to get some backup and to explain things. It's important that we can look back on data so we can see whether the use of antimicrobials has increased or hopefully reduced, but also looking at changes in the types of antimicrobials that are used. So it's very important to be able to access data from previous rounds of collection and then analysing that. The data can be downloadable, so the member countries will be able to access it and download it for use in other purposes. And then there will be a possibility to update any historical data too. The resources page will obviously include some user guides to help with training, and obviously having templates for guidance and the calculation. So this is probably the biggest benefit will be a reduction in workload, we're hoping. So the current manual processes are very time consuming to collect data from lots of different sources to input to the data collection. But something that's also important is that it can be difficult just because those sources maybe are very spread out. So it's useful to have this easy access system that hopefully will make it much easier to input the data. Obviously, having the calculations done automatically will be really useful. So calculating the quantities of antimicrobials, having that data collection and recording all the time, and hopefully reducing errors and enhancing quality of data. So this is kind of our the simple sum. So we have the amount of antimicrobials used over the animal biomass, and that's our simple calculation. But by linking to WAHIS, hopefully that will help to provide an analysis of the antimicrobial quantities adjusted by animal biomass. And there will be a public portal, so some of the information will be publicly available, obviously not things like product information or any sensitive data. There will be regional and global analysis will be possible. There will be access to the OIE report. So these are currently online on the OIE website, but these will be included in this new public portal, and also some other resources. So looking at future steps, so this is a very simplified diagram of what happens with antimicrobials. So we know that the main points that we get data currently are from imports, so maybe member countries which import some or even in some cases all of their antimicrobial agents. They have data from imports. Data from sales, and that's at a number of different levels. So again, it might be for medicated feed, or it might be for sale to farms at the veterinary practice level. Also combining that with data from prescriptions, as if you remember that one of the 5 only rules is to use prescriptions for selling and using antimicrobial agents. So data from prescriptions and also farm records to give us more information about antimicrobial use at different levels. And then hopefully that's going to lead to the possibility of reporting at species level as we try and see which antimicrobials are used in which species. So just a quick summary of the future database collection. So species level reporting, hopefully that will be allowed in the future system. We'll be able to integrate with OIE WAHIS. And this calculation module, I think, will remove a lot of headaches for the focal points for veterinary products who have to do this every year. There will be data ownership. So the data will be available to member countries to use for their own purposes as well. And they can use that to make informed decisions. And also allowing faster and accurate data analysis and reporting. So that will be all done much quicker. And my final slide is kind of going back to that very first image where I was showing this interaction between humans, animals, and the environment. And really, when we're thinking about antimicrobial resistance, we do need to consider that AMR, those antimicrobial agents, and the resistance that unfortunately comes with them is found really everywhere. So we need to combine across sectors and think in one health approach. I'm thinking about animals, thinking about humans, and also thinking about the environment. So wildlife and waterways and the environment itself, as we think about how to tackle antimicrobial resistance. Thank you very much for your attention. And if we've got some time, I'm happy to answer some questions.
Thank you, Lisa.
though we are running a bit late, but we started late also. So we can have few quick questions or comments. If somebody wants to have any questions or comments on that.
She has told about the country portal of the all data. So I just want to mention whether informed us about the country portal. Our country have the own portal like this for the information.
Sorry, I just caught the very end of your question. Could someone repeat that?
Can you repeat it?
Ma'am, you have mentioned country portal. So India have our own country portal. We were able to get all information about antimicrobial agents.
Okay. So the portal is really for part of this global database on antimicrobial use. So the OIE is collecting data from all member countries around the world so that we can look at the global picture. And if India already has its own portal, then we hope that perhaps that will make it easier for India to report, to easily transfer that data to the submission for the global database. That would be really useful. Thanks.
Exactly. Thank you.
Any other comments or queries? If nothing else, I would like to thank Dr. Lisa for wonderfully explaining the OIE initiatives, the way they are developing the communication material for AMR and offer to the member countries to translate those into their local languages and basically distributing them for better awareness. But the basic highlight was on the method of data collection on this. And if Lisa, you are there, I would like to have a request from your side to you. If you can develop some kind of tools to collect this data before this is reported. Lisa, are you still there? Yeah.
Yes, I'm still here. What we have been doing is we have been having workshops within a number of different countries to try and understand. Obviously, within each country, they have different means of making and selling and using the antimicrobials. So there are different points. So I mentioned import and different sales points. So understanding that chain of how antimicrobials are sold and used within a country is the first step. So trying to understand that is the first thing. So looking at the countries, certainly we can help to go through that with each member country if they need help. And additionally, there is in the near future there will be a new tool coming from FAO and OIE on monitoring of antimicrobial use at farm level. So just trying to look at one point in that chain of antimicrobial use, but probably, hopefully this year, but that's in the pipeline too.
Great, thank you very much. So if we do not have any other further questions or comments on that, I would like to thank profusely Dr. Lisa Thompson to highlight those issues, especially the AMU. Thank you very much, Lisa.
Thank you so much.
Now I would like to invite, this was the perspective from the animal side. Now I would like to invite Dr. Anush Sharma to basically highlight the human perspective into the AMR issues, the WHO perspective on policy. Dr. Anush Sharma, please.
Thank you very much, sir. First of all, I'd like to thank the organizers for inviting me to share my perspective. And before I start, I'd like to just share WHO is different from OIE. OIE is an intergovernmental organization, international. WHO is a UN organization, United Nations Lead Agency for Health. It's a member state organization. So what that means is the 194 member states that we have, the health ministers of the 194 member states dictate the WHO agenda. Our highest governing body is the World Health Assembly, and all these 194 health ministers of these member states are our bosses. The Indian health minister is our boss also. So the Indian Health Minister, Dr. Harshvardhan, is also the chairman of the WHO's executive board, and there are weekly meetings which are held through teleconferencing with the 35 member states who are part of the WHO's executive board. I will focus my attention on what is the WHO India perspective on policy options for a One Health containment of antimicrobial resistance. And this is an adaptation of a presentation that we made at the Prime Minister's Advisory Council meeting for Science, Technology and Innovation, which was chaired by the Principal Scientific Advisor, Dr. Vijay Raghavan, and co-chaired by Dr. Vinod Paul. All of you are aware of the impact of AMR, and I will not reiterate that, but I would like to draw your attention to everybody is bothered about COVID. And if we put that into perspective, COVID has up till now taken 2.6 million deaths, slightly over a year. But antimicrobial resistance, if it is not checked, is likely to lead to 10 million deaths every year by 2050 onwards. So 4 times more lethal than COVID by 2050, and 1 death every 3 seconds. Also, India has reported high levels of resistance, and we have resistance in the disease control programs also. But it's very important to understand why we need One Health action for AMR containment. It's not only human health, it's also animal health, but it is also the food sector, not just the food animals, but plant health, agriculture is also equally important, and so is the environment. Because antimicrobials are used in both human health and animal health. And you find AMR bacteria in all these 4 sectors, and you'll have antimicrobial residues in the environment and the food. 70% of the antimicrobials are used in the animal sector. And this is the antimicrobials that are manufactured at the gross level. And a lot of these antimicrobials are excreted in the urine and feces, both by humans and the animals. So the area around the farms and the hospitals is likely to be high in antimicrobials. Also, a lot of these wastes end up in open dumps, and a lot of people throw away antimicrobials in household waste. So you can also have that cluttering up the environment. And also, aquaculture is another area which can lead to a lot of antimicrobials leaching into the environment. Also, pharma industry is another area which can also lead to environmental pollution. India has the highest consumption of antimicrobials in human health in absolute terms. We are number 1 consumer of antimicrobials, although per capita consumption in India is much lower than some of the developed countries. But in the food animals, India is roughly 5th, much after China, USA, Brazil, and Netherlands. But again, it is likely to increase, and India is likely to become the 4th highest consumer of antimicrobials by 2030 because of the increasing focus on food animals and the consumption patterns. And also the antimicrobial consumption, especially in chickens, in India is likely to be quite high in the next years. Now, a little bit about some of the issues which have been done. At the global level, we have the One Health tripartite, FAO, OIE, and WHO, and Lisa beautifully sort of enumerated all the activities which are being done. In addition, UNEP is being roped in to address the environmental perspective of AMR. WHO identified AMR as a theme of the World Health Day 2011. And then we had the Global Action Plan. In India, the Ministry of Health organized this international conference on AMR in Feb 2016, in which WHO Regional Office and the India Office jointly supported this. And we had more than 500 experts from around the world, especially in the Southeast Asia region. We had 6 ministers from the region who participated in this meeting, and everybody agreed to work together. Another a landmark event was the high-level meeting on AMR at the United Nations in September 2016, in which a lot of other organizations like the World Bank, Asian Development Bank, and UNEP got roped in. And again, AMR is one of the critical issues which needs to be addressed if we have to achieve the Sustainable Development Goals. One of the recent things which has happened last year is the establishment of the One Health Global Leaders Group on AMR. This is follow-up to the Interagency Coordination Group on AMR, which was established by the United Nations. Their recommendation, the Secretary General has established this One Health Global Leaders Group on AMR. This actually has leaders, either heads of state or heads of ministries from a number of countries around the world. And from India, we have, it also has people, representation from industry and NGOs. And from India, we have Sunita Narayan, the chairperson of Center for Science and Environment, who's a member of this group. Also, I think Dr. Malik mentioned the Prime Minister Modi talking about antimicrobials in his Man Ki Baat in 2016. But before that, we had the Jaipur Declaration. All the member states of Southeast Asia region signed this declaration. WHO India office put together this background document, AMR and its containment in India, in collaboration with the Ministry of Health, NCDC, and ICMR. And this led to the development of the National Action Plan and the Delhi Declaration on AMR in April 2017. And these two documents were launched at the Interministerial Consultation on AMR in April. Also, there's a BMJ special edition on AMR in Southeast Asia that WHO helped to put together. Now, this is India's National Action Plan. And I will subsequently go through each of the strategic priorities, but it's important for you to understand Dr. Praveen Malik beautifully enumerated the 6 pillars of the National Action Plan and what needs to be done for each of these areas. But I would like to draw your attention to the 16 focus areas in these 6 pillars. And it is important to understand everybody has to work together with a One Health approach. So if a focus area has a white background, that means all sectors have to work together in that. If it's a blue background, that means it's only for human health. And if it's yellow, it's only for food or the animal sector. And if it's green, it's only for the environment. But a white background means it's a One Health issue. And just to exemplify this, human health has to work across the 6 pillars. All 6. Only few areas it is not working because they are domain specific to the other sectors. Now let's look at the food, animals, and the agriculture sector. Again, they need to work across all the 6 strategic priorities, all the 6 pillars. So except for 2-3 areas, just like human health, they also need to work across these pillars for a One Health approach. Same is true for the environment. If we talk of awareness and understanding, surveillance, regulations, infection prevention in the environment, they need to work across all these strategic priorities. So let me go through each of these strategic priorities and give you a brief overview of what are the key things which were promised in the National Action Plan. And National Action Plan is already 4 years old. Now it only has 1 more year, and we need to start thinking of reviewing it and developing a new one. In fact, we had planned a review of the National Action Plan last year. Unfortunately, the pandemic put this on hold, so we were unable to organize this last year. But the first strategic priority talks of two focus areas to strengthen awareness and understanding of AMR. One is to raise awareness through communications and IEC. Target group is the general population, policymakers, and the farmers. But if we talk of the technical stakeholders, we talk of education and training and building their skills. So both of these two focus areas go hand in hand, and it needs to be across all sectors. Again, Dr. Malik talked about behavior change strategies, and that's something which was promised in the National Action Plan, and we need to think about it where we are. Also, we need to revise the training curricula to ensure AMR is strongly focused in the curriculum of students, not just veterinarians, not just human health, but also dairy technologists, fishery professionals, or the other allied sort of services. Also food technologists. Also, we need to think about how can we consolidate communication and information resources. Because recently there was a question in the parliament on what has the government done on AMR, and the reply focused on a very small set of initiatives done by the Ministry of Health. Even all the initiatives of the Ministry of Health were not answered in that written reply, which is there on the PIB website, and it was released, I think, 2 days back. Also, we have a lot of e-learning platforms. How can we consolidate the Diksha portal, or the iGot portal, or e-Gurukul portals to strengthen education? And school education is important because children are the future citizens and leaders. To ensure that we have effective antimicrobials, we need to focus on them to bring about a behavior change in the consumer attitude towards antimicrobials. Also, is it time that we stop focusing on World Antimicrobial Awareness Week and have a continuous campaign throughout the year rather than just focusing our attention in 1 week in November? And this needs to be there at the national level, state level, and in the facilities. Second strategic priority focuses on labs and AMR surveillance, strengthening the laboratories and surveillance of AMR. India's National Action Plan has taken the surveillance of antimicrobial use to strategic priority 4 because that is where the synergies lie, because that is used for antimicrobial stewardship to optimize its use. Surveillance of AMR is closely linked to laboratories, and these laboratories have to be there in hospitals, environmental labs, veterinary labs, fishery labs, food labs, across all sectors. And we need to also be able to strengthen our labs to detect the residues. And we need to have networks in all sectors and all levels. We have the INFAR network in animal husbandry and fisheries. We have FSSAI network in the food labs. In human health, we have one network by NCDC called National Antimicrobial Resistance Surveillance, a network called NARSNet. And then we have the ICMR network, which is the AMRSN, Antimicrobial Resistance Research and Surveillance Network. Both NCDC and ICMR have 30 labs each. In addition, WHO has established 3 more surveillance networks, which are state-level surveillance networks. One in Maharashtra called Mahasar, another in Kerala called Karsnet, and a third in Delhi called VINSAR-D, WHO-IMM Network for Surveillance of AMR in Delhi. And we are looking at having these networks in all the states and union territories of Delhi. We are doing this in a phased approach. We also have a third national level network, which is still not operational as yet because of the pandemic. It is called VINSAR. In human health, it is in partnership with the IMM. Again, the questions I would like to pose to all of you is it time that we talk of national standards for AMR surveillance, which are aligned to global standards, but are according to India's needs? So the breakpoints that we use are not dictated by any Other country, they are dictated by Indian scientists, Indian researchers, and based on India's experience in this area. Currently, we have 3 AMR repositories, one led by CSIR at MTech Chandigarh, another by Department of Biotechnology under the Mission AMR program at NIMR in Pune, and third is ICMR's NICED repository. Is it time that we align all of them so that we have a cohesive approach for the AMR repository? Also, is it time that we have a national AMR report or a dashboard so that when anybody asks or anybody wants to seek information, right now information is fragmented in silos. I am aware of a number of initiatives, but even me, I am aware of only a small fraction of all the work being done in the country. So is it time that we have a national AMR report or a dashboard which captures all this information for policy making and for spreading awareness? Third strategic priority, very important for all the 3 sectors. And here the 3 sectors are working not in silos. They need to work in coordination, but there's a strategic thing because IPC in human health has different connotations from IPC in on the farm. It's biosafety and biosecurity on the farms. It's not infection prevention and control as in the human health. And again, Swachhata, the mission or the Swachh Bharat mission will ensure sanitation and hygiene. So we need to prevent healthcare associated infections, have good IPC in veterinary care, animal husbandry, good biosafety, biosecurity, and also in fisheries and agriculture to reduce the spread of resistance pathogens and the genes and the residues in the food and the environment. Is it time that we have a national program for infection prevention and control? The pandemic focused on infection prevention and control in a very big way. I was given additional charge of IPC in addition to AMR and labs, which are my core areas. So IPC, because it has synergies with AMR, and a number of countries and WHO advocates for a national program on IPC because it's a cross-cutting area. And the pandemic strongly highlighted the role of IPC in healthcare. So we need to have HAI surveillance in place, also have HAI surveillance in place in veterinary hospitals so that you don't have healthcare associated infections in the veterinary sector as well. Also, Swachh Bharat mission, Ayushman Bharat are strong synergies where if we can work together with accreditation agencies. Also the Kayakalp program, which looks at improving the quality of healthcare facilities. We have the national IPC guidelines and the national patient safety implementation framework, which strongly highlight the importance of IPC. Fourth strategic priority is optimizing the use. Again, as India, we need to balance access to antimicrobials with access. In our remote areas, a number of people, especially children, still die due to pneumonia because they do not have access to antimicrobials. And in urban areas, we have the problem of excess. People popping antimicrobials. And this needs to be in all the sectors, not just human health or the food animals. Primarily in these 2 sectors. We need to have treatment guidelines for use of antimicrobials and also the implementation. Some where we do have guidelines, but a lot of times they're not implemented. How do we ensure implementation? And we need to have antimicrobial stewardship programs at all levels and sectors so that we can optimize the use of antimicrobials. And how do we include AMR in accreditation checklists, whether it is healthcare facilities or the veterinary hospitals? Fifth strategic priority focuses on research, innovations, and financing. And the first focus area spreads across both the human and the food animal sector, where we need new medicines for treatment, we need diagnostics, we need vaccines, we need alternatives. And I'm very glad that today the university has entered into a collaboration with Dabur Ayurved for a collaborative work, and I hope this will lead towards newer alternatives to antimicrobials. Also, we need research on new drugs, novel treatments. Diagnostics are also very important. How do we ensure to differentiate bacterial from a viral infection? And DBT is doing a number of, supporting a number of projects. So is ICMR and ICAR and CSIR and DST as well. And all of them are focused over and aligning it with Make in India. Also, we need to have an operational research program on AMR. Every institution or a research organization focuses on their own priorities. How do we bridge these silos and see what works, what doesn't work? We partnered with the Department of Biotechnology to develop this Indian priority pathogen list to guide research, discovery, and development of new antibiotics in India, aligned to the global WHO's priority pathogen list, but this is completely aligned to India's needs. And this was launched at the recent Bio India by the Dr. Harshvardhan, who's also the Minister of Science and Technology who oversees DBT. Also, we need to have a One Health AMR research agenda for informed, evidence-informed policy making, cutting across sectors, across departments, across ministries. And most importantly, how do we ensure funding for AMR? That is the biggest challenge. And this is something I think has been a challenge for us. We are trying to work on the impact of AMR in India, a paper, so that the policymakers start investing in AMR. Some of the states which have already invested in AMR, and I can give you examples in the next slide. Kerala, for example. Why do you think they prioritized antimicrobial resistance? They did it actually back in 2011 because they realized the impact. But more importantly, it got a big sort of boost. They started a liver transplant program a couple of years ago. And as I think Mr. Monji Saxena said, surgery is successful, but the patient doesn't survive. The first successful liver transplant, patient died due to an AMR infection. And that really boosted Kerala's state action plan. And the last strategic priority is on collaborations, international collaborations at the global level, regional level. India is leading in the Southeast Asia region. Also in BRICS, it is always AMR has featured on the BRICS agenda, ASEAN agenda, and the G20 agenda. All the last 4 G20 meetings, there has always been antimicrobial resistance, which all the G20 countries agree that they will work together for AMR containment. And the AMR hub is one of the outcomes of this G20 efforts for AMR containment. DBT is India's a member on the AMR hub, which is located in Germany. Again, we have a number of disease control programs at the national level. And I'm aware of the ones in the human health, but even the animal health, we need to have collation of this information and sharing, maybe on the AMR portal, across the programs, so that we can get that information. But most importantly, health, both for humans and for animals, is a state subject under the Indian Constitution. So we need to have the states on board. So the last focus area is extremely important, state action plans on AMR. And for this, we already have guidance documents which we prepared along with the Ministry of Health on how to develop state action plans. And we've supported the development of state action plans in Kerala called CARSAP, in Madhya Pradesh called MP SAPCAR, and in Delhi it is called SAPCARD. And again, it's very important to have them as multi-sectoral, One Health, AMR coordination efforts. How do we ensure regional engagements amongst the states and how to align them with the state implementation plans of the NHM, the National Health Mission, or the other programs of the DHD, which are there at the state level. Again, this is my second last slide. AMR containments need human resources. In all sectors, we need microbiology and the lab professionals. We need teachers, we need trainers, scientists, researchers, economists. We also need communication experts, media professionals. Again, the various things. And then we also need specific people, and this is an incomplete list. This is just to give you an idea that how many people are involved. And AMR is everybody's business, but we also need to keep in mind training has to be kept in mind. How do we build their skills? How do we provide them the infrastructure or a system in which they are able to deliver? This is the list of key stakeholders, the key ministries across. You have the Ministry of Health, Ministry of Agriculture and Farmers Welfare, Ministry of Fisheries, Animal Husbandry and Dairying, Science and Technology, Environment, Forest and Climate Change. We have the Jal Shakti Ministry now. We also have Niti Aayog, which is above all this, and the principal scientific advisor to the government of India, which is above these ministries and monitors their work. We have the Ministry of Ayush, the Ministry of Chemicals and Fertilizers, where Department of Pharmaceuticals is important, Information and Broadcasting, External Affairs, and a number of other stakeholders within these ministries, which are extremely important. Again, how do we align them with Swachh Bharat mission? The Jan Arogya program, I think the PM Jan Arogya Yojana, the Make in India, Atmanirbhar Bharat Abhiyan, and most importantly, not to forget the private sector. Again, this is an incomplete list. There are a lot of professional bodies, a lot of associations, NGOs, which also need to be a part of these efforts. So last slide. Again, I'm raising more questions than giving answers. I think we need to review, revise, and develop a new national action plan. Also have redefining the priorities, achievements, stakeholders, and timelines. The National Action Plan had promised the establishment of a national authority for AMR containment. Maybe we need to relook at it. Do we need it? Or do we need to have another mechanism to have AMR coordination across all the ministries, departments, and stakeholders? And also, do we need a drug regulator for the non-human health sector? Because that is also a relatively weak area where drugs get smuggled across borders in huge gunny bags. It's not small amounts, and so better regulation of those also need to be there in place. How do we elevate AMR to a sort of a mission mode? How do we budget AMR into the work plans, including staffing? Because unless, and the reason I say staffing is, we need human resources if we need action against AMR. And the government, the biggest challenge is, first thing which is cut is if you ask for more manpower, that's the first thing which gets cut off. So we need to start thinking, how do we do it? How to strengthen AMR labs to monitor the AMR trends? And can we have centers of excellence for AMR containment? And last thing to think about is, how do we have coordination mechanisms for One Health AMR? The existing AMR governance mechanisms, there's an intersectoral coordination committee, which is chaired by Secretary Health with joint secretaries of all the other ministries, which has not met very frequently, only 2 or 3 times. We have a technical advisory group where all the stakeholders are not there. And also a core working group, which is chaired by the director of NCDC. The tag is chaired by DG ICMR and Director General of Health Services. Do we need task forces on AMR across 6 strategic priorities, which are chaired by senior professionals across sectors? For example, for research, could we have DG ICMR, DG ICAR, Secretary DBT, and DG CSIR be co-chairs of this task force, and the secretariat revolves amongst the four on a quarterly basis so that everybody is accountable. Same way we can have the task forces for the other five strategic priorities, two or three or maybe even four chairs depending on the sectors involved, and have cohesive sort of outcomes and outputs for AMR containment. So thank you very much for inviting me for this program, and I would like to especially thank Dr. Amit Kumar for inviting me and the university for organizing this. Thank you.
Wonderful lecture, sir. Thank you very much. And before we finish, we can have few quick questions or comments. If anybody wants to say something on this.
Sir,
Some questions on online. Right, doctor, please.
Sir, Dr. Pankaj Chauhan here. Sir, excellently, Dr. Sharma, you have spoken well, and we are living at presently in COVID pandemic. So definitely for us, this is the very interesting and resourceful lecture. So you talk about the surveillance, AMR surveillance. So what is your working policy of this surveillance? How do you work?
Surveillance is the first priority that WHO identified after the World Health Day. In fact, in India, we identified AMR surveillance even back in 2008. WHO India, along with the WHO Regional Office, established INSAR, Indian Network for Surveillance of Antimicrobial Resistance. Dr. Rajesh Bhatia was there from the regional office. I was there from the country office. And we handpicked 20 labs across India, got them together. They had minimum standards in place to be a member, so that we have quality data. And then we started sharing of data on an annual basis. And this continued for 4 years. After that, sadly died. But all those sites are still a member of either the ICMR surveillance network or the NCDC surveillance network, most of them. Now we are focused on, for a country the size of India, we need to have more surveillance labs. We work on the human health, but we need INFAR at the national level, but then we need INFAR at the state level also. So we are focused on supporting 30 labs of NCDC, 30 labs of ICMR, 30 labs of WINSAR at the national level. But for each state, we are looking at establishing state surveillance networks where the state government takes ownership and shares this data at the national level. So that's how we are doing this. And at the global level, we have the GLASS system, the Global AMR Surveillance System, where all our member states are expected to share the data. India has been sharing the data for the last 4 years now. India is a member of GLASS and shares data, and data from both the NCDC and the ICMR network is shared in GLASS.
Sir, you raised the issue of some financial problem. Sir, you told about the great collaborators are with you. CR, BRICS, Asian, G20. I think there is some financial issue. So
see, financial issues I talked about, the financial issues are not at that level. Financial issues are at the local level. Within the ministry or the department, AMR gets left behind. So we need to have cohesive action. And even right now, there is no separate budget for AMR. The National Action Plan, unfortunately, in India is not a costed action plan. A lot of other countries have budgeted, costed action plans. So maybe the next time when we develop our action plan, the National Action Plan, we'll have it as a costed action plan, and we'll have clear, our aim was the same. We actually identified which ministry will be leading which initiative or department. And we actually expected that they will come back to us that we are investing this much into this. That didn't happen. So we need to have that action happen, that AMR is budgeted in the department or the ministry's financial plan.
In fact, just to inform, just to supplement this, Dr. Sharma, when Infar has started 4 years back, is the same position till now. In this EFC, we are planning to put some budget. Otherwise, this was an activity started by 20 labs without budget. So it's like we have to create these kind of positions. Dr. Sharma, you want to say something?
And I think, sir, we should proud of our India. Such conditions we are providing our vaccines to other countries also.
Explained all the things. Now, on these two counts, sir, I have a basically request to you as well as to you. Eventually comes the question, budget comes from where? If clean milk production program adopted by NDDB, supported by Department of Animal Husbandry way back in 2004, when every milk sample was found to be contaminated with antibiotic, and India was preparing itself for cheese production. So whatever companies were trying to produce the cheese, they had this problem that milk does not curdle. And if you all recall, दही जो है घर में जमता था. जब मदर डेयरी ने दही जमाना शुरू किया, अमूल ने किया, ब्रिटानिया ने किया, तो सबके पास ये समस्या आई कि कल्चर हम डालते हैं, दही अच्छी क्वालिटी का नहीं आता. तो वहाँ इकोनॉमिक इशू आया कि भाई अगर कोऑपरेटिव्स को ट्रेन करें और उनको प्रीमियम प्राइस दें कि भाई एंटीबायोटिक बल्क सोमेटिक सेल काउंट देखेंगे, मेस्टाइटिस कंट्रोल करेंगे, तो वैल्यू गॉट क्रिएटेड, जिसमें कस्टमर को अच्छा दही मिला, इंडस्ट्री को अच्छी प्राइस मिली और फार्मर को अच्छा दूध मिला. इवन केरला में भी श्रिम्प एक्सपोर्ट होती है और हर बार उनका क्लोरमफेनिकॉल और नाइट्रोफ्यूरेंटाइन की वजह से जापान उनको नेगेटिव लिस्ट में डालता था, यूरोप ने तो बैन ही कर दिया था, और एक बार बैन लिस्ट में आ गए तो पॉजिटिव आने में 2 साल का समय लगता है. अब यहाँ पे इशू ये आता है कि मिनिस्ट्री ऑफ कॉमर्स फॉर डॉलर दे आर रेडी टू डू शीर्षासन एक्सपोर्ट बढ़ाने के लिए, लेकिन इस प्रोग्राम को फंड करने के लिए कि अगर आप AMR कंटेंट करोगे तो हमारी कॉस्ट ऑफ प्रोडक्शन ऑफ मिल्क, मीट, इवन फिशेस इज लोएस्ट इन द वर्ल्ड, और अगर हम उसको एक्सपोर्ट कर सकते हैं तो वी विल क्रिएट द वैल्यू एंड वी विल गेट द फंडिंग ऑल्सो. नाउ सर, एक रिक्वेस्ट हमारी है सर, आपसे भी है सर, ये हमारा पर्सनल ऑब्जर्वेशन है, पर्सनल बेसिकली एक्सपीरियंस है कि पहले यूनिवर्सिटीज को सफिशिएंट फंडिंग मिलती थी टू कैरी आउट रिसर्च वर्क ऑफ नेशनल इम्पोर्टेंस. आज सर क्या हो गया है कि somehow universities are being starved बन गया, लेकिन मैन पावर और रिसोर्स मीन्स रिएजेंट्स एंड टू कैरी आउट रिसर्च, वो उनको कॉन्फिडेंस में नहीं ले रहे हैं और उनको एक ऐसी नज़र से देखने लगे हैं कि अगर आप देखेंगे कि जो प्रोजेक्ट्स अप्रूव भी होते हैं उसमें उनको लो प्रायोरिटी मिल रही है. प्राइवेट की हम नहीं बात कर रहे कि आप सर्विलेंस की लैब आप बनवा रहे हैं. सर, एक यूनिवर्सिटी और प्राइवेट के साथ भी एक्सपेरिमेंट करके देख लीजिए, नहीं होगा तो आप उनके ऊपर मुकदमा करके फांसी लगा दें.
उसके लिए ये तो लंबा टाइम हो जाएगा बिकॉज टाइम इज लिमिटेड, सो वी कैन डेलिबरेट ऑन दैट ड्यूरिंग लंच टाइम एंड आई कैन एक्सप्लेन देयर आर ample opportunities for the universities also.
यूनिवर्सिटीज एंड प्राइवेट भी सर, नहीं तो फिर
अल्टीमेटली
आधार पूना वाला ने देश को बचाया है सर.
दे आर नॉट कमिंग फॉरवर्ड टू दैट. R&D का जो बन रहा है उसके लिए मैं बात करूंगा कि हाउ DHD एटलीस्ट इज बेसिकली वर्किंग to bring the private industries also into the R&D ecosystem.
Very good, sir.
इकोसिस्टम नहीं बन रहा है, और इकोसिस्टम ना बनने में हमारे रेगुलेशंस भी रेस्पॉन्सिबल है, हमारा माइंडसेट
हाँ
भी
सर,
रेस्पॉन्सिबल है,
बिल्कुल
खुद हम भी रेस्पॉन्सिबल हैं फ्रॉम द प्राइवेट सेक्टर
नहीं,
एज वेल.
सभी लोग हैं सर, अब
So
सभी लोग हैं
we are trying on that, and probably we can discuss in detail during the lunch time if you want to respond quickly. Quickly, sir.
Just one thing, we also, आपने research का जिक्र किया, we actually had an ESBL E. coli tricycle APX project, which was based in Kolkata, and everything was done, and the pandemic actually played spoil sport. We had 4 institutions, PGI Chandigarh, and the Neel Ratan Sarkar Medical College, the Kolkata Medical College, and the West Bengal University of Animal Husbandry and Agricultural Sciences. These were the 4 institutions which were part of this project, and research, a lot of funds were allocated, procurements were done, reagents came. Because of the pandemic, the thing couldn't be taken off. But we are again exploring. The equipment is there, the expertise is there. It's just a matter of restarting it, and we will be doing that soon.
Just one query with there online. A simple thing. Your views on that, Dr. Sharma. Do you think AMU policing, it's basically law enforcement, is better than the persuasion?
Antimicrobial use, policing has to go hand in hand with the individual persuasion efforts. And we need to make a distinction between consumption and use. Consumption is the bulk amount which you estimate from sales, procurement data, etc. But use is the quality of use, where you're looking at individual patients, individual animals, how much is being used on individual animals. So both of these go hand in hand. And from the strategic perspective, I would advocate a joint effort where some policing and some persuasion is both there.
Thank you, Doctor. Wonderful deliberation on this, where tripartite plus has been explained, the 6 plus and the role of different sectors on 6 pillars were explained quite nicely. And the major emphasis was on the time and need for standards for AMR surveillance to be adjusted to Indian global benchmarks, and human resources need from different sectors, not only from the science but also from the sociology, psychology, and other sectors, economy. So from these point of views, thank you very much. Dr. Amit, you want to?
Sir, with the permission of Chair, I would like to inform the House that Dr. Professor A.P. Garg has joined us, Honorable Vice Chancellor of Soviet University.
Okay.
So I request Dean College of Biotechnology to welcome Dr. A.P. Garg, sir, by presenting a flower bouquet, sir. With this, I request Dr. Malik to please.
Welcome, sir. Now, without wasting much time, because we are already ahead of schedule, so late in the schedule, sorry, I would like to request Dr. Anup Kalra to deliberate on the alternatives, the herbal medicines in AMR, restricting AMR. Dr. Kalra, please. Sir, अगर आदेश किया जाए तो सिर्फ 25 मिनट है आपके पास, सर, इंक्लूडिंग क्वेश्चन एंड आंसर. नहीं, 30 मिनट दिया है सर मैंने, 5 ही मिनट घटाए हैं, ज़्यादा नहीं घटाया. प्लीज सर, क्योंकि टाइम नहीं है, अगला सेशन लेट हो जाएगा सर.
Okay. Yeah. Thank you, Chair, for giving me the opportunity. Respected Dr. Malik, Honorable Vice Chancellors, present Deans and Directors, it is indeed a great opportunity for me, along with Mr. Saxena, who is our managing trustee, to share a few thoughts on microbial resistance to antibiotics and human health, challenges and opportunities, antibiotic usage in food production. Well, before I do that, because my presentation would be overlapping with some of the important points which my predecessors have already discussed, two or three points which I surely would like to complement the industry and the academia, that in spite of all these challenges, of the funds, of the manpower, which they have been facing, but the research is still on. So that is one very positive point, that talks about the passion, that talks about the interest we have. Only thing is that this kind of interest which we have, the opportunity we have, should get translated with the policy planners to result into the equal understanding for making the resources available. Second point which was brought out was that the animal industry uses 70% of the medicines, the antibiotics per se. Now, again here, what was discussed in the first session, that we don't have the realistic data. These are some assessed data, that 30%, 70%, soil doesn't come in between, where soil usage is much, much higher. That's number one. Also, looking from the animal health, you look at the cow or a buffalo, that's 400 kg. So the requirement naturally is going to be higher. So there are some paradoxes, which at the moment I would not like to deliberate. But yes, the animal husbandry sector is now gradually getting more aware and trying to ensure that how do you use the antibiotics in a more rational manner. And a lot of these industries have shifted to, whether phytogenics or using organic acids, because they are also conscious, as we all are, about our own health. Because ultimately, whatever food they produce is going to be consumed by their own children as well. So in this backdrop, I would just like to share some of our views in this. Now, there are important production diseases, whether they are for dairy and poultry, and they are all of economic importance, on which the industry has been working, the academia has been working, the ministry has been working under the guidance of Dr. Malik and his predecessors. But what is more important here, as we discussed in the morning, AMR since inception got missed out. Gradually, it is now gaining more attention and more importance. Okay. Now, with this, sorry, can someone help me? यहाँ से जाएगा ना, यहाँ से, okay. And this point also has been, discussed by my predecessors, that the usage of antibiotics, indiscriminate use of antibiotics. Some people say livestock industry uses. I say I also use it. People like me also use it, and others also use it. So indiscriminate use. Second is irrational use. Morning Dr. Malik was sharing with us that, a guy had an infection. He said, 4 days I took this antibiotic, another 4 days I took another antibiotic. And this happens with the learned people. What do you talk about the labor category? They use indiscriminate. 2 दिन ले लिया, फिर छोड़ दिया. बच गया, बोला साहब ये एक्सपायर हो गया, नाले में फेंक दिया. So ultimately all this is going to create problem for all of us in our ecosystem. And irresponsible use. So all the three are equally important, and we all are responsible towards it. And we cannot exclude anyone, any academia, any ministry, or any individual. So this needs to be looked at very closely. How do we address this? Now, this data is known, so I would not dwell on it, that, there has been ban in number of countries, mostly, how the situation started. And today in the Europe, there is a ban on antibiotics, but there are several other countries, if I can share the map with you, that there are certain good number of countries where still the usage of antibiotic as growth promoter is in use. India is one of them in the livestock area. But, I remember I had a meeting with Dr. Malik and a lot of industry associations approached him. He says, sir, ye cheez aap approve kar dijiye. He took a stand. He says, no, you call for the meeting and you explain why these kind of antibiotics should be used as growth promoters in the antibiotic. So there is definitely a transformation which is coming, but I think we are on the right track, but this would still take some more time, so that we come to a level where we say, yes, we are using the antibiotics rationally, prudently, we have the sufficient withdrawal period. So those are challenges, but I think over a period of time, things would improve. Now, Eliza also mentioned this point that people are not venturing into new antibiotics. Why? Because it requires time, it requires money, but the antibiotic, the resistance which they gain is much faster. So if you look at, the total investment which is made for research is 38 billion, and out of that, only 1.8 billion dollars is spent on the AMR, the research on the antibiotics. The reason is this. So why should anyone invest? Now, for growth promotion in food animals, people have choices, people have options, which they are using today. So one of the option people are using as antibiotic. So people refer them as antibiotic growth promoters. So if I use antibiotics, possibly it would, reduce the the growth of the harmful bacteria and possibly might help in growth, which factually doesn't happen. Some people use probiotics and enzymes, some people use phytoadditives. All these have got their merits and demerits. Now, in one of the meeting, I was sharing that use of AGP. Is it Dr. Kalra? You are talking about herbals. Why are you talking about AGPs? AGPs are antibiotic growth promoters. So then I had to share with them, look, it is not always antibiotic growth promoter. It can also be an alternative to growth promoters. So alternative growth promoters, which are phytoadditives. And they have a distinct advantage. So these phytoadditives have not been in practice in India for last several years now, but the acceptability has now gained even in Europe. The products have been approved and they are being rationally used and they have found the benefit. The only reason which I say this has happened is because now this has been based on the merit of science. We have proven it, the industry has proven it, the academia has proven it, we have got tremendous support from the government, and we have been able to prove this on the basis of science, which the Honorable Prime Minister also said that we have wealth of traditional knowledge. How do you prove it on the scientific lines is important. And our organization is working on those lines, and which is what, while I had requested the university through Professor Dr. Malik, that these are some of the strengths we have. And I think I'm grateful to the university and Dr. Malik and to the Honorable Vice Chancellor for having accepted our request for an MOU. And we're very, very hopeful that something good would happen out of this MOU and we will be able to bring some good value to this. This also has been talked about, host, environment, pathogen. I would not deliberate because, whenever you do, so host and environment and pathogen, they are all very closely related and are responsible, for the host pathogen environment relation, and which is one of the reasons which also needs to be looked at. Now, one reason is that, okay, I have used phytogenics or I have used some alternatives. Now I am free. I can do whatever I want. But what happens to the biosecurity? What happens to the management? In his inaugural address, Dr. Malik did mention that biosecurity is very, very important. Management is very important. Nutrition is very important. So it's not only that you're managing, if I take poison and then I take an antidote, now I am free. So I can do anything. It's not important. When you are dealing with your own body, you are the best judge for your own body. If you talk about the livestock system, you must know your livestock. You must know, what your poultry deserves, what your cattle deserves. Otherwise, you would say, oh, my bird is in stress. It has got infection. Pump more antibiotics or pump more herbals. I'm saying why? Ultimately it is cost. So therefore, biosecurity is important. health management is important, and therefore we have to be very, very careful and cautious. I'm not deliberating much on this owing to the paucity of the time. But one of the most important approach which the phytoadditives or the herbals have been focusing is the pro-host approach. You know, when I say pro-host approach, in short, is that how do you build the immunity of the livestock or our own body? I'm sure, you know, owing to the COVID, the terms or the herbs like ashwagandha are very, very common and commonly known to all of us. Giloy is very, very common, known to all of us. And this has been used, Giloy is most favorite to be used in kada. But then there is a science behind it. What we have tried to do as an organization, we have based the entire, looked at right from the quality of the planting material, that is the plantlet or the seed, the cultivation practices, the kind of organic matter the soil has, so that you can get the good quality material, the content of the herbs. It's not every ashwagandha is equally good, but then, you know, the contents are important. The contents of Giloy are important, which actually make the pro-host approach happen. So as an organization which has a lot of values to the research, we have made this happen and tried to ensure that whatever solutions are made available to the livestock industry or to the poultry or to the pets are creating value in terms of improving their health. We had some of the visitors who, when they visited our R&D and the factory, they said, do you produce the products for the animals here? I said, yes. He says, we have never seen such a big laboratory which is certified. We have never seen such a clean environment where you produce medicines for the livestock. So he said, sir, this is his saying, Mr. Saxena, आप और हमारे में प्लेसिबो इफेक्ट हो सकता है, गाय और भैंस के अंदर प्लेसिबो इफेक्ट नहीं होता है. इफ यू गिव देम राइट, इट will result into right. So therefore it is more important to look at from the angle that, you know, the quality herbs are used, the source of the herbs is certified, and contents are known, which is what we have been working for promoting the quality herbals to the limelight. So validation is important apart from the quality. Some of these herbs, I would not go into details. I'm sure all of us know Eucalyptus glabellus, Allium sativum, Gingiber officinalis. ये सारे नाम, कहीं पे अश्वगंधा, कहीं हल्दी, कहीं लहसुन, कहीं अदरक, ये सब हमने सुने होंगे और हम लोग इस्तेमाल भी करते हैं किचन में as a traditional use. But what is equally important, when we use them as feed supplements in India or Europe, we look at the contents. We look at when are they harvested, are they stored in the right condition. So all these aspects we look at, and which is what, you know, whenever you talk about the name in the herbals, definitely you would have Ayurved as the front runner. Just unke product use karo, wo quality products hote hain. Not that I'm trying to promote, but then this is what is the feedback of the industry, feedback of the academia, India and abroad. And these solutions of these herbs, what we have used, have been documented for their results in number of scientific validations. And now the results, we have gone to the studies where we are mapping the herbs, their roles on the what do you call, and on the cellular level, and the results are just fantastic. So some of the solutions are, as an organization which we have developed, one is N-biotic, so you can very happily and comfortably replace the antibiotics in the feed. We have recipes where, you know, this is one of the major challenge in the poultry, so you can replace these antibiotics for the respiratory challenges in livestock or poultry. Salco-check, you know, digestive or the gut health is very important. And ultimately good gut health means good immunity and good production. So we have solutions, you know, where all these three can very comfortably, scientifically have capability to replace the antibiotics. And we have been able to do that to a considerable effect. But there's still a long way to go. And in doing so, We are thankful to the academia, to the veterinarians who have helped us all along, and this has helped us while communicating that health is more important. So please focus more on the health. So if the health is better, the disease automatically will be less. Don't wait till you get the symptoms and say, now what do I do? So the principles of Ayurveda is how do you keep yourselves healthy and to improve the quality of life, which is what we have used in Ayurveda to help the industry and our stakeholders for improving their farm profits. As I said in the beginning, whatever solutions we have had, herbs we have, we put a lot of science behind them. And today we are happy to share with you, we have more than 700 scientific published reports. 40% of them are in the international journals. And this is where Ayurveda Research Foundation is located. This is in Haryana, Sonipat district, where we have a DSIR certified lab, one of the best labs we have. Now we are in process of getting NABL certification to the lab. The objective of getting these certifications is how do you take the research to the farmers? How do you help the farmers? Whether it is testing of milk, testing of feed, testing of water, testing of raw materials, guiding them, helping them to produce quality food for themselves and for us. And it is an integrated manner, 360-degree integration. And this is through a program which is known as 5F. It is sustainable integration of livestock and agriculture, which talks about how do you have food, fodder, fuel. So food for you and me, feed and fodder for the livestock, fuel and fertilizer for meeting our daily needs. And this can only happen when you have agriculture and livestock in a sustainable, integrated manner. Today, there's cow dung which is wasted. So we said we will use it in improving the soil quality, in improving the fuel needs. We got into hydroponics. We are one of the leaders today, and we have systems where you can have a 50 kg to 1000 kg machine, and you can produce every day without use of soil and very little water. So all these initiatives as an organization we have took is to promote the quality food production and sustainable integration of livestock with agriculture. So proposed action plan, possibly this has been deliberated earlier, so I would not dwell on this, but all these parameters are important. We have a special facility at our lab where we are taking initiatives to detect work on the AMR. We have a lab. We are working on testing of the antibiotics in the feed, in the chicken, and milk, and then recommending the way forward as well. And we would be very glad if we get an opportunity to collaborate with for the research or for any kind of alliance which the university feels it is fit for, or ICMR, to see that we are able to take these initiatives to the next level, whether in the area of research or education or in monitoring or surveillance. Antibiotics are a precious resource. Ultimately it is very important. So the action has to be taken today, which we have already discussed. Otherwise, there's no cure for tomorrow. And Bill Gates, in his TED talk, had mentioned biggest threat to the human mankind is not the nuclear arsenal, but the microbes resistant to all known antibiotics. So it is very important. So जैसे मैंने पहले कहा कि हम तो सचेत हैं ही हैं, हम कोशिश भी कर रहे हैं, लेकिन जब तक हम पॉलिसी प्लानर्स को इसमें इन्वॉल्व नहीं करेंगे, शायद हम अपने उस मुकाम तक नहीं पहुंच पाएंगे जहां हमें पहुंचना चाहिए. और मुझे लगता है कि अब पॉलिसी प्लानर्स भी जागरूक हो गए हैं और माननीय प्रधानमंत्री जी ने इस और फोकस किया है, और इन्हीं इंटरवेंशंस को लेके हम लोग आगे चल रहे हैं. और मेरी रिक्वेस्ट ये रहेगी कि आप में से जो भी कोई ARF विजिट करना चाहे, आपका स्वागत है. हम लोग वहाँ आपके साथ शेयर कर सकते हैं क्या हमने इनिशिएटिव्स लिए हैं, और आपका मार्गदर्शन अगर वहाँ पर भी हमें मिलेगा तो हम एक नई कोलैबोरेशन के साथ मिलकर हम आगे बढ़ सकते हैं. इन्हीं शब्दों के साथ बहुत बहुत धन्यवाद. नमस्कार.
Thank you, Doctor.
5 मिनट ज़्यादा हो गए सर।
हाँ सर, अगली मीटिंग आपको ARF में ही रखनी है, तभी देखेंगे सर।
थैंक यू।
ये वाली मीटिंग अगर आप पहले बताते तो ये ARF में ही रखनी चाहिए थी। तो ये अगली मीटिंग आपको ARF में रखनी है सर, उसको आप प्लान कर लीजिए। एनी कमेंट्स, क्वेश्चंस? हाँ डॉक्टर प्लीज। डॉक्टर AP गर्ग सर, वाइस चांसलर शोभित यूनिवर्सिटी मॉस्को।
No doubt, the herbals are better than antibiotics. But at this time, we are conducting certain experiments during the last 6 months, and we have found that even these herbals, they are disturbing the gut microbiome. So when you are taking in excess quantity, because during this COVID pandemic, many of the people who were taking the regular khana, we tried to isolate the gut microbiome of these people, and we found that some of the hefty bacteria, they diminished. Absolutely. Yes. Bifidobacterium populations, these were reduced. So before recommending even this Ayurvedic, we should also focus on the quantity.
It has to be a balance, sir.
Balance. Because if we are disturbing the gut microbiome, then we are disturbing the entire body. 70% of your immunity, it is dependent on the gut microbiome. There is no doubt about that. So when we are focused and I think this Ayurvedic, when we are focusing on the pure preparations of the Ayurvedic, then we have found that then the effect is multifold. So we should try to focus on the natural product. When we are focusing on the natural product, then we found that certain components in that preparations are which are protecting the gut microbiome, etc. So the raw herbal preparations and the pure herbal preparations should also be looked into for their effect on the gut microbiome.
Thank you, Dr. Thank you very much, sir. So, हाँ डॉक्टर साहब, quickly, सर एक क्वेश्चन और बहुत
Sir,
जल्दी।
सिर्फ only one question. Sir, this is the general question for house doctor, not only for the Dr. Karla. Sir, most of the countries, as sir have also mentioned, banned antibiotic. So why in India antibiotic should be banned?
See, Dr. I think I am the right person to answer this thing. In fact, we have got list from OIE, we have got list from WHO, which are critically important for a particular sector. So we'd like to see that these antibiotics should not be used too much on the other side, right? So we have already banned colistin, and bacitracin is on the line, okay? And we are trying to see that the things should be banned when they are to be used on a specific purpose in a specific sector. So वो जो balance करके हम लोग बात करते हैं, we deliberate on that, and in case it is found that this has to be banned for use in another sector, we do so, and that's why colistin was deliberated and was banned 2 years ago. Now we are talking on the bacitracin, which is also on the pipelines, and in case some other critical or very critical, most critical antibiotics on the list of the WHO, we can deliberate on those to see what kind of usage is there on the animal side, and if animal side can do without using this particular antibiotic in their sector. So we wanted to ban colistin for the growth promotion, but somehow the DTAB has taken a decision to have a blanket ban, where we have got certain issues on that, and we are writing back to DCGI to open it partially for the veterinary use, because in mastitis this is being used.
So just say both several times we have seen most of the doctor prescribe with only the such type of the very small symptoms they prescribe the heavy antibiotic. They should be
सर उसी के लिए रेगुलेशंस की बात कर रहे हैं, और इनफैक्ट अगर हमें जब जरूरत ज़्यादा पड़ेगी, एक लेवल आएगा, अभी शायद इतना हम पॉलिसी मेकर्स नहीं समझ पा रहे हैं, पर हो सकता है 10 साल के बाद आए, जब हम आपसे ये पूछें कि आपने ये एंटीबायोटिक प्रिस्क्राइब की है तो क्यों किया है, राइट?
सो लोग कर रहे हैं, अभी हम प्रिस्क्रिप्शन की बात करके H1 को फॉलो कराने की बात कर रहे हैं, अभी H1 ही फॉलो नहीं हो रहा है। सो जब H1 फॉलो होने लगेगा कि प्रिस्क्रिप्शन को हम रिकॉर्ड में लेके आने लगेंगे, शायद ये भी सवाल आएगा कि प्रिस्क्रिप्शन लिखा क्यों है, रेस्पॉन्सिबिलिटी फिक्स हो। सो लंबा टाइम है, बट यस डेफिनेटली वी हैव टू गो टूवर्ड्स दैट। जब हम बिल्कुल क्रिटिकल स्टेज पे पहुंचेंगे तो शायद ये वाली भी बात करेंगे। Thank you, Doctor. Thank you very much, sir. Wonderful explanation on the effects of herbs and other things, and then the principle of Ayurveda, which is focusing mainly on health rather than diseases. So, with this, I would like to thank Dr. Anup Kalra, and would like to invite the last speaker for this session, Dr. Anu Rahal, to talk on ethnopharmacological interventions to overcome AMR. डॉक्टर राल प्लीज। एंड आपको 2 बजे ना लंच में सबको भूख लगी है, चले जाना है, हम उठ के चले जाएंगे, है ना। आपको 2 बजे तक खत्म करना हो खत्म कर लेना, नहीं तो सब उठ के चले जाएंगे बस। फाइनल। थैंक यू मैडम। मर्जी आपकी, 2 बजे का टाइम है।
Respected Chairman, Co-Chairman, dignitaries, and the audience, good afternoon. The topic of my talk is the ethnopharmacological interventions to overcome the antimicrobial resistance. By definition, you can just say that AMR is the ability to withstand the lethal doses, the doses which had been previously effective, but now they are no longer effective because either there is either a surface modification in the bacteria or the metabolic pathway which the enzyme with the drug was using has been altered. WHO has already taken the initiatives, and it has launched GLASS and has undergone a massive surveillance, and then it has also gone for a joint initiative for a public-private partnership, and has aimed to develop and deliver the 5 new treatments for these drug-resistant bacterial strains. So the final target is a drug. So how does the drug act in the body? The drug, once it is administered, it gets absorbed passively and it's distributed to the different tissues, and as it gets distributed, it is also metabolized and excreted from the body. To exert a pharmacological effect, it must be reached the site of action in at a certain threshold level, and then only it can lead to a clinical response. So once the drug and the bacteria are plain, the drug has to enter the bacteria by a passive diffusion, and once it enters the cell, the bacteria is going to actively exclude it. These two factors will play a major role in deciding the intrabacterial concentration of the drug, and the killing is both time and concentration dependent. So if the bacterial efflux is higher, then it will lead to the decrease in the intracellular concentrations, and there will be a therapeutic failure. And so it will also give an increased chance for the selection of the resistant strains. So if the activity of the bacterial efflux pumps can be reduced, the drug concentrations can be maintained, and the treatment can be effective. These efflux pumps are actually the plasma membrane proteins, and which actively exclude any xenobiotic that is entering the cell, and overexpression of these efflux pumps has been already regarded as the hallmark for the failure of the treatment, and they also act synergistically with the other resistance mechanisms. In the complete eukaryotic family, there are 2 main families for this efflux pumps, the ABC family and the major facilitator family. The ABC family is further subdivided into 7 subfamilies, of which 3 are of the drug resistance importance, And the most important is the versatile molecule is the P glycoprotein. And it has been encoded by the MDR1 gene, the multi-drug resistance gene. Hypothetically, to make the treatment effective, we can either inhibit the pump, we can downregulate it, or we can modify the drug molecule so that it is no longer a substrate for the pump. Modification of drug molecule, as already discussed, is a unfavorable task. So reducing the efflux through a potent efflux pump inhibitor or a moderator seems to be the best option. Now, how does this overexpression takes place clinically? It can be induced by the antibiotic itself or by the host producing molecules or even by unrelated molecules like disinfectants and the household chemicals. To what magnitude does the uh, pump modulation affects? You can appreciate that induction of the efflux pumps can lead to a 1000-fold decrease in the concentration of the antibiotic. And the inhibition can lead to a 2-fold increase in the concentration. So if there is 1000-fold decrease in the intracellular concentration of a drug, then no treatment can ever be expected. On the other hand, if we can increase the concentration, we can double it for making the treatment effective. So the way forward can be, we can selectively inhibit the host, the pathogenic efflux pumps, so that there are greater intracellular concentrations of the drug in the bacteria, and this will lead to a better pathogen killing and will reduce the chances of development of drug resistance. And on the other hand, we can go for a selective activation of the host pumps so that the concentration of the drug in the host cells is reduced, and so the therapeutic index of the drug is improved. There's a huge list for these efflux pump inhibitors, but none of them is till date clinically available, but it can certainly affect the efficiency of the available antibiotics. The best example to is the NSAIDs. We all know that NSAIDs are always used along with antibiotics as a supportive treatment, and they're actually moderating the antibiotic concentration, they are prolonging its effect, and that is because it leads to an increase in the intracellular concentrations. Several natural alkaloids are also reported to show similar effects. Now, how can the efflux pumps inhibiting activity be done? Either we can go for the inhibition of the energy sources as they are requiring the protons, or we can go for the altering the membrane potential of the bacteria or the generation of ATP. The membrane potential alterations and the ATP generation, these two will also target the host cell. So they are of no clinical use, but we have several natural compounds which are proton scavengers, like the betulinic acid or ursotic acid. So they can certainly be used as proton pump inhibitors. As far as the bacterial killing is considered, it can be either cidal or static. The cidal is just producing the membrane disruptions, so they are ultimately making the cell more vulnerable to lysis, while the static mechanism is weakening the bacterial defenses and it is allowing the host defense to overpower the bacteria. And both the mechanisms are being affected by the quorum sensing, a system of the bacteria in which it tries to evade any hostile conditions present in the surroundings. So once the bacteria is phagocytized, it experiences a lack of oxygen, nutrition deficiency, and a respiratory burst. So to just avoid damage, it goes to a dormant stage, or it leads to other mechanism. Once it enters the dormant stage, the most of the regular enzymes will downregulate, and the other pathways like glycoxylate shunts will be active. So if we target the alternative pathways, then certainly the static mechanism can be more potentiated. As far as the herbals are concerned, the side effect can be either direct, it can directly affect the membrane, or it can lyse the cell wall, or it can interact with the genetic material, and it can even chelate the cofactors with enzymes. And it can also have the indirect effect to moderate the efficiency of the present resistant antibiotics, like the penicillin's inhibition by epigalactocatechin, which increases the sensitivity of the drug in MRSA. Similarly, we can, for the static also, we can go for the translational pathways and the disruption of metabolic enzymes, and ultimately we can use the herbals to weaken the bacterial defense. At CIRG, at my institute, we have been using this for the development of polyherbal formulation against Haemonchus contortus, and we have targeted the two vital enzymes for this, the glutamate dehydrogenase and the tubulin protein. And through in silico studies, we had identified 5 phytomolecules, which were showing a better efficiency than albendazole, the routine dewormer. And then we conducted the synergistic studies, and we backtraced the extracts, and then we could develop 2 formulations which could maintain clean animals for 5 months. One of this product is already available in the market as Vermolex, and the other is under modification. In bacterial line, we are working on Brucella melitensis, and as the Brucella enters the dormant stage, it activates the glycoxylate cycle for energy production. So we have targeted the isocitrate lyase and the malate synthase enzymes, the 2 consecutive enzymes of the glycoxylate pathway, and we have developed a crude combination. And in December 2018, we went for the field screening, and we found that it could decrease the bacterial load for about 2 months, and we are further modifying it. But these animals which we treated, when we tested it in 2020 breeding season, all the animals were found to be clean. None of our treated animals have till date reported Brucella positive again. Sir, goats. So till date we have 67 goats treated. To conclude, the efflux pumps modulators can be used as an effective mechanism for controlling the AMR. The plant derived molecules can play a critical role in the development of effective therapeutics, either alone or in combination with conventional antibiotics. And the reverse pharmacology needs to be considered to characterize the biomolecules along with their mechanism to circumvent AMR and control infectious disease. Thank you. Sir, I finished well off in time.
थैंक यू डॉक्टर साहब. एक तो निकल के भाग गए पहले ही, मैं पकड़ नहीं पाया, पीछे थे मेरे. पर thank you very much for like finishing the things in time, and in fact conveying a wonderful message of how herbal preparations can be scientifically used rather than simple observations on some of those things. So this is the kind of science I was talking about in the morning also. So if anybody has got any questions or comments on that, कोई online comment है? So wonderful. Either it is so clear that nobody wants to question anything, or it is so confusing that it goes above the heads. Thank you very much. Thank you, Dr. Anu. Thank you very much. And it's a wonderful session. So before I close,
Thank you, sir.
I would like to request my co-chair, Dr. Anup Kalra, to say something in case he wants to say something on this session.
It is close to lunchtime. I would not take much time. The only point, which I wish to state and share is that we have a lot of such conferences, whether they are here or in Delhi or world over. There's plethora of material which is available in terms of knowledge, and they're all very keen to participate, deliberate, and share. But then, what is most important here is that out of this, what are those deliverables and how do we make them actually happen on the ground is important, and which is what I think the bureaucrats and the policy planners are now involved into the entire thing. I'm sure we have a bright future, not only related to AMR, but all such initiatives which the academia is taking, where the researchers are taking, that we should be able to reach to some positive conclusions. For example, one of such thoughts which was shared is that we have deficiency of resource and the funds in AMR. So I think that is one way. We must definitely plan some time-bound actions to work towards an actionable goals. These are some of my thoughts.
Thank you, Dr. Sab. Dr. Sab, before your turn, I would like to request my reporters, in case they want to say something, Dr. Harshit and Shalja.
Sir, it's a quite wonderful session, and what the things is related, like we have a deficiency between regarding the data collection and surveillance is most important thing, and we really need some laboratories for the surveillance and the data collection, because whenever we applying for the project, so the question is raised whether the data is authentic or not. So we need some scientific collaboration, and the paper should be published and it should be collected, and the repositories are required, whatever this. So sir, from this session, the main thing what we data generation is required. Some of the platform should be prepared to take the data initiative, and the surveillance is required, because in Meerut, some bacteria is become resistant, while in some other like in Noida, the other bacteria is become resistant. The prevalence of the bacteria is required, sir. Whatever the prevalence of the bacteria is scenario nowadays by the sir, this city wise. So I think that is sufficient for this, sir. Okay, thank you, sir.
अच्छा, आप कुछ कहना चाहेंगे सर? आप कुछ कहना चाहेंगे सर?
The four lectures were there, and I thought the main emphasis of this particular session, emphasis on this particular session was to have the data, as the Dr. Harshit said, data collection of all the antimicrobials used and misused. Whatever the doses is being given has to be recorded well. And alternatively, we have to look for the other approaches apart from the antibiotics, because we cannot rely only on the antibiotics. As I heard Dr. Nukala, who was just showing one slide, that industries are not looking for new antibiotics because they are not looking it beneficial for them. So obviously we have to look for alternative approaches. I think this is the key for this particular session. Thank you. Thanks all.
Thank you, Dr. Akshay. And before I close the session, I would like to request Dr. Manji Saxena to say.
Well, thank you, Doctor. There are 2 points I want to make. That normally we look into the pathogen and try to understand what pathogen does to our body, and that is the right approach also. The other way is how pathogen establish the relationship with the host and proliferate to create the clinical response. What we see is in the form of the symptoms. Now, the point what I am trying to make, the maximum research which is going on the herbals world over is how do they influence intestinal mucosal barrier for enteric diseases? How do they influence the bronchial linings of our trachea and also the lungs tissue? Because ultimately, the clotting factor which is produced by this virus is by a protein. And then there was a research carried out that how ashwagandha is going to neutralize that very enzyme which result into the fibrosis, that is fibrin production in the lungs. And US universities have been ahead of time. By the time vaccines got developed and everything, US started using ashwagandha not just as a supplement, but they started cultivation of ashwagandha also in their state of Arizona. Now, same thing when it comes to whether it is metritis. Again, uterine mucosal lining is there. Now we ignore, in fact, what happened, we as a pharmacist, we always ignored the small molecule. Salmonella control has been through the use of mannose sugar, where it will not kill the salmonella. But salmonella will get hooked to the mannose molecule. That is the antigen which is there on the salmonella surface, and without producing any pathogenicity, it will be passed through the feces. So what I'm trying to say, she has done a very good work. In fact, we would like to collaborate on that side. Look, besides the parasite pathogen, host also needs to be looked into, and that is where this microbiomics comes into the picture. What you said, sir, the good bacteria get destroyed. So this is perhaps going to be one of the very interesting direction. And the other mechanism, which just 1 minute, sir, ultimately bacteria's pathogenic, they develop resistance by memory recognition. So the moment they see there is a molecule single, they start producing RNA and DNA and then protect themselves from that antibiotic. At least you all can basically search Google and find out whether neem has ever got resistant against any of the pathogens. You know, now why it is? Because the neem has got more than 200 chemical molecules. And therefore, for bacteria, the mechanism it can do only for single molecule. Lot of people basically question, why don't you have a single herbs? Here the combination of the herbs is to potentiate that action, to minimize the side effects, and to enhance the optimum usage. And that is why you find 4, 5, like we take in our food, not just the fat. We take unsaturated, saturated, then we take carbohydrate, simple sugar, polysaccharide sugar. Same way, ultimately, we take protein, not just for the protein, but to meet out our requirement of amino acid. So it's a whole complex system where we have to basically see that how we can minimize these pathogens also, rather than to killing is not just the only approach. This is what's up.
Thank you very much, sir. Wonderful points. So just to conclude, it's a wonderful session, and where people have been talking about
international policies, the national perspective from the international agencies, the alternatives and the science behind those alternatives. It's really a wonderful session, and as rightly pointed out by Dr. Kalra and Dr. Manji Saxena, we are talking not only about microbes, we are talking about not the disease, we are talking about health, and health means we have to talk about the complete picture of host pathogen interaction, including the environment. So with these words, I would like to thank the speakers for their wonderful deliberations, and organizers for arranging this wonderful seminar. And I hope it's already three minutes over the time, and I am speaking for those 3 minutes. Thank you very much.
Thank you, sir. Before we end the session, uh, I request Chairman of the session and Honorable Vice Chancellor Sobit University to kindly honor the speakers who are present here. So, please, sir, uh, Chairman of the session, Dr. Malik, sir, and Dr. Garg, sir. Uh, doc, First, I would request to honor by presenting a memento to Dr. Anuj Sharma, WHO consultant. Now to Dr. Anup Kalra, Director Corporate Affairs, Dabur Ayurved. Thank you, sir. Now I request to honor Dr. Anu Rahal, Principal Scientist, ICR, CIRG Magdum. Thank you, sir. Thank you, Chairman, sir. Thank you, Professor Garg, sir, for honoring the speakers. I thank one and all. Now we'll proceed for lunch and we'll assemble at 2:30 sharp for the next session.
And now, without wasting much time, I think that we should start with the session. We are already behind the schedule. I think we are 1 hour back, late or something like that.
Around 15 minutes. We are seeing online 18 lectures.
All three are online? All three are online lectures?
All three are.
Yeah. Okay, so I have been told that all 3 lectures, these are online. So we will start. I think that the speakers have joined. Yes. Okay. Fine. So the first lecture, it is by Dr. B.R. Singh, Head and Principal Scientist, Division of Epidemiology, IVRI, Bareilly, from Bareilly, I think. Yes. Unusual causes of emergence of antimicrobial resistance. This is a nice session, and I invite now Dr. B.R. Singh to start his talk. Dr. B.R. Singh, please.
So thank you, Chairman sir, and organizers for giving me this opportunity. Sir, can you hear me properly?
हल्की सी
Audible,
sound.
sir? Am I audible?
Yes, now audible.
Yeah. So thank you for organizers for giving the opportunity, and thank you, Chairman and co-chairman and reporter and all the people who are attending the lecture. And this is unusual in the sense that most of the time we believe that emergence of antimicrobial resistance is due to veterinarians and due to medicos and to some extent due to non-concerned Indians who go and buy the antibiotics on the counter and take it. So my views are a bit different, and I want to share with you here. So 1 in 10 people hospitalized worldwide, they die due to failure of antibiotics, failure of treatment. And failure of antibiotic means there is antimicrobial resistance, and the drugs which are given, either they are not working due to antimicrobial resistance or they are not suitable to be used. And this the problem is much worse in developing countries or middle income countries or the low income countries. So around 40,000 infants died due to sepsis, and most of this sepsis is due to the infections which do not respond to the antibiotics. And we can see news in so many medias that antibiotic resistance is increasing day by day, and superbugs surging, harbored even on doctors' hands. So these type of news scare us, and they induce us to think that what is happening to the world. Worldwide, each year 7 lakh people die due to antimicrobial resistant infections. And it is estimated that by 2050, 10 million people, that is 100 lakhs or the 1 crore people will be dying every year due to antimicrobial resistance. And of these, more will be in India than in any other country. So because we are more in number, and that's why we will more of Indians will be dying. 10 million deaths attributable to AMR every year, and 2 million will be in India. So this is very serious state, specifically for we Indians. Why no or rarely new antibiotic is introduced in last 1 decade? There are several reasons. Antimicrobial drug development and then its release to the market that cost heavily. And The bigger reason is that by the high cost, the antibiotic is released. That antibiotic is not having good life. And very rapidly there are reports of emergence of resistance to almost every new antibiotic. And that's why since last many years there is no entry of the new antibiotics in the market. And you can see it is decreasing year by year. And so people say that it is global antimicrobial consumption, which is increasing, and India is the largest consumer of antibiotics. But people forget that we are the one of the largest population also. So when population is more, certainly we have to use more doses. But there is population also in China. Why in China it is not increasing? So one of the statement of our learned Prime Minister is that we are having money there and we can buy the antibiotics. That's why we are using more antibiotics. But that is not justifiable. And here comes the misuse of antibiotics by we Indians. So we are misusing the antibiotics. We can go without prescription to any pharmaceutical shop, and we can purchase any antibiotic of our choice, and we can eat either a day or two, or we can complete the course or we cannot complete the course, and we can take it at our wish. And that is the reason, instead of becoming rich, I think that this is the major reason that we are having the freedom to buy any antibiotic on counter without any prescription. So antimicrobial uses in human versus livestock. India is the biggest consumer of antibiotics of human use, but it is not the case with the animals. In livestock, India is at the, you can see that India is at the 5th place in antibiotic consumption, though we have the largest number of livestock heads in India. That means veterinarians, who are usually taken as the culprit of using antibiotic indiscriminately, they are not the real culprit. Real culprits are some other people who are responsible for emergence of drug resistance, not the veterinarians or not the livestock owners. You can see the India's place is much behind, and even after end of this decade, India will be much behind in use of antibiotics in livestock and livestock industry. People say that in animals, usually antibiotics are used for mass treatment. One animal is sick, the antibiotic is given to all. It is not the case. Indian farmers or Indian livestock owners are not so rich who can afford. It may be case of use of such type of medication in some of the organized farms, but most of the livestock in India are not in organized farm. They are in houses, they are in backyard of farmers, and they cannot afford mass treatment of their animals, and they are not able even to buy the required amount of antibiotic. That's why so many animals die without the proper antibiotic treatment. But being weak at many fronts, veterinarians are taken as the soft, we can say, the soft target by politicians, by medicos, by administrators. They all come to blame the veterinarians. And you can see that in India, a total of 1,500 tons of antibiotics are used, while in human being we are using 2,000 tons every year. And you can see in other countries also, Asia is the biggest user after Europe, and total Europe population is almost equal to India. So total Europe is using more antibiotics than India. So we are blaming India, we are blaming veterinarians without giving any consideration to the real facts and figures. So why the antibiotics are so important? Why people think of the antibiotics? Particularly, I am talking about the administrators and politicians, those who take more care of the pharmaceuticals rather than the livestock owners and people. Antibiotic constitute 12% of the Indian pharma market. India's antibiotic market is 16,000 crore as on today, and 30 to 40% of these are injectables. The pharma sector contributed about 1.72% of the GDP, but they forget that almost 3 times is contributed by the livestock sector, for which we hardly think of, but we often think of the pharma sector. And agriculture GDP, more than 1/4 of GDP is contributed by animals only. So if veterinarians are trying to treat the animals with suitable antibiotic, then what are the options with the veterinarians? They have no facility to get tested the samples of animals. And the livestock owners do not hold the capacity to send their animals tested for antimicrobial sensitivity test. Even they are not in position to afford that cost of antibiotic sensitivity testing for themselves also. Means when it is not available for humans, how we can think of the animals? And we are always ready to blame the veterinarians. I am very much against this notion that veterinarians are playing any role in emergence of drug resistance. Popular belief that says that misuse and overuse of antibiotics by human and routine use of antibiotics to grow faster and faster the food animals are the key causes. It has been claimed by OIE and WHO that this is the major cause. Certainly, we are in position to misuse because many a time we purchase the antibiotic without prescription and we use, but we are not in position to overuse. So only one aspect applies on India. Exposure of bacteria to antimicrobials. Antibiotic exposure of bacteria in irrational, inappropriate subtherapeutic doses. That is more important for emergence of the drug resistance. But what are the reasons of these inappropriate or suboptimal concentration of the antibiotic use? Human consumption of therapeutic and preventive purposes. Lot of the antibiotics which human are consuming, they are coming in the excretions, and in excretions are going without any treatment to sewage, and sewage is again causing the spread of the antibiotic resistance. So new dimensions in emergence of AMR, what are those? Overuse and misuse of antibiotics have long been believed to fuel antimicrobial resistance, but new research has shown that simply lowering consumption is not enough. Poor sanitation means without releasing sewage without any treatment. Corruption and low public health spending have bigger role of pushing up drug-resistant infections in low and middle income countries, including India. And this is not my statement, but this is of WHO statement, the recent statement. Means it is poor sanitation, corruption. Where is the corruption? That is allowing the non-usable drug to be sold in market, allowing drugs to be sold without any prescription. There are so many things. And low public health spending. Many of the countries, they spend 10 times or 100 times more than India on health. But we are not spending that much, and we are blaming our people that they are not hygienic, they are not good people, they are responsible for emergence of drug resistance. And it is not only India, it is everywhere in middle income countries. Sanitizers, use of sanitizers. Though most of the sanitizer claim that 99.99% they kill the microbes, but Ontario research has shown that just 46 to 60% of the bacteria are killed with the hand sanitizers when applied on the hand. So what are those rest? More than 40% bacteria. They are resistant. They become resistant not only to sanitizers but to many of the antibiotics, because antimicrobial resistance pattern and antimicrobial resistance um, capability is not specific to any antibiotic. Many a time certain genes are activated and they confer resistance to a broad spectrum of antibiotics, particularly those genes regulate the efflux pump regulators. Load of hand sanitizer in India are of loads of sanitizers. Last year only 20 batches of hand sanitizers have been caught substandard or of non-standard quality. And you can imagine that only 1% of the samples, only 1% of the batches are sampled. Means it may be 100 times more. That is 2000 batches per year might be there which are substandard in the market. The antiseptics and disinfectant has 10% povidone iodine, 4% chlorhexidine, 4.8% chloroxinol, Dettol, Lysol, all these, they also contribute to the emergence of antimicrobial resistance due to selection of the resistant microbes or due to induction of the efflux pump regulators which make them resistant to many of the antibiotics. Biocides, exposure to low concentration of variety of biocides and dyes has shown to be resulting into induction of the efflux pump activity and resulted into increased drug resistance. Triclosan induces heritable drug resistance. It has been shown in E. coli. The oxidative stress induced by triclosan caused mutation in so many genes. Mutations lead to resistance by upgradation of beta-lactamase and multi-drug efflux pumps. Chlorhexidine has induced vancomycin resistance. In Enterococci, chlorinated phenols usually induce this efflux pump genes, MAX-AB and OPR. These are the antibiotic resistant phenotype mediating efflux pump. So, use of non-antibiotic antimicrobials, which are not for the consumption or not for the treatment, but they are usually used for disinfection and as the agent of our better hygiene. Biocide disinfectant and antiseptics, non-antibiotic antimicrobial chemicals are used in much larger quantities than antibiotics, resulting in high residual level of these chemicals in wider environment, in soil, in water, in rivers. For example, triclosan, a common biocidal agent used in over 2000 kind of products such as toothpaste, hand washing, and certainly it is not in the amount which should kill the bacteria. It is just added due to one or other reason to show that yes, we are using antimicrobial. And if you are using any biocide in concentration lower than the required, that lead to the emergence of drug resistance. Consumption of liquid disinfectant in 2008 in India was more than 7,000 tons, which share with a share of Dettol, chloroxinol, Savlon, and Suthol, so many others. In year 2000, worldwide production of chloroxinol was, you can imagine that it was 30,000 tons. So if this much amount is going to the water, going to the soil, when we use the disinfectant, we wash our hand, we use this hand sanitizers, later on we wash our hand, it goes into the water, it goes into the sewage, it goes into the river, ponds, and all get sublethal doses of antimicrobials, which lead to the emergence of antimicrobial drug resistance. Now I come to the important point, that is role of government and regulators in emergence and spread of AMR. Subtherapeutic exposure of microbe to antimicrobials comes from foul play of administrators and pharmaceutical companies in India in much more amount than it is usually come through the real use of antibiotics. Misuse of antibiotic as preservative, use of enrofloxacin has been allowed to be added into the FMD vaccine in India, and enrofloxacin or any floxacin group antibiotics is not permitted internationally to be used in animals even in therapeutic doses, but we are using in subtherapeutic doses in as a preservative in FMD vaccine. So where are the administration, where is the honesty and where is the transparency? Therapeutic use of substandard and fake antibiotics in therapy. I will be showing the graph how it is increasing every year, the substandard and fake antibiotics in the market. They are coming because this government officials, they are allowing it to go to the market. In China, when Jinping came to the power, China was the top most fake and substandard drug producer. But Jinping realized that, and in a month, he hanged more than 3,000 drug controllers who were giving certification to the substandard and fake antimicrobials at that time. And since that, China is no more at number one. India is now number one in production of the fake antibiotics or substandard antibiotics. Therapeutic use of non-approved antibiotics in the therapy, it is rampant in India. Why it is rampant? Why the our administration is not looking into that? Emergence of AMR due to pharma companies. It is very important as far as this emergence of drug resistance is concerned, because contamination of environment, soil, water and air, pharmaceutical waste is coming from antibiotic producing pharmaceuticals, polluting water bodies and soil to make natural resources the breeding ground of AMR bacteria. And there are several reports. Indian and Chinese drug makers routinely release untreated waste into the natural waters or surrounding soil. They dump. If you go through Gajraula on the way to Delhi from here, from Bareilly, you will smell that Gajraula is coming. So many antimicrobials and so many nasty chemicals are dumped into soil. The water around Gajraula in range of some 20 kilometers is not drinkable. And we are in name of supporting the pharmaceutical companies, in supporting the new and emerging industries, we are relaxing them on front of the environmental pollution. And even if we are not relaxing, our officers, they are minting money for allowing them to release the waste without any treatment. Reducing antibiotic-laden pollution from pharmaceutical manufacturing plant is relatively easy, affordable part of the solution. However, in India, releasing all noxious wastage into Ganges, its tributaries and other sacred river is much more easier and cheaper through using bribes. So pharma pollution, shut the back door on superbugs. If we can stop, we can shut the door for entry of the superbugs, but we are not doing that. So pharma pollutions, drug resistance through back door. India coming from back door, because we are allowing these companies to do the back door management of their polluting sewage or polluting effluent. India has become hot spot of antimicrobial resistance, largely due to contamination of natural resources through the disposal of untreated waste, not only of pharmaceuticals, other industries. How big pharma industrial waste is fueling in the rise of superbug worldwide. It is a known fact and I need not to comment. Occurrence and abundance of antibiotics and resistance gene in rivers, canals and near drug formulation facilities is rampant. In one of the study conducted here on wildlife of Gir forest and in Tirupati jungles, we found that superbugs are more common in wild animals dying due to one or other kind of infection. Why? No, who veterinarian is going to give them antibiotic there? It is the antimicrobial resistance which has emerged or microbes has emerged in river going through Gir forest and other forests which are contaminated by pharmaceutical companies and untreated sewage. So pharma effluent is one of the most important cause of emergence of the drug resistance. And you can see how pharma companies in India are enjoying the freedom to dispose of their waste without any restriction. And this figure shows about the largest of the some of the large generic and we can say the proprietary companies which are releasing so much without any observation by government or without any external waste treatment management plant or without any restriction they are doing. So Indian drug companies, when this year in January, last year in January, India government drafted a bill. Bill is still bill, and it was trying to, on recommendation of several players who are concerned with the emergence of antimicrobial drug resistance, they tried to put some restriction and they suggested that 121 common antibiotics, they should be the companies, those are producing those, that should be discharged into river and surrounding environment with effective treatment. And the Indian Drug Manufacturers Association has lobbied both the Ministry of Environment, Forest and Climate Change and Central Pollution Control Board. And they wrote letters and they lobbied and they stopped the nation to bring such any kind of restriction which can prohibit them from contaminating our environment, which is leading to emergence of drug resistance. If we will be sick, we will be consuming more medicines, not only of antibiotic type, of other types. And that means we will be more dependent on pharmaceuticals, how pharmaceuticals can bear the fear that people of India will be healthy in future if it is done. So there are several problems. It is a multi-billion-dollar business of substandard and spurious antibiotics in India. And the leaders of this spurious antibiotic and substandard antibiotics are nowhere but in Delhi. Delhi is the lead place. So estimated business of antibiotic in India in 2009 was 64 billion. And in 2016, India consumed antibiotics of more than 150 billion. And you can think that in India, as per the CDSCO estimate, about 10% drugs, antibiotics are fake. Means at least 50 billion rupees we are paying for the fake antibiotics. So you see how government is promoting the that people should fall sick and people should consume the fake or we can say wrong antibiotics. These fixed dose combinations, they are prohibited in many of the countries. But in India, we are allowing those. So not approved fixed dose formulations, they are 64% in Indian market. So you can believe that 64% antibiotics which we are consuming, they are not to be used in the way we are using. But these are allowed. Nobody is taking action against these nasty peoples. So you can see here that how the market of hate drug is increasing year by year. So this is the uh, graph made from the data extracted from CDSCO website. They every month they publish that how many batches they have caught substandard or of not of standard quality. They do not say it's substandard, they say not of standard quality. So I do not understand that what is the use of this type of nomenclature. Rather than directly saying substandard, they use not of standard quality. So in 2013, 41 batches were caught substandard. And in 2020, 97 batches. And in 2013, 31 pharmaceuticals produced these substandard antibiotics. But in 2020 and 19, they are nearly 100. So they have increased 3 times over last 7 years. And where we are going, this is the trend line you can see, we are going in an era where we will be having more substandard antibiotics. And as per CDSCO, they sample only 1% of the batches in the market. That means these may be 9,700 batches, which would have been substandard in 2020 if all would have been sampled. And to my surprise, when I inquired from CDSCO in an through an RTI, they said that they do not sample any product of the big pharmaceuticals. I asked them by name of the big pharmaceuticals that how many batches they have sampled in last 10 years, and their reply was astonishing that they have not sampled even a single batch of big pharmaceuticals. And that means big pharmaceuticals, they are playing major role and as they have the immunity, they are not sampled. They are playing with the administration at much higher level. I don't know
Can you please conclude in
how they are managing it, but they are certainly managing. And these antibiotics which have been caught, these are small players only. Large players have not done it. And you can see in veterinary also, this is not only of humans. In veterinary also, you can see how many, there are much more number every year, which are the batches caught substandard. So these are even almost, I can say 50 or 60 times than in human beings. Because when we are not ready to care the animals, human beings, who has to care about the animals? And veterinarians usually do not raise the voice about the substandard, either their vaccines or the drugs, because of one or other reason I cannot explain here. But about 5% of the total medicine samples intended for use in livestock were detected NSP or fake over last 10 years in India. And every year their number is increasing. So with this, I thank you, the audience, for their patience hearing, and I thank you organizers, and thanks to ICR for providing me opportunity to express my views. So thank you one and all, and your queries are requested.
Thank you, Dr. Singh. Any questions, please? Very interesting talk. I will conclude all the 3 lectures at the last, so that we give enough time to the speakers first. So any questions, quick questions? No? Just one query, Dr. Singh. You
Yes,
have
sir.
given a wonderful lecture, and this is the, in fact, the pollution which is being created deliberately or intentionally or due to economic reasons. The government is also making the legislations and the controls, but these are not working effectively. So why we are not raising this issue with at the government level to effectively control this emission problem? Because this is not only the pharmaceutical industry, but the textile industry, leather industry, chemical industry, and organic chemicals industry. These all are contributing a heavy load of the pollution on the soil, which is disturbing the natural microbial populations, including the resistance in the microbes.
So, sir, why we are not raising the voice?
Yes,
I will
why
give
we are
you
not raising the
example
voice?
why we are not raising
That
the
is the
voice.
question, why we are not raising the voice.
Yeah, I will try to answer that, why we are not raising the voice. In 2014, you might be remembering when I raised the voice that FMD vaccine in the market, they are substandard. And government not supported me, that supported the pharmaceutical companies. My department not supported me, and OIE supported me, and all international organizations supported me. That's why they were not able to hang me. Otherwise, preparations were there to hang me. And I am still fighting a case of 122 defamation case by leading pharmas who were producing that substandard vaccine. So everyone cannot afford to fight to such an extent. Everyone need to raise his or her kids. And that's why keep silent. Nowadays, it is very difficult to raise the voice against this corruption, which is rampant everywhere. Which is rampant even in giving the license to these pharmaceuticals for releasing the waste into the environment. And as you told that so many different pharmaceuticals they are releasing one or other antimicrobials, dyes, they are also antimicrobials. They all lead to increase in the antimicrobial drug resistance, and that is a good market. That is creating good market for future. More the drug resistance will be, more people will be falling sick, more will be consuming the drugs, and bigger will be the pharmaceutical market.
I would like to make one comment for the all audience. We all are eating all foods, and all the food in India, they are marked as used well before this date, and there is no expiry date. From this platform, I would like to invite the attention of the FSSAI that why we are not having this system that the all food materials should also have the expiry date. In all the drugs, in all the things we are having the expiry date, but not in the food material what we are eating every day, even the bread which we consume. And after that particular date, not only in the taste, but it is dangerous also. It is having the contamination also. And similarly, the drugs, Dr. Singh has rightly pointed out that it is very difficult to raise the voice, and even if we raise the voice, then who will listen to it? But my opinion is that the universities should take a lead role, at least in putting their voice in the ears of the politicians or the policymakers. Because unless we protect our environment, we are at a very dangerous stage, very dangerous stage. So thank you, Dr. Singh. Now I would like to invite the next speaker. Next speaker, Dr. Ajit Singh, is the scientist emeritus from Hisar, and he will be speaking on resistomes informed combating of antibiotic resistance in the frame of One Health. Very important topic, very important. And please, please focus on the. So, Dr. Singh, you are having the 30 minutes
okay.
Your lecture, and then followed by 5 minutes discussion. Okay. So it will be better that if you stick to the schedule. Thank you very much.
Okay. Am I audible and visible?
Slightly more volume. Slightly more.
Okay, okay. Okay. Thank you very much for inviting me to speak on this topic, Mr. Chairman and your team. So I was asked to speak on something related to AMR, which is so important and everybody is discussing this. I don't know whether much actions we have taken to translate and to combat AMR. Still today, it is a problem which is challenging the globe. So the first thing, the topic that I chose was registomes informed there is some disturbance. Informed. I'm sorry, there was some disturbance. Registomes informed combating of antimicrobial resistance in the. I've divided my talk into three sections. So first of all, although in the previous session, it has been talked about in detail that we are facing the pandemic of the antibiotic resistance. So it is a global problem. And if you see just briefly, the history of antibiotics starts, the use of antibiotics starts with in 1940s with the discovery of penicillin and sulfonamides, and then it was golden age during 60s to 80s, but the problem of AMR had begun during the same time. And then enter the 21st century, we are facing the global rise of AMR. In fact, it has spread all over the world, and we are in very much in the pandemic of. And as this slide has also been shown previously, we see that the major problem of AMR, the projections are for 2015, and Asia will be the largest casualty for antibiotic resistant bacteria they will be causing. Now you see the AMR is a problem not only of one pathogen, there are several different pathogens. All of them have some sort of resistance to antibiotics, one to several antibiotics rather. Some of these are having say resistance to all kinds of antibiotics in use, starting from penicillin to cunipristimine and daptomycin. Emergence of resistance has taken. Now, this is also a point to be noted that resistance to different antibiotics arises in different, say, duration, say, after start of the use. Say, streptomycin and cunipristimine, the resistance emerged just within 1 year of their use. And then most of the antibiotics take, say, 3 to 4 years to emerge resistant. And to some of the antibiotics, chloramphenicol and vancomycin, it took more than 10 years to emerge resistance. Now, what we see that new antimicrobial drugs which need to be replaced with the ineffective ones, the pace of emergence has already crossed, and we are not more in a position to, to take care of, say, bacteria with newer antibiotics. And this is just in short AR mechanisms. There are 4 major mechanisms, say efflux, inactivating enzymes, target site alterations, or target amplification, etc. Now, microbes use resistance emergence as evolutionary strategy. But we are also contributing to the emergence of resistance against microbes. Say microbial communities carry various antibiotic resistance genes. AMR emerges by both intrinsic mechanisms and horizontal gene transfer. It emerges under selection pressure of antibiotics, and there are, inadequate disposal systems, poor hygiene sanitation, and poor monitoring surveillance system, all of them contributing to, say, emergence of and spread of AMR. There are, say just in a more detail about the pre-existing evolutionary strategy of the microbial world. That means the antibiotic resistance is a pre-existing strategy that microbes use. And this is genetically determined and results from mutations, transposons, integrons, HGT mechanisms. And interestingly, AMR is slow to revert. And there are a lot of ARGs now known, and databases have been created that contain information for thousands of genes and antibiotic types, hundreds of antibiotic types, and many bacterial species, say thousands of species and hundreds of genera, and different mechanisms of resistance, as I have already talked about in the previous slide. So the emergence and spread of AMR, what is our contribution to emergence and spread of? Dr. Bhojraj Singh, the previous speaker, has talked about many of these, in fact, that relates to, say, dishonest practices or whatever, say, very complex issues. But these issues, say, inappropriate use of antibiotics in health and agriculture, biocides and toxic metal pollutants in soil, wastewater, etc., poor disposal system, poor hygiene, sanitary practices, biosecurity lapses in animal production systems, poor monitoring and surveillance, knowledge gaps, and inadequate intersectoral cooperation for One Health, lack of incentives for research on new antibiotics and alternative approaches also has. So let us recapitulate some interesting facts about AR emergence and spread. Bacteria are ubiquitous and live in communities of mixed species. This is a fact that, and another fact is that bacterial penicillin A was identified much before the use of antibiotics. That means they are pre-existing. Antibiotic resistance genes are pre-existing, and a large number of ARGs exist in natural microbial populations. And which came first, the antibiotic or the resistance? In fact, resistance originated in antibiotic producing bacteria, first of all, 3 billion years ago, according to, some studies that they have estimated that they are already existing. Conjugative plasmids transfer ARGs between pathogens and environmental bacteria. This is also important. It's not exclusively a problem related to pathogens alone. There is exchange of antibiotic resistance genes among pathogens, commensals, and environmental bacteria. And these plasmids were discovered in Japan during mid-50s, so very early, soon after, say, use of penicillin started and, say, streptomycin, etc., came later. They were found that they can be transferred. Resistance can be disseminated in microbial communities by HGT mechanisms. Another point is antibiotics cause selection pressure for emergence of AR bacteria. So the presence of antibiotics, the use of antibiotics enhances or the emergence of and then spread of AR bacteria. Clinically relevant ARGs are found in pristine environments with no human activity. That shows that, in fact, they are pre-existing antimicrobial resistance genes in environments like in the northern hemisphere, say Alaska region, or where no human activity has been noticed, the microbes isolated from there and then studied, the ARG genes, which are clinically relevant, were also found in those locations. So ARGs are co-located with, most often they are co-located with resistance genes for biocides and say toxic elements. So they are co-transferred. In fact, they are, so that to a point to be noticed. Say, and then discovery of new antibiotics and waiting for emergence of AR is no longer tenable. So because the speed of antibiotic development has been, say, outpaced by the emergence of AR. So something needs to be done and serious steps have to be taken now, and alternatives have to be found. Then the pace of emergence and spread can be slowed. But seeing all this, there is one fact that even if we have no control over the antibiotic resistance genes which are pre-existing in nature, our actions can be controlled. We can intervene in those, say, activities due to which the speed of emergence has increased. And for that reason, WHO, FAO, OI, they made global action plans, which was talked in detail in the previous session. And then India's action plan has also been talked about in the morning session. And state-wise action plans, although they are, they have been suggested, and they have to act in conformation to the national action plan. Then coming to the smart strategies to combat, and say, when we know that AMR problem is used as a strategy by bacteria in a very smart way, and it's difficult to control, so we need to invent and use such as a smart strategy to combat AMR. And this topic, antibiotic resistome guided pathways and the One Health approach, they are considered the smart strategies that can help combat AMR in a better way, better than the traditional approaches. So what is antibiotic resistome? In fact, resistome, as you see, it is a term derived from the omics branch of sciences, and resistome is the entire number of antibiotic resistance determinants in a microbial community. Now, you know, the microbes are present everywhere in diverse environments. So there are diverse microbiomes in diverse environments. More than 20,000 potential ARGs have been found, and more than 400 different types. And they are predicted from available bacterial genome sequences. The number of functional AR determinants in pathogens is, however, much smaller. Now, see, how do we determine antibiotic resistomes? What are the tools and technologies? There are two major tools, say, sequencing metagenomics and functional metagenomics. Both put together, say, you do metagenome sequencing by NGS technologies to determine total number of ARGs. Then you confirm or validate these ARGs by using functional metagenomics. And then using bioinformatic tools, you annotate and analyze ARGs, and you create databases from which you can store AR genes information, retrieve or catalog. And then using, say, further technologies, the AR diagnostics are being developed, and AR predictions for new antibiotics have been made, say, possible, and resistome guided discovery of antibiotics and alternatives, as these information contained in the database is being used for these purposes. No, just in a diagrammatic way, say there are different environments from which you can expect samples, microbiome samples, and then you extract genomic DNA fragments. From these fragments, there are two approaches can be run parallel, that you create the sequencing libraries, then you sequence the DNA fragments, and this will give, because this is the original DNA taken from many different microbes living in communities in a particular sample, you create a metagenome sequence. And from these sequences, you can then map these sequences to the known resistance genes, and also identify mutations known to cause resistance. So this will give you a picture of resistance potential in a particular sample. And this approach is known as sequence-based metagenomics approach. The other approach is functional metagenomics, in which you create fragments and put them in vectors, transform the E. coli host, and then you create a metagenomics library. From this library, you can screen the transformants for expression of here it is the expression of the resistance genes, which is important. And then these resistance genes can be screened on these Petri plates containing different antibiotics. So, and then you can clone these resistant genes, resistant for erythromycin or vancomycin or other or no antibiotics. Then you can create sequenced clones, and you can know with this approach novel resistance genes also. So both, often these say this is first done and then followed by functional metagenomics. These approaches are used in. Say it is just further that you can, how can you refine using the bioinformatics methodologies to identify antibiotic resistance using a sequence-based metagenomics approach. So there are various bioinformatic tools that are used to have all the resistance genes, say abundance of the genes, you can know the mutations, and in a way you know the resistance potential. And then functional metagenomics, again, say it has been explained, you have a sample, extract DNA, shear to make fragments, put it in vectors, make a library, metagenome library, and then you can select, sequence them, and from these sequences, you can annotate, assemble, and put them in database. So this is how the information about ARGs is being created, and the number of more and more genes are being added to the databases. Again, there is integration of NGS sequencing and screening technologies. Often these two kinds of screening for drug development and resistance surveillance, they are being used by integration of these. So this gives information about risk prediction for new antibiotics, informing combination therapies, then resistance inhibitors, informing resistance reversal, and then resistance surveillance, screening of hotspots, for example, in agriculture, hospitals, waste water, and then identification of new resistance genes, risk assessment and prediction, improvement of bioinformatics and algorithms. So these technologies are being used to know the registomes of different environments, and even say in different hotspots. So just to have a brief overview of diverse environments ARGs, both the terrestrial and aquatic environments contain microbiomes, and they have ARGs in them. Soil has the largest number of ARGs, including ancient ARGs. And then livestock waste and cow dung manure mixes air bacteria with plants, vegetables, and land. So no environment is spared of having antibiotic resistance genes. Water spreads air bacteria, and then urban water cycle, drinking water, sewage, and wastewater treatment plants, they create hotspots of air exchange and spread. Human activities allow mixing of pathogens, commensals, and environmental bacteria. So they add antibiotics, biocide, toxic metals from industrial effluents to allow air bacterial selection. So these are, say, interconnected activities, and the gene flow can be traced using the registome analysis. Now, wastewater treatment, just a glimpse of that, we know that a huge gap between wastewater generation and its treatment is seen in India. And this data I have taken from some study that wastewater by industries, about 60% is treated by industry, whereas the wastewater treatment is only about 30% in, say, class 1 and class 2 towns and cities. The suboptimal performance of existing sewage treatment plants. This is another, say, important thing. Even if we have treatment plants, their performance is suboptimal. And say this data is a bit old, but not much improvement I can expect during these years. Water resource management is going to be the most serious problem of the 21st century, all of you know. And say industrial and municipal segments account for 90% or more of the water treatment market in India. It's a huge market for water treatment. The wastewater treatment capacity in the country, although has increased, if you see the base 78, 79, has increased 2.5 times, but still it is about 10% of the sewage generated is treated effectively. 90% goes into the natural ecosystems, causing large-scale pollution of rivers and groundwater. So this is a serious problem that we are already having. And say just a brief introduction to One Health concept has already been talked about. So there is, this is an intersectoral activity involving almost, say, all spheres of human activity and, say, concerning human health, animal health, environmental health. So they are all related. So what can registomes guide to combat AMR in a better way in the frame of One Health? See, first thing is registomes give far better information about large-scale surveillance of that will point out hotspots of AMR, emergence and spread, in a holistic way, not just as we do with the traditional method. We select only, say, by exogenic cultures, cultivable. These registomes can be, say, studied cultivable and non-cultivable due to metagenomics approach. And so this will better point out the hotspots of AMR emergence and spread, will discover new AR genes, will evaluate efficiency of treatment methods, say sewage treatment or effluent treatment methods. And because of this, this fact, you can perform risk analysis in a better way. And then you can select appropriate waste treatment methods based on the registome analysis. And you can also improve stewardship practices in hospitals, not only medical hospitals. I would suggest that veterinary hospitals should also have stewardship practice if you are really to tackle this AMR issue and in a more systematic way. Then resistome guided pathways to combat say you, in addition, you have a better screen. Resistome is a better screen for predicting AR potential against new antibiotics and lead compounds. There are some interesting investigations in this area. You can also use resistome information to develop alternatives and antibiotic adjuvants. This term, antibiotic adjuvants, are, say, there are certain compounds that help antibiotic to work better. Say, like loperamide can help, say, uptake of tetracycline and some other compounds. Then combination therapies, resistome information can be more useful in deciding on the combination antibiotic therapies and say, including alternatives. And it also tells about resistance reversal. Next. So, in short, it was just my purpose was to introduce you and to put before you a strategy that can work better than the traditional approach in tackling AMR issues, particularly surveillance and evaluation of treatment methods and selection of methods, and in addition, new drugs and all that. So to conclude, resistome paradigm, which we are currently having, will impact the strategies to combat AR pathogens in the frame of One Health concept. Antibiotic resistance of microbial communities can now be determined by the NGS technology and tools. We have these technologies. It's not a future thing. We can already avail them and use them for our purpose. Resistome guided pathways will improve AR surveillance and risk analysis, effectiveness of waste treatment methods, stewardship practices, discovery of new ARGs, new antibiotics and alternative therapeutic options, and then resistomes of different environments in India also need to be studied more for more effective combating of AR threats. I think in India we have not taken this strategy at a large scale, which needs to be done, and for our own betterment and for control of this problem. And there, for further reading, I have included some very recent articles, although there is a huge literature available, and in all monthly journals you can find very good articles on this problem. And thank you very much for patient listening. Now,
thank you, Professor Singh. It was very interesting and informative. Any questions or clarifications or comments? No? But in fact, I will sum up at the last. So maybe that, but extremely important talk, because you know that the resistomes, but particularly in the microbial communities, this is a problem. We are mainly focusing only on the pathogens, but we are avoiding deliberately or intentionally the other microbes which are beneficial. And this is really a problem, and many of the scientists are not working on that. This is a new aspect where we must focus, particularly on the soil microbes, soil microbiology, which has been totally disturbed during the last 30 years. I will not 30 years, 50 years, I will say, after 1970. After 1970, when the industrialization of the things have come, then the entire soil micro, if you compare the soil microbiology of any soil before 1970 and now in 2020 or 2021, you will find a big difference, not only in bacteria, but even the fungi, protozoans, algae, and all. So we are mainly focusing on bacteria, but we are also avoiding the fungi. which are also getting the resistance. So I will sum up at the last. So let me invite the last speaker of this session, Dr. Mumtaz Saxena, Professor of Animal Biotechnology from the GB Pant University of Agriculture, and he will be speaking on molecular basis of development of multiple drug resistance. Dr. Saxena, please.
Very good afternoon, sir.
Very good afternoon.
Very good afternoon.
Have you?
My voice is clear, sir?
Yes.
Yes.
Clear.
Yes,
It is clear.
It is clear. Are you able to see my presentation? My slides?
Your screen is
Your
there,
screen
but
is there,
the
but
presentation
still
is
the
not coming.
presentation is not coming.
Network
Network
visualization.
visualization.
You have to come
You
on
have
the
to come on the
presentation.
Yeah, sir. I'm trying, sir. Give me 2 minutes.
Take the time in
Take the time in the mean.
Let me talk, because this is multi-drug resistance. This is one of the most invited or most interesting topic present day. All countries are focusing on it. And very unfortunately, in India, we are not focusing. As Harshit has pointed out in the first session, that the surveillance, which is one of the most important aspects that we are avoiding. Surveillance, and that surveillance must be genuine, correct, and then these strains should be, there should be a repository, and it should be at a local level. We are India is a vast country, and if we think that in Pune or in Delhi or 1 or 2 or 3 centers we will create and it will be able to solve the problem, no. The resistance repository, it must be in each district. I say that in each hospital there should be, in each agriculture university there should be, in all universities there should be, so that the genomes or the microbes, they can be stored and they can be consumed and used for the future reference. And Harshit, I know that he is doing good job, and I think Dinesh is also here. Dinesh from CCSU. In the morning he was here. Has he left? Has he left? Very unfortunate. So we are also working over here in Soviet University. Since I took the charge, then we are collecting the good number of the microbes. And when I was in CCS, I was having a more than 4,000 bacterial strains, fungal strains, and actinomycetes. These were the I were focusing. So, Dr. Mumtaz, are you back? Dr. Mumtaz, I am just taking the time. When you are back, then you can start. So
I'm trying, sir. I am facing some problem in the sharing of the screen.
Okay, so that is the thing, that the universities, they should focus on the culture collection, and these collections should be adequately, they should be stored and maintained. The microbiology, unfortunately, before COVID-19 pandemic, most of the people were not knowing it. If you talk anyone that I am working in microbiology or I am a microbiologist, that the people will ask you that what is microbiology. But thanks to coronavirus, that it has taught everyone that what is microbiology. Yes, everyone, even if you talk to a common person who is totally uneducated, and if you ask him, then he will also tell you that microbes microbiology, or these are the bacteria, virus. So, and many things in COVID-19, they have been positive also. They have been positive. We are sitting over here, we are discussing the things from a distance. That is the one very positive aspect. And now, recently I was reading the Nature journal last week. There was a survey. Dr. Mumtesh, are you back?
Sir, I'm facing problem in the sharing of the screen in my system. So if you allow me, I can discuss on Extreme boards.
Okay, fine. That's fine. That's fine. You can. Most welcome. The purpose is the interaction and the dissemination of the information and knowledge. Okay, Dr. Mumtesh, welcome.
Yeah, sir. Give me 1 minute, sir.
Yeah, okay.
Presentation.
So
Just a minute, sir.
Last week there was a survey, and 74% of the scientists, they were of the view that the conferences, seminars, they should be in virtual mode. 74%, because we can connect easily from the far off places, and we can interact easily. And had there been no COVID-19 pandemic, India would have taken further 10 years to come on this technology. So that is the one very positive aspect, that at least the universities or the higher education institutions, or even the primary or the middle class institutions, they have come on the online system, and they have been able to interact positively. They are contributing to the science. So this is one. So Dr. Mumtesh, should we start? Okay, let me sum up in the meantime the 2 lectures which have been conducted. The first lecture was by Dr. Bhojraj Singh from IVRA Bareilly, and he has beautifully summarized the AMR. Mainly, he focused that in developing countries, this is a problem, and the 2 million Indians are affected. Yes, but my guess is that maybe the more than 2 million people, they are affected by this antimicrobial resistance disease, and a lot of money is invested on their treatment, and many times these people, they will die because of the non-availability of the effective medicines. The antibiotics are not good. There was a time when in 1945, after the Second World War, that the antibiotics, they were introduced after the surgery. So that was the time when they were required for the surgery or to avoid the secondary infections. But we have taken it as a routine diet. If we are suffering from some cold, or if we are suffering from some mild infections, then we are taking the antibiotics. And in India, this is the most unfortunate, that anybody can go to the pharmacy store and can buy any antibiotic without any prescription. And what to talk of the prescription? Even if you go to the doctor, then the doctors, most of the doctors, they will prescribe you the antibiotics, not even one, they will prescribe you 2, 3, 4, and they will think that either of the 4, at least one will work. So this is the factual condition what you will find in the prescriptions, or even the companies. Now you will find that that the capsules which are available, these capsules, they are having the powder, and these are not having the single salt. They are having the multiple salts, or even the multiple tablets inside the capsule. So they will prescribe you one, so that you think that only the one medicine has been prescribed, but in fact it is having the several medicines. So these, and out of these several, only one is effective. The rest of the medicines, they are not effective, and they are mainly responsible for the induction of this multiple drug resistance. So that is the one very, very important thing. The government has started thinking only, I will say it, because very frankly, the government still in India at least, we are still not having any policy to regulate the marketing and prescription of the antibiotics. And we have not even adequately educated the people about the use of the antibiotics. The antibiotics, when we use, then many times you might have focused that after taking the antibiotics, your gut, it gets leaky.
And that
And
is
that is
the gut
the gut
microbes.
microbes, they are affected. Their population is affected. And until now you might be knowing that all the signals, they go from brain to the all other organs. But now the scientists have confirmed that the signals go from the gut to the brain and then they get distant. And in gut, from where the signal comes, they come from the microbes. So the gut microbiome is one of the very important aspect where we people should focus. Last time I was discussing with Harshit that we should work together and we should start focusing on the gut microbiome. And now the recent, during this pandemic era, corona pandemic era, you might have seen that the Ayurveda has come in a big way. And this Ayurveda, or the prescription of the herbal medicines or the herbal plants or the herbal antimicrobials, we can say it, these herbal antimicrobials, don't think that they are the Sanjeevani or they are the remedy. If you are taking only single herbal, then it will also disturb your gut microbiome. Take multiple herbals in lesser doses. So one of the very important aspect in the Ayurveda is, we were discussing in the lunch also with Dr. Praveen Malik and Dr. Amit, that Dr. Kalra, that whenever we are going to focus on the Ayurveda, we should focus on its prescription, on its validation of the scientific data. And if we are able to focus on the validation of the scientific data of the herbal system, we will be able to beat the world. We are in India having the 30% of the total biodiversity of the world. 30% of the total biodiversity. 15 different agroclimatic zones. Whatever the herbs you can think of, it will be available in one or the other part of the country. The only thing is that we have to validate its data, we have to focus it. Whatever the amount of money we are investing on allopathic drugs or allopathic investigations, if we are able to spend even 10% of it on Ayurveda, then we will be able to improve the condition of the system. So I think that Dr. Mamtesh has joined. So, well, Dr. Mamtesh, welcome in this session.
Thank you, sir. Thank you, sir.
Yes, Dr. Mamtesh, please. Yes, Dr. Saxena, you can start.
Yes, sir. Yes, sir. Yes, sir.
So Dr. Bhojraj Singh had emphasized on that the globally 10% is being used in humans the antibiotics and 90% in the livestock. But one thing is very important, that in India, we are not mainly focusing on the livestock, and mainly our focus in the antibiotic consumption is in the humans. And overdue and misuse was another highlight which is mentioned, and the very important thing what he mentioned, that is the hand sanitizers. And he was of the opinion that the 46 to 60% of the microbes, they are killed and rest not. But I think that this Canadian investigation, I have also seen this paper, but this paper has been criticized by the other microbiologists also, because this is based on the only few types of the hand sanitizers. And now you will find that in the market there are several hand sanitizers. I was looking here that the one very important, I will just try to classify, that is the one type of the sanitizers which are having the glycerol also, about 30% or something like that. So we have tested that those type of the sanitizers, and those sanitizers are not very good. I'm not criticizing, but microbiologically they are not very good. So Dr. Mamtesh, have you come?
Yes, sir. Should I start, sir?
Okay,
Hello?
yes, please. I was just taking the time.
Okay, sir. So
Yes,
I'm sorry,
please.
sir.
Yes, welcome.
I have taken some time in this acclimatization of the system. So in next 15 to 20 minutes, I will try to sum up my discussion, sir. Basically, once we talk about the multiple drug resistant, uh, the major issue which raised that why this drug resistance is increasing. So I am trying to elaborate some of the causes and some of the means by which we can avoid this development of the drug resistance. You know, once the human being started their life on the earth, the pathogens come with that. So there is always a competition between the health and the disease. So what the human did, they tried to develop some tools in the form of the drugs and to overcome on the pathogen. But very smartly, the pathogen developed their own tools to overcome these drugs, and the result is like that. Most of most of us have known that these pan resistant who are having the and resistant for more than 6 drugs, even 7 drugs, and most of these drugs are very new drugs also. So what the people tried, they started thinking about the combination therapy rather than using one drug. They started thinking of 2, 3 drugs. In the 2015, it was the WHO recommended this thing, though some of the countries was practicing it. But within the 3 years of the introduction of the combination therapy, the questions were raised on this approach also. So why the MDR is developing so quickly? There are several reasons. There are transposomes, there are plasmids, there are accumulation of the drugs, there are multiple drug efflux systems. So I'll try to touch one by one. The most commonly reason at the molecular level is the mutation in the targets. See, most of the drugs which we are used, they are targeted on a particular target molecule. Say, for example, DNA polymerase or the specific proteins. So what the organism used to do, they used to modify them, these target organs, and these target molecules. And once they are successful in the modification of these target molecules, the drugs offer no choice. Similarly, plasmids are equally playing their roles. This example of how the erythromycin resistance was developed. The plasmid encoded the gene ENB was transferred. Apart from that, sometimes the overexpression of some of the drugs, like some of the enzymes or some of the genes also contribute in that. Has been seen in the cases of the sulfur drugs, that the high overexpression of the dihydrofolate synthesis resulted into the resistance against the sulfur drugs. Enzyme inactivation, the well-defined mechanism by which the plasmid provide the gene to the susceptible bacteria and they develop the resistance. This is a classical example of how the penicillin resistance was developed. People try to develop the semi-synthetic antibiotics to modify it to resolve the problem of this development of these enzymes. But bacteria is even more smarter. They have modified even their enzyme against these semi-synthetic antibiotics also. Then acquisition from the other species. As the earlier speaker was discussing, Dr. Ajit, Professor Ajit was saying that we have to think about the soil because most of the drugs which has been disposed in the soil, they are contributing for this development of the antibiotics. So sometime what happens, pathogenic or non-pathogenic organism, they are contributing the resistant genes to the pathogenic organism. Then this is again the modification of the target molecules, vancomycin, which used to bind on a pentapeptide, that is DLA. And once the resistant developed, they slightly modified the target molecule, and the organism is no more susceptible for the vancomycin. Then organisms have developed some very specific systems. They develop the efflux system, means they develop the tools by which they can throw out the antibiotics from their cells. They develop some of the systems by which they can inhibit the drugs at the local levels by using some of the very specific proteins, such as QR proteins, which modified the enzyme topoisomerase. So then non-specific inhibitions, we know that we are all aware about the outer membrane proteins or the porins, which has been targeted as a molecules for the drugs, for the development of the vaccines. But on the other hand, that these porins play very important role in the transport of these molecules. So if there is a mutation, if there is a changes, if the organism are able to to modify their porins, they become resistant for the drugs. Biofilm formation is a well-known phenomena, so need not to discuss about it. Then what are the sources of these antibiotics resistant genes? See, there is a very classical example. We have isolated the vancomycin from an organism, and the pathogenic organism also obtained the same gene to develop the resistance against these vancomycin. So this is how nicely the system works against each other. Then this microenvironment, we can say some of the bacteria like Proteus or Serratia, which are very common organism in the soils, they also transfer the genes which can help in the development of the drug resistant. And you'll be surprised to know that more than 70% resistant genes came from the soil. So here is that very important issue that we should have to think about not only your health, not only the animal health, but also about the soil health. Then our plasmid, it is a well-known plasmid which have contributed for the development of the plasma resistance. So why this antibiotics resistance has been developed? First, very commonly we have misused it. We have contaminated our environment. We have irrationally used it in the feed additives. We have used it against the viral diseases. And once we have used it in the low dose or in the substandard dose. So in the near future, this is a report which was published by the CDC recently, and they during the COVID outbreak, and they have anticipated that by the 2050, if the progression of the multiple drug resistance remains at the same speed, by the 2050, 300 premature deaths will be resulted. And that will cost in the term of economy more than 100 trillions. Definitely the antibiotic resistance will develop. But if we can delay this resistance, even just 10 years, it will save $65 trillion, which is much more higher than the total GDP of the India. And this development of the multiple drug resistance will reduce the GDP of the world up maybe more than 3%. So it's a very important issue. If you see the recent scenario, what we are trying to do is not correct. In very few cases, we are trying to determine the pathogens, especially in cases of the ICU, the antibiotics are not used rationally. So most of the multiple drug resistant organism or the organism like pan resistant, they have been isolated from the ICUs. And then some subtherapeutic levels, as I have told you earlier in earlier slide. Then if I'm just giving a classical example of the ofloxacin, which is very commonly used for the treatment of the gastroenteritis. If we see the USDA recommendation, a person should use 300 milligrams dose twice a day for 5 to 7 days. And normally what we used to do, we used to take the 200 milligrams dose for 3-4 days. So this is a classical example that how the antibiotic resistance has been developed. We have worked on the 2 projects. One was funded by DST, another one by DBT, in which we try to work on the gangetic water and the commercially poultry eggs which are supplied to the market. And we isolated more than 150 salmonella isolates. We are working on salmonella. So for our study was focused on salmonella. So, and what we could observe that most of these organisms were having the resistance for the commonly used antibiotics. So if we take the classical example of the Salmonella typhi, earlier the chloramphenicol was used, and but as very quickly the resistance was developed against most of these drugs, and so finally in 2016, this NCDC has to decide for the option of combinational therapy, which has also been questioned. Now the WHO has provided some of the recommendations to the different classes for the human practitioner, for the veterinarian practitioners, that don't prescribe these antibiotics if they are not necessary. Use sensitivity trials before use. Don't use it in suboptimal concentrations, and try to develop a database of the antibiotic responses. For the people, for the general people, we used to take the antibiotics. What the best thing we can do, we should not dispose the antibiotics in the environment, because this is the most common way by which we are contributing for the antibiotic resistance. Then for the people, for the agriculture and for the farmer sector, use the antibiotics under the use of antibiotics in the food chains, and then the government has to take the risk that the development of the new drug antibiotics has this movement for development of resistance. These new antibiotics resistance is developing very quickly. So to give the subsidy to develop the rational, and of course, and this is what I am talking about. The industry has to think that what is more important for them, economy or the health. Time is running out very fast if we are using the antibiotics in the same way. For a good health to all of you, because still we are facing the pandemic. We are in the midway of the pandemic. I pray that all of us should stay safe and healthy. Thank you very much.
Thank you, Dr. Saxena. Any questions or comments or clarifications from the audience? No? Yes, please. Yes. Yes, Nilesh.
We are continuously using huge amounts of these antibiotics, and we know that bacteria becoming resistant to these antibiotics. Whether we have to move toward the alternative of these antibiotics is best way, or creating new antibiotics is the one way.
Yes, Dr. Muntesh, would you like to reply to his question? Nilesh is asking a very important question, that to combat this MDR, should we go for the new antibiotics, or
we can search for alternative.
Yeah. Yeah, we have to think about the alternative, because right now what is happening, industry are no more interested for the development of the new antibiotics, because industry always think in the term of economy. So we have to think that whatever the tools available with us, we have to explore them in the best possible way.
Dr. Nilesh, this is a very interesting question which you have put. And the microbiologists and the doctors are threatened to answer this question, because no pharmaceutical industry, as you know, that during the last 2 decades has focused on any new antibiotic, and they will also not. And you will find that COVID-19 pandemic, this is the only first vaccine which has been released in less than 1 year. Before that, it was the mumps vaccine in 1964, which was released in 4 years. Because this is a trial system is very lengthy and very long and a huge investment. And by the time this antibiotic comes in the market, we get the new strains. So we produce the antibiotic in the pharmaceutical industry against a one particular resistant bacteria, and then by the time it will come in the market, the another strain has come. So that is why you will find that this is a very difficult answer for this. The question is very simple, but the answer is very difficult. And I don't think that we are having the answer. The answer, what lies, which was Dr. Saxena and all people were speaking in the morning, that is we have to switch over to the Ayurvedic system. Ayurvedic system, I'm not very supportive like that, but scientifically, you know, that the Ayurvedic system of the herbal medicines, besides the active component, they are having several other components which are protective in nature. So if one compound is doing some harm to the natural system, then there are the other compounds in that and they will be able to protect. So that is why now you will find that in next 10 years, you will find that the entire system of the medicine will change. The probiotics, functional food, Ayurvedic, and all these things will take the place of this allopathy. And the allopathy will lose its significance in the next 10 years. I'm pretty sure you will also live for next 10 years, and I will also live. And and then after 10 years you will get the answer.
One more thing is coming in the market, na yoga.
Yes. Yoga, that is also a perfect science. That is also the combination of the yoga and the Ayurveda. It can combat. We are focusing on the disease, while we should focus on the health. This is very, very strange that all the pharmaceutical industries, they are focusing on that this antimicrobial, this drug for this disease, and unfortunately we are not focusing on the health, while our ultimate goal is the health. So we should focus on the health, that how the person can remain healthy, and for keeping the person healthy, there have to be several ways. We have to live happily, psychological factors. Then the gut microbiome, जैसा अन्न वैसा मन, बहुत पुरानी कहावत है. Whatever you will eat, you will think the same. Why you will think the same? You will get the same type of the bacteria in your gut, and the gut bacteria will give the signal to the brain, and the brain will think. So positive thinking. So we have to focus on the health. So let me any other question? Let us first finish Dr. Montes' question, then I will sum up. Any other question or comments? Dr. Nilesh, thank you. You have put a very nice question and very interesting, which will be answered in next 10 years. Any other question? Dinesh? No? Pankaj?
We are talking about today the antimicrobial activity. First of all, I think we should improve our immune system.
Yes.
Definitely, this is the best alternate. We should not talk about the antimicrobial system. We have to मस्त रहें, स्वस्थ रहें, व्यस्त रहें.
So thank you, Doctor. Yeah. So thank you, Dr. Muntesh. Thank you very much.
Thank you, sir.
So the first lecture, Dr. Bhojraj Singh, as I have also emphasized, that he had also mentioned that the use of the neem, turmeric, marigold, and the disinfectants, liquid disinfectant, the neem, marigold, or turmeric, or the other herbals, they need to be studied. Do they induce the resistance? In my opinion, or I have a strong opinion, that in the consumable quantity, they do not. because the neem is having more than 200, 250 antimicrobials, and to my knowledge or to my information, it has not caused any resistance in any of the microbe. So the biocides at the low doses was also the another factor what he has mentioned, the new dimensions. And the new dimensions, several new dimensions were discussed by him. Beautiful lecture, and he had focused on the use of this disinfectant which has been used, rather commonly used during this corona pandemic. And this has certainly, yes, I do agree that this has also introduced some sort of resistance. So young microbiologists, whosoever are present here, I would like to invite their attention that they should focus on their PG thesis or UG thesis, that they should see that how these hand disinfectants, they are causing the resistance, or how much microbial load they are able to reduce under the natural conditions. Then he had uh, also focused on the substandard drugs resistant. The very important question what he has raised, that is, we are using the word not of standard. Yes, this is very misnomer. Not of standard is the no word. Why we are trying to use the word substandard? So from this platform, I would like to invite the attention of the organizers that in the recommendations, they should write that the government should focus and should clearly spell out that these are the standard and these are the substandard. So more than 60% of the antibiotics which are in the market, they are substandard. And they are misleading the consumers by using the term not of standards. So these drug maintenance, Indian Drug Manufacturers Association, they are lobbying. So at the same time, we the scientists, microbiologists, should also lobby and should also focus and should invite the attention of the policymakers that that these things should not happen. 121 antibiotics. He has said that these are the antibiotics which were said that their effluent should be discharged in the nature after treatment. And due to the lobby of the drug manufacturers, this bill was withdrawn. It is very sad. We, the environmentalists, we, the scientists, we, the agriculturists, we, the people, should focus and invite the attention of the government that such type of the bills which are based on the scientific inventions, which are meant for the protection of the future, they should be discussed at scientific forum, and they should be withdrawn, or they should be considered only on the recommendation of the scientific committees. Dr. Ajit Singh has focused on the resistance. That is true, very true. He had pointed out, if you remember, that he had said that antibiotics came first or antibiotic resistance came first. If you see the evolution of the cell, you will find that the antibiotic resistance in the evolution, it came first. The enzymes which are responsible for the induction of the resistance, they originated more than 3 million years ago in the nature. So these type of the enzymes, they were already known when the antibiotics were introduced after the Second World War in the surgery for treating the secondary infections. And the scientists were knowing very well that one day, sooner or later, these antibiotics will introduce the resistance. But the pharmaceutical lobbies, again I am coming on the same thing, that the pharmaceutical lobbies, they came in the market, they sold their product, and now the antibiotic industry is almost finished. You will find that, but still you will find that there's more than 1600 crore rupees in India. They are still earning, knowing fully well that the antibiotics are causing the great harm and loss, not only to the lives, but to the environment. into the economy, into the future generations, into the evolution of the species, and they are damaging the environment. But he has rightly pointed out that the resistance for most of the antibiotics, it takes 3 to 4 years. So now your answer to your question is here, that you said that what is the new challenges. In 3 to 4 years, no new antibiotic can be introduced in the market by any of the pharmaceutical industry if they follow the right protocol for the testing or for the trial. And that is why the pharmaceutical industries are not focusing on the new antibiotic. And new antibiotic is not the solution of the antibiotic resistance. You may introduce the new antibiotic, the microbes will again learn this and they will come out. The microbes are intelligent than you. The single microbial cell is containing more than 10,000 of the genes. So you can just think, and we are having more than 3 trillions of the cells, and our each cell, each chromosome is having only 1,000 genes. Our each chromosome, and that single cell is having more than 10,000. So that single bacterial cell is more intelligent, more during the course of evolution. The bacteria, archaeobacteria, they have not changed their genetical system. It is stable. And now you can see that during this course of evolution, every species, every major species, the big, larger species, they are changing their genome to survive in the nature. So the smart strategies to combat the AMR was discussed by him. Beautifully, he has mentioned that the metagenomics and the several other methods, they can be used for studying this smart, to combat the antibiotic resistomes. Antibiotic resistomes, that is, you will find that the microbial communities, in all microbial communities, that is, which I was mentioning, that we are focusing just on the pathogens. But what about the saprophytes? What about the major beneficial microbes? They are also developing the antibiotic resistance. Or why only antibiotic resistance? Even the resistance for the fungicides, resistant for the insecticides, resistant for the every agrochemicals what we are using. And that is why, which is mainly responsible for causing the infertility of the soil or the decrease of the fertility of the soil. So that is why we should focus not only on the antibiotics, but we should focus on all chemicals which are dangerous in threat to the microbial system. The last topic, which was discussed by Dr. Mumtesh, a wonderful topic. He has discussed that the transposomes, plasmids, multiple drugs, efflux, and the accumulation. These are the four major factors which are mainly responsible for induction of the multiple drug resistance. So from the mic, he has also isolated some of the MDR isolates, and he has also mentioned and emphasized that more than 70% of the resistant genes, they are coming from the soil. Yes, right, correct. And we are disturbing the soil intentionally or unintentionally, using the different type of the agrochemicals, using the different type of the systems, using the monoculture also. That is also disturbing the soil microbiology and soil ecology. So that is why we should focus on the balanced system. We should come on the Vedic system, Vedic microbiology, Vedic science, which is sustainable. Which is sustainable. So the WHO recommendations to avoid the MDR, that should be strictly followed. We would like to make a recommendation through this platform to the government agencies that the antibiotic susceptibility, without the antibiotic susceptibility test, no doctor should be allowed to make the recommendation for the use of the antibiotics. There should be a policy. In the policy paper, the government should focus and introduce. that government should also introduce that the full course of the antibiotics should be must. If you go to the Western countries, you will find that the doctor only identify or the disease, diagnose the disease. He does not make the prescription. He will have to go to the medical store and the pharmacist will make the prescription. And the pharmacist will give the entire course. There will be the envelope, the entire course will be given, and this course will have the all precautions, that what type of medicine is to be taken, what precautions he has to take, what food he has to take, what not he has to take, and he has to follow and there should be a follow-up action that the entire course should be. Otherwise, in India, we will face the multiple problems. Minimum use of the antibiotics in the food chain. That is also the recommendation of the WHO, which should also be endorsed through this platform. And all the antibiotics should be used and should be prescribed under the specialist supervision, and there should be a strict surveillance, as Dr. Harshit has mentioned in the morning session also. Without the surveillance, we cannot combat the multiple drug resistance. Then this is one of the major threat for the human health. I don't say for the industries, but it is the major threat for the human health. And we should focus on the human health instead of on the pathogens. So thank you very much. Thanks to all the three speakers who made a nice presentation, and thanks to the organizers for giving me an opportunity to chair the session, and thanks to the participants for the patience hearing, and thank you very much. Yes, Dr. You were saying.
This morning, I raised the question to Dr. Malik also, but I didn't get the answers. Today, the system of Indian system of administration was to approve heavy dose of the antibiotic.
Actually, you know that my personal opinion is that if you ban the antibiotics today, suppose, if we want to know the importance of any of the system, you just think negative. You go back. If you ban, then the pupil will have the secondary infections, and how will you control during the surgery? So before banning, we should make and find out the effective alternatives. And the effective alternatives lies in the herbals and the Ayurveda. So we should first authenticate them. We should find out. So the Ayush Mantralaya is working on that. And once we are having the documentation of these herbals and the Ayurvedic system, and when we are, and you know that there are very highly effective herbals which are more effective than the antibiotics. We have to document them. Once we have these, then we should make them popularized. We should make their good manufacturing system, and then in the phased out manner, because they have established into the system. Anything which has established into the system cannot be eradicated immediately. For example, the corruption has invaded in our system, and if you want to eradicate overnight, it will not. You can phase out. You can phase out slowly and slowly, and you have to work strategically. So if you have to eliminate the antibiotics, so we have to first find out the alternatives, and these alternatives should be first made available to the public. And once they are made available to the public, then phased out, they will die their own death. You make it popularized. You come in the competition. Now, during this pandemic, you have seen that the several people, they migrated to the Ayurvedic system, to the herbals, to the vegetarian food. So the thing is that the popularity, you have to popularize, you have to come in the competition. Then they will be phased out. So thank you. I think that now, Dr.
Thank you very much, sir, for enlightening us and chairing this session. It is great privilege to our college and itself from the side of participant that you have enlightened us by summarizing in a very massive way, like whatever the recommendation from these three lectures that will be putting in a frame that it should be recommended to be scientific community, social community as well as the governmental system, that they will implement something on focus on those aspects which you have recommended. On behalf of the College of Biotechnology, I am again thank you, sir, for chairing this session. Thanks a lot. I am also thankful to the co-chair of this session, Dr. Anil Sirui, sir, Director Research of this university, for supporting this session and enlightening the other audiences. I'm also thankful to all the parties, all the speakers, Dr. B.R. Singh from Division of Epidemiology, IVRI, Dr. Ajit Singh, scientist from Luwas, Hissar, Dr. Mumtaz Saxena from SVPUAT, Pantnagar, for enlightening us with the new approaches, new things that should be implemented in our scientific community. Especially the concept of One Health issues that will be more beneficial. Now we have to target it on these issues. Second, you have suggested about the things that we have searched for the alternative for the antibiotics, which is more beneficial and eco-friendly for our society also. At last, I am also thankful to the reporter, Dr. Shalini Gupta, Associate Professor from College of Biotechnology, and Dr. Akshay Garg sir from Veterinary Colleges, for I know that they will get it the pain to make it the report of this session. And on that report and the recommendation of our chairman sir, we are having some valuable thoughts which should be put it in front of the government for making some funding, and that should be implemented for the society welfare. Thank you all. Now I'm proceeding this end of this session with the permission of the chairman and co-chairman sir. If you permit it, we will end it this sessions, technical session third. Now, again, we have moving towards the valedictory sessions. And for the valedictory sessions, the session, as we are already delayed, the session is continued. So I will invite Siroi sir for the welcome address and during this valedictory session. Welcome, sir.
Good evening. It's my privilege to welcome again Dr. A.P. Garg, Honorable Vice Chancellor of Shobit University, to chairman of this valedictory session. Friends, in this valedictory session of important topic, antimicrobial drug resistance, which is a burning topic of the worldwide nowadays. Today we had 7 lectures from eminent speakers from, I must say, globe. We had a speaker from Japan also. We had a representative from WHO also. So we had a very nice compendium of speakers with us, and there are many recommendations which have come up. I'm going to briefly present you the salient highlights, though we will further improve these recommendations before presenting to any forum for actions. Sir, today morning in the inaugural session we had with us Dr. Praveen Malik as chief guest. He is Commissioner Animal Husbandry, Government of India. Besides him, we had with us Mr. Mohanji Saxena, Managing Trustee Dabur Ayurved, he was guest of honor, and the session was chaired by our Honorable Vice Chancellor Professor R.K. Mittal sahab. We had very significant insights on the topic from all these dignitaries. In brief, the recommendations which come up from the day-long proceedings are, sir, some of the major concerns which emerged during the day-long proceedings. First was that it was made aware by all the speakers and delegates available here that there is a limited awareness regarding the use of antibiotics and their after effects, particularly in dairy farmers, poultry breeders, and other stakeholders. Also, there is a lack of facilities to detect drug of choice and perform antibiotic sensitivity to support the selection of effective drug. Simultaneously, lack of cost-effective, safe, and easily available alternate therapy that is lacking in the country. Lack of doorstep diagnosis and biologicals to avoid the exposure of infectious diseases. The antibiotics are being used indiscriminately, which is causing antimicrobial drug resistance for treatment of diseases and as growth promoters. So these are the major concerns which were highlighted today. As far as workshop was concerned, the thematic areas which were discussed, first was public awareness, behavior changes, and communication. Second was national action plans on antimicrobial resistance, optimizing use of antimicrobials, innovation, research, development, and access, surveillance and monitoring, and besides that, governance and alignment with sustainable development goals. So first recommendations that has come up is to ensure equitable and affordable access to existing and new quality assured antimicrobials and their careful use by competent licensed professionals across human, animal, and plant health. This is, I think, one of the most important recommendations coming out of this workshop. This recommendation needs to be supported by efforts both to reduce the need of antimicrobials and improve access through lowering the prevalence of infection through clean water, sanitization, and hygiene. If we do this, we will certainly will not need antibiotics. Then decreasing the likelihood of diseases and their spread through delivery of existing vaccines and strengthening infection prevention and control measures. Ensuring best practices in animal and plant health, food production, and waste management. This is important. Then supporting behavior change through effective communication and incentives targeted at the public and professionals in human, animal, and plant health, as well as food production and the environment. Lastly, strengthening national surveillance, regulatory, and accountability mechanisms, they need to be strengthened. The next important recommendation that has come up is to accelerate the development and implementation of effective One Health National Antimicrobial Resistance Action Plan within the context of Standard Development Goals. Under this, what needs to be done is we have to prioritize actions and interventions that are specific to the national context, particularly in our own country. Then strengthening key national systems for infection prevention, monitoring, integrated surveillance, procurement of health commodities, and waste management. Then technical cooperation, capacity development, research, and advocacy components, including support at national and local levels to mobilize action on antimicrobial resistance. Effective national coordination, accountability, and governance mechanisms. By these, we can take up this recommendation. Next important recommendation that has come up, we have to phase out the use of antimicrobials for growth promotions. They are being used as growth promoters. They have to be phased out in due course of time. Next is to increase investment and innovation in new antimicrobials, particularly antibiotics, diagnostics, vaccines, waste management tools, and safe and effective alternatives to antimicrobials for human, animal, and plant health. This needs to be done. Then strengthen research collaboration in a One Health context. This needs to be strengthened. Under this, we have to undertake coordinated global mapping of research and development activities and funding to address antimicrobial resistance. We have to establish and maintain a platform for sharing information on research and compounds in development in both ongoing and completed research and development activities so that we can work on that. Promoting synergies and opportunities for collaboration among funders and researchers in human, animal, and plant health, besides protecting environment. And lastly, the promoting openness and transparency in data. That is important. We have to have data, and that should be open to everybody from all research and monitoring and surveillance sources. Next recommendation, which I think comes up, is the systematic and meaningful engagement of civil society groups and organizations as key stakeholders in the One Health response to antimicrobial resistance at regional, national, and local bodies. Here we have to strengthen their roles in accountability, advocacy, monitoring progress, and ensuring careful use of antimicrobials, promoting synergies with consumer and civil society groups active in other sectors, including in climate change, disease management, and the environment, and other aspects that are described in standard development goals. Provision of political, financial, and technical support for civil society organizations to enhance their engagement, including the work with governments. Then next is to take care of unusual causes of emergence of antimicrobial drug resistance. You have explained it in the last session in detail. There is immediate need for robust risk assessment mechanisms for identifying risk of antimicrobials on the emergence of AMR. Then along the major causes of emergence of AMR, other factors like poor public health indicators, abolition of substandard antibiotics in therapy, and the availability of inexpensive antibiotics over the counter without a prescriptions needs to be taken care of for the control of dissemination of antimicrobial resistance in India. Then the next generation tools and technologies are developed to improve the strategy to combat the antimicrobial resistance threats. This is also explained by you in detail, Sir Rabi, in the last lecture. You have talked about the resistance. I'm skipping that. You have deliberated in depth on that. Then to overcome AMR, understanding the mechanisms of efflux pumps regulations, especially during disease pathophysiology and drug treatment, is inevitable. If the pathogen efflux pumps are selective inhibited, it will lead to a great intracellular concentrations of the drug in the pathogen and better killing, and will overcome the rising problem of the drug resistance. If the efflux pump of the host can be selectively activated, it will reduce the chances of drug accumulation and toxicity in the host. This will also increase the safety of the drug. Based on these recommendations, sir, I have listed a few points. For example, the diagnostics and laboratory support system that needs to be established, extended, and strengthened in institutions and throughout the country. Then there is a need of laboratories to detect drug residue in milk, meat, egg, and their products at smaller levels, village levels or block levels. Then there is a need to modify and revise the FSSAI standards for animal and poultry feed and fodders. Then surveillance of antibiotic residues is an important parameter and need to be addressed on priority. Focus is required for the development and availability of low-cost, effective disease diagnostic kits and biologicals. We have taken our lead on this, and we have already taken a project in this college on this lines. Then promotion of herbal and ethno-veterinary medicines is of course needed. Simultaneously focus on indigenous technological know-how based ethno-veterinary medicines with expected development and validation of ITK based preparations. This is required. Surveillance of antibiotic residue in animal products. It is imperative to develop and execute an awareness program among stakeholders, including farmers, veterinarians, industrialists, and even consumers. And lastly, development of IT-based interconnectivity platforms for the farmers, scientists, researchers, and industry to understand each other's requirement and develop strategies considering the interest of all stakeholders for this antimicrobial drug resistance. So these are some of the points which I could gather from the day-long proceedings. I myself, as well as our faculty members, we will sit together and further improvise these proceedings. So once again, I thank you, sir, for giving me this opportunity to present these recommendations. And before I leave, I must thank all the delegates, my faculty colleagues, my dean, college dean, Dr. Ravindra Kumar, for organizing this one-day workshop. And I also thank World Bank supported innovation, NAHEP innovation grant for financial assistance. Thank you, sir. Thank you very much.
Thank you very much. Thank you.
Now I invite Professor A.P. Garg for a presidential address.
Well, Dr. Sarohi and my colleagues, it was a wonderful experience and opportunity for me to be present amongst my colleagues and to interact with them with a true science. As you know that antibiotics, we are listening since morning till now. They should be better referred as antimicrobials. And that the antibiotics term which was introduced long back, that is those which are produced by the microbes and they are against the specific microbes. But now no antibiotic is active against the specific microbe. You will find that either against the gram-positive bacteria, against the gram-negative bacteria, or some antibiotics against the both types of the bacteria. So the antimicrobials will be a proper word, that is, they are working against the microbes. One thing. Second thing, you will notice that now the term has less meaning. Antibiotics terms, antimicrobial term has become very wide. That several chemicals which are produced by herbs, as I was mentioning, produced by even the food materials produced, they are also working against the microbes. For example, onion, lasun, ginger, black pepper, turmeric, and several spices. Almost all spices are working against the bacteria and against several microbes. So to call that term, we should use better term, the antimicrobials, which will include everything, which will include even fungicide, which will include insecticide, which will include everything. And we are giving more and more emphasis to the bacteria. I wonder that, and if you see it, you will find that how much percentage of the bacteria are pathogenic. You will notice that a large number of viruses, 1.3 million of the unidentified viruses, they are living in the wild animals. And they can be transferred anytime, any day, from the animal's host to the man, as we have seen in case of the coronaviruses. If you see the origin of the coronaviruses, you will find that in 2002, SARS, then MERS, all they have been transferred from the animals to the humans, even this COVID-19. So we should also focus on the control of the viruses. If you trace the history of the deadly microbes, you will find that the viruses, several viruses, they have created the pandemics and they have devastated a large population in comparison to the bacteria. So we should have a balanced approach. We should work on the viruses also. We should work on the protozoans also. We should work on the fungi also. We should work on the bacteria also. And we should try to find out such antimicrobials which are working against a wide range of the microbes, wide group of the microbes. Any antimicrobial which is working against the narrow range, this will not survive for a longer time. This will be, it will be specific, and it will be specific only for a short time, and it will, in due course of time, it will cause the resistance. And we, the scientific community, should be open and should express our scientific data and should focus on the problems which are relevant to the society. And this workshop, which is focused on the antibiotics resistance, which is the one of the most important thing, which is the most burning topic, and it was rightly selected because the most of the doctors, they are prescribing the antibiotic, and even the agricultural scientist or the veterinary scientist or the food scientist, they are putting the antibiotics in the food chain without even knowing that what impact they will have in the food chain itself. So they need to be regulated strictly. And in India, such a large country which is having the more than 130 million of the population, and in this case, still we are not focusing on this. More than 138 billion population. So we have to focus, and we have to make the strategy, and the universities like agricultural universities or the science universities, they need to take a focus. They need to take a lead by making the recommendations, by doing the research, by educating the pupil, by making the I mean to say communication with the general public. And with that viewpoint, all the 7 lectures which were delivered in the morning lectures, I was only present in the 2 lectures, but I know Professor Malik since long, and I know that he must have spoken very well. He is a wonderful speaker. And Anup Kalra, I know him since long, I think more than 30 years. He is also a wonderful researcher and a wonderful speaker. And he also focused on the Ayurvedic system, the antimicrobials. And we people should also focus on such type of the things which will change the scenario. And even the GDP, our economy, can be changed if we just focus on the agriculture. If we just focus on agriculture, herbal medicine, we can still come to the 30% of the global economy. If you see in the Vedic period, the contribution of India in the entire world economy, it was 30%. And now we are 3% only. And if we have to become Atmanirbhar, if we have to become Vishwaguru, then we have to focus on agriculture, we have to focus on herbal medicines, we have to focus on the scientific validation of data, as Dr. Sirohi has also given the wide range of the recommendations. And I hope and expect that these will be sent to the policy makers. And on behalf of Shobit University, on behalf of the faculty members and the students, I assure that whatever the policy, whatever the system the Agriculture University would like to make in the research or education, we are with you. Always. Anytime you can, because I don't, I have many persons to focus on the administration in my university. And whenever you will call me for the academic activity, for whole day or for whole week, I am available. No problem. It is always my pleasure. And thank you for inviting me to chair the session and to interact, not to chair the session, that is a thing, but to give me the chance to interact with the young faculty members and the participants. And it's wonderful that you have organized a dual mode conference. We made the last week the 3-day conference, and then you have also started the dual mode conference. This should be the right approach for the dual mode conferences, so that we can have the both flavors of the networking as well as the academic networking from a globe. So thank you very much. Thank you.
Thank you, sir. Thank you for your motivational words. And sir, one more thing, that when we planned this event, then we thought of the offline participation. So what you have just mentioned, we also thought in the same way, that at least at this scenario, at this juncture, we microbiologists from the different local institute like CCS, University from the Soviet Subharati and the LLRM, we should come together to work on this platform, and we should have some interaction. So we try to accommodate all those which we can accommodate in this short short period of time. And another thing, sir, by the grace of the God, some things they are decided by the destiny. What we used to say, prarabdha. So when I requested you to chair a session, sir. And now you are the guest of honor for the valedictory function also. So being a microbiologist, sir, you better know the need of how important role these microbiologists has to play for the welfare of the society and the veterinary profession, particularly because I am a veterinarian. So that always remain in my mind. So in this context, sir, we have here from Dr. Amit Pandey. He is from the Bikaner. He traveled all the day yesterday from Bikaner to reach this place, because he is working on this aspect there. And we have one person from the Isar, Dr. Sarma. So he traveled whole day, but because we gave only 3-4 days to apply and to get the permission and to reach this place, so we get some limited number from outside. But here we have certain faculty members. Yes, please, Daksab, you please introduce one by one from the other institutes. Yeah.
Very thankful for you, all of you, that I got a lot of knowledge about the microbiology.
My name is Rishika. I'm a PhD scholar at Soviet University.
Okay, thank you. So likewise, we have one more person, and other they might have left. They are all our students. I know them. I would like to get introduction of.
Dr. Neetu Kumari from Meerut College, Meerut, Biotech Department, and I am very thankful that I got a chance to come.
So.
Look after कर रहे हैं. अच्छा. सुधीर.
So likewise, I would like to introduce our microbiology team from college.
Yes, we should make a strong microbiology team here in Meerut.
So we have young promising team from College of Veterinary Sciences. We have two of them here. Dr. Harshit Verma is here. He is a veterinary microbiologist. We have Dr. Akshay Garg. He has a very good experience in the field, and he has also the international exposure to work on different aspects. Basically, a cell culture specialist. And Harshit is basically a bacteriologist. And myself, a microbiologist, particularly specialized in bacteriology. Here I am posted in immunology department.
I have seen few papers of Harshit. They are wonderful.
Sir, he is very energetic.
Yes, and
so,
we have interacted, I think, 3-4 times. Yes, we have interacted.
One more faculty, Dr. Selja, she has some other assignment. Otherwise, she would have been here. Selja Katoch is there also in the microbiology department, and the department is being headed by Dr. Rajiv Singh. He is from a PhD from IVRI. So likewise, we have a team here, and you are free to come here. We have the option to collaborate, and we have initiated one step taking the MOU with the Dabur Ayurved, so that we can have certain large project in multidisciplinary projects. So in that you can cooperate with us, and we will be very happy to cooperate with you. With this, sir, I would like to request.
I would like to recommend here that if, because last time we were discussing with Harshit that he is working on the immunogenic aspects of the herbals. So in the next 10 years, you will find that there will be a big scope. Those young people who wants to collaborate and want to work on the immunogenic activity of the herbals. You can do a part of the work in Shobit University and with Harshit also, and we will be glad because we were discussing last time that, and we should come in a big way. This will change the scenario, I tell you. And microbiology is such a wonderful subject that we get the results within 48 to 72 hours. That is the most important advantage. We need not to wait for 6 months for the crop to come. We need not to wait, and we need not to wait that the rains need to come, or we have to give. Just you put in the BOD and then you will get, after 48 hours to 72 hours maximum, you will get the results. And if you have to repeat, you can repeat the experiment in 1 week. So the microbiology is a wonderful and fascinating subject. And this will be one of the best aspect, those who wants to work, that is the immunogenic activity of the herbals. Just focus on that. You will get good patents, you will get good papers, and not much work. You can evaluate the one herbal, I think, within 6 months. Within 6 months, you can evaluate the complete aspect. So in 1 month, you will be able to get 2 papers, good quality two papers.
So, sir, there were 2 technical sessions, and in those sessions we were having some reporters. So I would like to request you to bless them by presenting a memento, so that it may be a memorable moment for them, and particularly those are youngsters. So I request the chief guest of the function, Dr. Garg sir, and Dr. Saroi sir, the PI of the project and DES and Director Research of the college, university. Dr. Ravinder Kumar Dean sir, to please come over on the top of the stage, so that I can call them one by one. I request first to Dr. Harshit Verma. Although he's senior in a job, sir, senior to Akshay, but in his college and his university time, he is the junior most. So I always starts with Harshit. So Dr. Harshit was there the whole day, and for last 3 days he worked very hard to get this book in the public form. एक फोटो अच्छा नोटियाल साहब चले गए ना सर, आज वहाँ 2 फंक्शन थे। हाँ सर। Now I request Dr. Akshay Garg. जी सर। Now we have Dr. Naresh Pratap Singh, who is the co-organizer of this, co-organizing secretary of this event, and he has worked hard and he managed all the way to the experts. Dr. Naresh. Now Dr. Nilesh Kapoor. And now in this continuation, we have with us Dr. Jiten Singh, sir. Dr. Jiten. Sir, he reached here from Delhi by overnight traveling. He was having some accident in his family, so he has to rush Delhi. Then he came back. He reached here by 3:30 in the morning. Now I invite Dr. Shalini Gupta. Ma'am, please. Today, ma'am was might be remembering the back days of the Pamnagar because Dr. Mamtesh Saxena was in the same lab in which Dr. Sarati was during her PhD. Now, Dr. Rekha Dikshit, ma'am. Without team leaders, nothing can be possible. So, sir. So, in this program, sir, we were having double engine Sarkar. One is on right of yours and one is left on yours, sir. So I would like to request Dr. Harsit to accompany and present this small memento to Dr. Ravinder Kumar, sir, Dean College of Biotechnology, for this type of engine which has given this energy to organize this event. Nothing is possible. So now I request Dr. Akshay to please give Dr. Anil Saroy a memento so that we can remind him time to time that this double engine is required again, sir. And now at last I request Dean College of Biotechnology and Dr. Anil Saroy, sir, to give a memento to Professor A.P. Garg, sir, who have been here, sir, to make this memorable, sir. Sir. So with this, I invite Dr. Rekha Dikshit for vote of thanks. That is a ceremonial part, and we have to, in Indian tradition, we have to have this opportunity, and then we will go for the valedictory. Ma'am, please.
Good afternoon, Honorable Vice Chancellor, Shobit University, Professor A.P. Garg, and Director Research, Director Research, Professor Anil Rohi, Dean College of Biotechnology, Dr. R. Kumar, and members, faculty members, students, illustrious speakers, and it is indeed a pleasure and privilege to propose the vote of thanks to all the valued guests and participants in the one-day international workshop on antimicrobial drug resistance, current scenario and future prospects. On behalf of organizing committee, I extend heartfelt gratitude to all the speakers for gracing the event and sharing with us their findings and ideas today. I'm thankful to Honorable Vice Chancellor, Sardar Vallabhbhai Patel University, Dr. R. Kumar Mittal. He always encourages and motivates us to organize such events. He guided us for the financial support from World Bank by institutional grant under the aegis of NEHEP and ICR. We were truly inspired by his great words in his presidential address today. I sincerely thank our chief guest, Dr. Parveen Malik, Animal Husbandry Commissioner, for generating awareness on need for the optimizing the use of antibiotics in the era of rising population, environmental degradation, and wildlife-human conflicts. I extend my thanks to guest of honor, Mr. Mohanji Saxena, Managing Trustee, Dabur Ayurved, for enlightening talk on resistant microbes that are threats to the humanity today, and suggested integration of environmental, agriculture, health sciences for developing long-term solutions for this huge challenge. I wish to express my gratitude to Dean College of Biotechnology, Dr. R. Kumar, Nodal Officer, ICNA Project, and Director Research, Professor Nils Rohi, and Principal Investigator of this ICNA Project, Institutional Capacity Building, leading to the accreditation of College of Biotechnology, Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut, for being the pillar of support. I thank both of them on behalf of organizing committee for guidance and contribution for organizing this international workshop. An event of this dimension was not possible without the sincere effort by members of organizing committee. I extend my sincere thanks to Dr. Amit Kumar, Dr. Jitendra, Dr. Pankaj Kumar, Dr. Shalini Gupta, Dr. Nilesh Kapoor, Dr. Naresh Pratap Singh, Dr. Pankaj Chauhan for organization of the event. I express heartfelt gratitude to Dr. Lisa Thompson for inspiring talk on OIE initiative on data collection on antimicrobial use on animals. I must mention a deep sense of appreciation for Dr. Anu Sharma, WHO consultant, for providing insights on WHO perspective on policy options for AMR containment in India. I take this opportunity to extend our most sincere thanks to Dr. Anup Kalra, Director Corporate Affairs, Dabur Ayurved, for explaining the role of herbs in AMR mitigation. I would like to extend the hearty thanks to Dr. Anu Rahal, Principal Scientist, ICR CIRG Magdum, for delivering the interesting talk on ethnopharmacological interventions to overcome antimicrobial resistance and insight. I would like to thank our distinguished speaker, Dr. B.R. Singh, Head and Principal Scientist, IVRI, for highlighting serious state and causes due to the emergence of resistant strains and unusual reasons behind that. Extended thanks are to Dr. Ajit Singh, Scientist Emeritus, UAS Hisar, for excellent presentations on resistome-informed combating of antibiotic resistance in the One Health frame. This omic branch of science comprises entire number of antibiotic resistant genes to determine the AR genes in the metagenome obtained by sequencing through next generation sequencing. I am thankful to Dr. Mumtaz Saxena, Professor Animal Biotechnology, GB Pant University Agriculture and Technology, Pantnagar, for making seminar interesting and meaningful by wonderful talk on molecular basis for development of multiple drug resistance. I'm thankful to all the faculty members, students for their enthusiasm and participation during this conference. And last and not the least, I'm thankful from bottom of my heart to the media person for extensive coverage of the event. Thank to all of them. If I have forgotten any name, it is not intentional, and I'm sorry for if I have already have forgotten. And I'm seeing Dr. Harshit Verma, Dr. Akshay. Their contribution is not less, and they have yes, sir. Okay, I consider the live streaming trainer. It is also, we are thankful to you for the extensive coverage of the event and all the students, Mrinal, Malyaj, and all the PG students who were a foundation for the successful organization of this event. Thanks to all of you.
हाँ लाइट भी है
आपने बोलियों को सबको मोमेंटम दे रिमेम्बरेंस के वेरी जरूर